ಸದ್ಯಕ್ಕೆ ಆ ಕಡೆ ತಿಳಿಸಿ ಗೊತ್ತಾಯ್ತು ತಿರ್ಗಿಸ್ಲಾ
ಎಲ್ಲರೂ ಇನ್ಚಾರ್ಜ್ ಆಗಿ ಅಲ್ಲ ಯೂಟ್ಯೂಬ್ಗೆ ಡೈರೆಕ್ಟ್ ಆಗಿ ಸರ್
starting present ಆಗು ಇನ್ವಿಟೇಷನ್ ಫೋಟೋ ಬರ್ತಲ್ಲ ಈಚೆ ಬಾ ಬಂತಲ್ಲ ಇಲ್ಲ ನೆಟ್ವರ್ಕ್ ಸ್ಲೋ ಇದೆ ಅನ್ಸುತ್ತೆ ಯಾಕಂದ್ರೆ ನಾನು ಅಲ್ಲಿದ್ದೆ ಈ ಅಲ್ಲಿ ಬಿಟ್ಬಿಟ್ಟಿದ್ದಾರೆ ಓಕೆ
ಕ್ಲೋಸ್ ಮಾಡೋದಿಲ್ಲ ಏನು ಮಾಡಿ ಕೆಳಗಡೆ ಇದಿರುತ್ತಲ್ಲ ಇದೆಲ್ಲ 3 ರನ್ ಆಗ್ತಿದೆ ಅದನ್ನ ಯಾವ್ದನ್ನು ಕ್ಲೋಸ್ ಮಾಡಬೇಡಿ ಲೈವ್ ಹೋಗ್ತಾ ಇದೆ
ಲೈಟ್ ಚೇಂಜಸ್ ಮಾಡ್ಬಿಡಿ
ಲೈಟ್ ಚೇಂಜಸ್
ಏನು ಬೇಡ
ಆಲ್ಟ್ ಟ್ಯಾಬ್ ಮಾಡಿಬಿಡು ಆಲ್ಟ್ ಟ್ಯಾಬ್ ಮಾಡಿಬಿಡು ಅದು ಅಲ್ಲಿ ಬರುತ್ತೆ ಅಷ್ಟೇ
ಬಟನ್
ಕ್ಯಾಮ್ರಾದಲ್ಲ
ಆರೆಂಜ್ ಹಾಕಿದ್ದೀರಾ
ಅಯ್ಯೋ ಬಿಡ್ಲಿ ಬಿಡು
ಬಂದೆ
ನಮ್ಮ ಜೀವನ ನಮ್ದು ಹಂಗೆ ಮೂಲೆ ಬಿದ್ದೋಗ್ಬಿಟ್ಟಿದೆ
ಬ್ಯಾಗಲ್ಲಿ
ನಾನು ಅಲ್ಲಿ ತಗೊಂಡೋಗಿದ್ದೆ ತಲೆ ಬಚ್ಚಿಸ್ಕೊಳಕ್ಕೆ
ಯಾರು ಬರ್ಸ್ಕೊತಾರದು ನನಗೆ ಗೊತ್ತಿಲ್ಲ
ಐವಾಸ್ ಬಂದರು ಹೋಗ್ಬಿಟ್ಟು ಸಡಾಗ
ಕನೆಕ್ಟ್ ಮಾಡ್ಬೋದಾ ಡೈರೆಕ್ಟ್ ಆಗಿ ಈಗ ಇಲ್ಲ ಆಫ್ ಮಾಡಬೇಕು ಡೈರೆಕ್ಟ್ ಆಗಿ ಕನೆಕ್ಟ್ ಮಾಡ್ಲಾ ಓಕೆ ಅದಕ್ಕೆ ಕೇಳಿದೆ ಕನೆಕ್ಟ್ ಮಾಡ್ಬೋದು ನಂಗೆ ಅದಕ್ಕೆ ಕೇಳಿದ್ರೆ ಅದೇ ರೆಡ್ ಕಲರ್ ರೆಡ್ ಕಲರ್ ಎರಡು ರೆಡ್ ಇದೆ ಟೂ ಮಾತ್ರ ಗ್ರೀನ್ ಅಲ್ಲಿ ಇದೆ 8 9 ರೆಡ್ ಇದೆ ಓಕೆ
ಅಲ್ಲಿ ಮಧ್ಯಾಹ್ನ ಸ್ಟಾರ್ಟ್ ಆಗಿದ್ದಾರೆ ಊರಲ್ಲೂ
ಸರ್ ಇದು ಇನ್ನೊಂದು ಆನ್ಲೈನಲ್ಲಿ ಬರ್ತಾರಲ್ಲ ರಿಸೋರ್ಸ್ ಪರ್ಸನ್ ಮಧ್ಯಾಹ್ನ ಬ್ರೇಕ್ ಇದೆಯಾ ಟೀ ಬ್ರೇಕ್ ಬರ ಅವ್ರು ಸೆಷನ್ ಸ್ಟಾರ್ಟ್ ಆಗಿದೆ
ಇಲ್ಲ ಸರ್ ಅದು
ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ಒಂದು 2 ನಿಮಿಷ ಟೈಮ್ ಬೇಕಾಗುತ್ತೆ ಯಾಕಂದ್ರೆ ನಾನು ಸ್ಟಾರ್ಟಿಂಗ್ ಇದನ್ನ ಲೈವ್ ಕೊಟ್ಟಿರ್ತೀನಿ ಪುನಃ ನಾನು ವಿಡಿಯೋ ಕ್ಯಾಪ್ಚರ್ ಮಾಡ್ಕೋಬೇಕು ಅವ್ರು ಗೂಗಲ್ ಮೀಟ್ ಲಿಂಕ್ ಆಗಿ ನಾನು ಜಾಯ್ನ್ ಆಗಿ ಅವರು ಜಾಯ್ನ್ ಆಗ್ತಾರೆ ಸರ್ ರಿಸೋರ್ಸ್ ಪರ್ಸನ್. ಅದಾದ್ಮೇಲೆ ನಾನು ಪುನಃ ಇನ್ನೊಂದು ಕ್ಯಾಪ್ಚರ್ ಮಾಡಿ ಪ್ಲೇ ಮಾಡ್ತೀನಿ. ಅದಕ್ಕೆ ಒಂದು 5 ಮಿನಿಟ್ಸ್ ನನಗೆ ಮ್ಯಾಕ್ಸಿಮಮ್ ಬೇಕಾಗುತ್ತೆ.
ಸರ್ ಇದು ಸೆಮಿನಾರ್ ಹಾಲಲ್ಲಿ ವೈಫೈ ಇದೆ ಅಲ್ವಾ ಅಲ್ಲಿ?
ಲ್ಯಾನ್ ಕನೆಕ್ಷನ್ ಇದೆ ಸಿಸ್ಟಮ್ ತಾನೇ ಡೈರೆಕ್ಟ್.
ಹೌದು.
ಸಿಸ್ಟಮ್ ಅಲ್ಲಿ
5 ಜನ ಏನೋ ಓರಲ್ಲೂ ಆನ್ಲೈನ್ ಮಾಡ್ತಾರೆ ಅವ್ರನ್ನ ಕೇಳಕ್ಕೆ.
ಸಿಸ್ಟಮ್ ಅಲ್ಲಿ ಜಾಯ್ನ್ ಆಗ್ತಾರೆ.
ಇದೆ ಇವ್ರು ಕೇಳಿ ಅಕ್ಷಯ್ಗೆ ಕೇಳ್ತೀನಿ. ಅಕ್ಷಯ್ ಅಲ್ಲಿ ರಿಮೋಟ್ ಸ್ಕ್ರೀನ್ ಇದು ಮಾಡಕ್ಕೆ ಚೇಂಜ್ ಮಾಡಕ್ಕೆ ಅಲ್ಲ ನಾನು ಹೇಳ್ತಾ ಇದ್ದೀನಿ ಇದು ರಿಮೋಟ್ ಇದಕ್ಕೆ ಸ್ಕ್ರೀನ್ ಚೇಂಜ್ ಮಾಡಕ್ಕೆ ಸ್ಲೈಡರ್ ಆಮೇಲೆ ಇದು ಚೆಕ್ ಮಾಡಿ ಸರ್ ನಾನು ಬಂದೆ ಆನ್ಲೈನ್ ಆಫ್ ಮಾಡಿದೆ ಈಗ ಲ್ಯಾಪ್ಟಾಪ್ ಅದೇ ಈಗ ನಾನು ಆನ್ ಮಾಡಿ ವಾಲ್ಯೂಮ್ ಕೊಟ್ಟಿದ್ದೀನಿ.
ಅವಾಗ ನಾನು ಏನಾಗುತ್ತೆ ಗೊತ್ತಾ
ಅವ್ರು ಒತ್ತರ ನೀ ಚೇಂಜ್ ಮಾಡ್ತಿರೋ ಅಷ್ಟೇ.
ನೋಡೋಣ
ಆಲ್ ಟ್ಯಾಬ್ ಮಾಡು, ಆಲ್ ಟ್ಯಾಬ್. ಯಾ not this. I know.
ಏ ಡೋಂಟ್ ಚೇಂಜ್ ಎನಿಥಿಂಗ್ ದೇರ್ ಇದು ಇದಕ್ಕೆ ಬೈದಿಲ್ಲ ತೆಗಿಬಿಡ ತೆಗಿಬಿಡ ತೆಗಿಬಿಡ
ಇರು 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 ನೋಡಕ್ಕೆ ಇದು ಫಸ್ಟ್ ಪೇಜ್ ಅಲ್ಲಿ ಇಟ್ಟಿರು. Bro, disconnect this, disconnect the
wait bro wait wait
ಅದು
wait
ಹೊಟ್ಟೋಗ್
ಇದು ಬಿಟ್ರೆ
wait ಲೋಡೇ ಆಗದೆ ಇದ್ರೆ oh my god the internet is very shitty bro. Wait bro, ಅಯ್ಯೋ ಅಮ್ಮ ನೀನ್ ಮಾತಾಡ್ತಿದ್ದೀನಿ ಬೈತೀಯ ನಿನ್ ಬಾಯಿ ಬಿಟ್ಟು. Where is it bro? ಅಲ್ಲೇ ಇತ್ತು.
ನೆಕ್ಸ್ಟ್ ಸ್ಲೈಡ್ಗೆ ಬರಲಿ ಅದು.
You be with me. I'll
For
be
that piece over here. ಇಲ್ಲ ನಾನು ಕೈ ಕಾಲು ಆಡಲ್ಲ.
ಅಲ್ಲ you give this to me separate ಮಾಡ್ಕೊಡ್ರಿ ಸರ್. You give this I will wait ಇದಕ್ಕೆ ಏನೋ ಕಟ್ಟಬೇಕಂತೆ. ಕಟ್ಟಿಗೆ ಬಂದು
ಅದೇ ಇರು ಅನಿಶಾನ ಹತ್ರ ಇತ್ತು.
ಅದನ್ನೇ ಬೇಕಾದ್ರೆ ಹಾಕ್ಕೊಡಪ್ಪ ಮೇಲ್ಗಡೆ ಇದೆಯಲ್ಲ
ಅಯ್ಯೋ ಈ ಕಡೆ ಇದೆಯಾ? ಇಲ್ಲೊಂದಿದೆ.
ಕಟ್ ಆಗಿದೆ
ಅದಕ್ಕೆ ಕೇಳಿದೆ ನಿಮ್ಗೆ ಪಿಪಿಟಿ ಹಾಕಿದ್ರ ಅಂತ. ಅಲ್ಲ ಇದು ಕಮ್ಮಿ ಮಾಡಲಾ?
ಸ್ವಲ್ಪ ಕಮ್ಮಿ ಮಾಡಿ.
ಸಲ ಚೇಂಜ್ ಮಾಡ್ತೀಯಾ?
ಯಾವ್ದು?
ನೆಕ್ಸ್ಟ್ ಸ್ಲೈಡ್.
ಇಲ್ಲೆಲ್ಲ ಇಟ್ಕೊಂಡು ಇದೇ ಚೇಂಜ್ ಮಾಡ್ತೀರಾ ನೀವು?
ನಾನು ಇಲ್ಲೇ ಬೇಕಂತ ಇಲ್ಲೇ ಚೇಂಜ್ ಮಾಡ್ತೀನಿ. ಇದಕ್ಕೆ ಈ ಸೊಬೈಲ್ ಈ ಸೊಬೈಲಲ್ಲಿ ಇದರಲ್ಲೇ ಮಾಡ್ಬೋದು.
ಇದೆಲ್ಲ ಮಾಡ್ಬೋದು ನೀವು.
ಅದ್ರಲ್ಲಿ ಮಾಡ್ಬೋದಾ? ಅದಕ್ಕೆ ಒಂದು ರಿಬ್ಬನ್ ಬೇಕಲ್ಲ.
ಸಾರಿ. ಏನಾಯ್ತು?
ಒಂದು ರಿಬ್ಬನ್ ಬೇಕು. ಒಂದು ಅರ್ಜೆಂಟ್ ರಿಬ್ಬನ್ ಬೇಕು.
ಅಲ್ಲೇ ಅವೈಲೇಬಲ್ ಇತ್ತಲ್ಲ ಕರೀತಾನೆ. ನಿಮ್ದೆಲ್ಲ ಏನಿದೆ ಈಗ ಇದೆಲ್ಲ ಇನ್ವೊಕೇಶನಾ
ಇಲ್ಲೇ ಇರ್ತೀನಿ
ಓಕೆ ಓಕೆ
ಇಟ್ಕೊಂಡಿರೋ
Om Shri Manjunathaya Namaha. A very good morning to distinguished dignitaries, all of the dais, eminent academicians, research scholars, delegates, and my dear friends. It is with immense pride and profound honor that I welcome you all to the inaugural ceremony of the National Conference on Recent Research and Innovations in Life Science, Forums 2026, organized by the Department of Life Science in association with SDM, SDM University, Harvard, and University Cultural Hub under the aegis of the NUSA 2.0 grants and Cultivation Initiative. Today, we gather not merely for a conference, but for a confluence of intellect, innovation, and inspiration, where research meets responsibility and knowledge meets nation building. As Helen Keller once said, alone we can do so little, together we can do so much. May the rain mark the beginning of many meaningful collaborations. To invoke divine blessings and formally commence this auspicious occasion, I now request the invocation team to render the prayer.
Sir, ಇದು slide ಹಾಕ್ತೀರಾ? ಹೌದು, document.
To extend a warm welcome to the celebrant for today's ceremony, I invite Srimati Asina Sami MD, Head and Assistant Professor, Department of Microbiology, to deliver the welcome address.
ಗೊತ್ತಿಲ್ವಾ?
ಸರ್ ಇದು ಯೂಟ್ಯೂಬ್ ಲೈವ್ ಇದೆ ಸರ್, ಅದಕ್ಕೆ ಅದು ಸ್ವಲ್ಪ
Very warm and
ಬೇರೆ ಅದಕ್ಕೆ ಹಾಕ್ಬೋದಾ?
ಹಾಂ ಸರ್, ಟ್ರೈ ಮಾಡ್ತೀನಿ.
It gives me immense pleasure and honor to welcome you all to the National Conference on Recent Research and Innovations in Life Science 2026,
Slide
organized by the
ಯಾರು ನೀವೇ
Department of
ಮಾಡಿ.
Life Science, NLA and SDM LLP, in collaboration with SDM Research Institute for Biomedical Science, SDM University Dhaka, and University
60
Career
years
Hub,
of
University of
60
Mysore.
years of this.
On behalf of our management, principals, organizing committee, faculty members, and students, I extend a heartfelt welcome to
ನನ್ನ
our
ಟಾಕ್ ಇದೆ
esteemed
ನೆಕ್ಸ್ಟ್ ಅದಕ್ಕೆ
leadership on the dais
ಗೊತ್ತಾಗಿದೆ, ನಾನು ಮತ್ತೆ ಕೆಳಗೆ ಬರ್ತೀನಿ.
So firstly, I would like to welcome our respected principal, Professor Bharati, for her
Guesting.
contribution in organizing this
ಇದನ್ನ ತೆಗ್ದು ಹಾಕಿದ್ರೆ ಆಯ್ತು.
So I welcome you, ma'am. I request Mr. Prashant, Head, Assistant Professor, Department of Commerce, to welcome ma'am to
ಒಂದು ನಿಮಿಷ ಮೇಲೆ ಬರೋಕ್ಕಾಗುತ್ತೆ ಸರ್?
It is a privilege to welcome our
ಒಂದು ನಿಮಿಷ ಸರ್, ಒಬ್ಬರಿಗೆ PPT ಹಾಕಬೇಕು.
and eminent scientists
ಪೆನ್ ಡ್ರೈವ್ ಕೊಟ್ಟಿದ್ದಾರೆ, ಅದನ್ನು ಕನೆಕ್ಟ್ ಮಾಡಬೇಕಿತ್ತು.
contribution the field of life science continue to
ಈಗ
inspire
ಮಧ್ಯದಲ್ಲಿ ಹಾಕಬೇಕು ಸರ್.
the regular results. I request our principal, Professor N. Bharati, to
ಹೌದಾ? ಇಲ್ಲ ಸರ್, ಇದೊಂದು ಸ್ಪೀಚ್ ಎಕ್ಸ್ಟೆಂಡ್ ಮಾಡಿ.
I extend a warm welcome to Dr. K. Sasimurthy, Professor and Director of Research, SDM University, Harvard, for graciously taking us about the conference
ಹೇಗೆ ಮಾಡೋದು
and
ಸರ್?
guiding us
ನಾವು
through
ಪ್ರೊವೈಡ್
the
ಪ್ರಜೆಂಟ್ ಸರ್ವಿಸ್
I request to please convey on this conference the recruiting work. I request Shri Shivriyama Sir, Head and Assistant Professor of Management, to welcome. I extend a heartfelt welcome to our advisory member, that is Dr. Ramesh Sir, from Mahajana PG Center, who is the advisory member, who has worked sincerely and who has guided us for the conference, and also for your support, guidance, and dedicated efforts. So I welcome you, sir. I request Srimati Rajameshwari, our Assistant Professor, Department of Microbiology, to welcome you. A hearty welcome to our IPC coordinator, Srimati K. Sukhuta, Associate Professor and Head Department of Computer Science, who always support us towards quality enhancement. I welcome you, ma'am. I request Dr. Shisham Sir, Head Assistant Professor, Department of Environmental Science, to welcome us.
ಈ ಕಡೆ ಇದೆ ಕಾರ್ನರ್
I also welcome Dr. Shweta, Assistant Professor and Head Department of Biotechnology. Happiness of responding us to efforts are appreciated. I welcome you, ma'am. I request Mr. Nilan MR, Assistant Professor of Management, to welcome ma'am. I extend my sincere welcome to all distinguished speakers who are present here. Dr. Srinivas D.M., Scientist, the Assistant Professor, ICMR, Bangalore. I welcome you, sir. I request Meghana, Dr. Meghana, to welcome sir slowly. I also welcome Dr. Jagadisha Koya, Assistant Professor, SDU Research Institute for Biomedical Sciences. I welcome you, sir. I request Mr. Syed, sir, Assistant Professor of Management, to welcome sir. A warm welcome to Dr. Ajay Kumar Oli, Associate Professor, SDU Research Institute for Biomedical Sciences. We welcome you, sir. I request Mr. Pandashri, Assistant Professor of Biochemistry, to welcome sir.
Laptop LED.
I also welcome one of the journalists, Dr. Subramanian, to the conference. I welcome you, sir. I request Mr. Abhishek, Assistant Professor of Biotechnology, to welcome sir. I also extend my sincere welcome to all the session chairs, research scholars, faculty members, and students, participants, and all media persons and sponsors who are gathered here from the welcome you all. I hope that today's dedication will enlighten us, inspire us, open new avenues for research and innovation. I once again welcome you all to this event. Thank you, ma'am, for your gracious words of welcome, beautifully laying the foundation of this national academic activity. I would like to take this opportunity to welcome ma'am as well. Welcome to you, ma'am. I would like to request Srimati Rajarajeshwari, Assistant Professor of the Department of Microbiology, to welcome ma'am formally. The lighting of the lamp symbolizes the triumph of knowledge over ignorance and enlightenment over certainty. May I now request our dignitaries on the dais to kindly come forward for the ceremonial lighting of the lamp.
ನೀವೇ ಹಾಕ್ತೀರಾ
ತೆಗಿಬೇಕಲ್ಲ ಅಲ್ಲಿ ಆಡಿಯೋದಲ್ಲಿ
ಕನೆಕ್ಟ್ ಇಲ್ಲ ನಂದೇ ಕನೆಕ್ಟ್ ಮಾಡಿದ್ದೀನಿ ಪುನಃ ಆಫ್ ಮಾಡಿ ಯಾವ್ದು ಕರೆಕ್ಟ್ ಆಗ್ತದೆ ಅದೇ ಕರೆಕ್ಟ್ I want your help. ವೈಫೈ ಕನೆಕ್ಟ್ ಆಗ್ತಿಲ್ಲ ನೋಡಿ ವಾಟ್ಸಪ್ ಓಪನ್ ಮಾಡಬೇಕು ಲಿಂಕ್ ಕಳಿಸಿದ್ದೀನಿ ವರ್ಲ್ಡ್ ಸ್ಕ್ರೀನ್ ಮಾಡ್ಕೊಳ್ಳೋಣ ಇದು ಹಾಕಬೇಕು ಇವಾಗ ಕೀಪ್ಸ್ಗೆ ಮತ್ತು ಗೋವಿಂದ ಪಿಟಿ ಹಾಕಿ ಬೇಡ ಇದರಲ್ಲೇ
As the Savior concludes, may you lead our minds to scientific temples and to responsibility and obey the law. Provide us with an overview of the concepts of ethics and now invite Dr. Lisa Murthy, Professor and Research Director, Indian University, Mumbai, and Chief Creative this conference, please provide a comment.
ಪಿಟಿ ಇಲ್ಲ ಇದು ಓಪನ್ ಆಯ್ತಾ
Organizing committee members, Professor Ramesh, esteemed speakers, and my dear participants. Maybe about a year ago, Dr. Shankar came up with the proposal, idea to be able to hold a conference of national and international status at his college. We were still debating on the theme of the conference, but then it took shape in the form of being able to assimilate different departments this college seamlessly so that it would encompass various aspects and topics which covers the area of life sciences. It was a significant effort to be able to identify the themes that are relevant to the participants, especially for the young student community. And towards this, organizers have chosen significant personalities in their respective areas to be able to come today and address you on the various topics of life sciences. In fact, we are very fortunate to have the Doyen of Indian Science, Professor G. Padmanabhan, Bhushan Awardee, who has shaped the biotechnology research and translation in India, and who is here with us today as our chief guest. We are very much fortunate to have him with us today. I hope that the participants will learn new concepts, new ideas, will learn new ways of doing research, will make friends, new friends, will also strike collaborations to be able to take forward their research, and hope that they will return home richer by knowledge of the proceedings of this conference. Thank you very much, and we welcome you once again to this conference.
Thank you, sir, for outlining the vision and academic framework of this conference so comprehensively. It is now time for the release of the conference key souvenir, which reflects the quality contributions and research insights of this gathering. When requested to release on the dais to kindly release key souvenir.
ಎಷ್ಟು ಸ್ಲೋ
Hearty congratulations to all the contributors and the editorial team for this commendable academic effort. It is now my privilege to invite Dr. Shweta S., Head and Assistant Professor, Department of Biotechnology, to formally introduce our esteemed inaugurator and keynote speaker.
Shri Vachanataya Namaha. Good morning, all. It's my immense privilege to introduce Harvard scientist Dr. Govindaraj Padmanabhan. His work has influenced global efforts against infectious diseases and elevated India's scientific landscape. Dr. Govindaraj Padmanabhan, born 20th March 1938, is an Indian biochemist and biotechnologist renowned for his pioneering contributions to molecular biology, particularly in eukaryotic gene expression, T biosynthesis, and malaria parasite biology. He earned his PhD
60 plus years, just a slide.
ಆಯ್ತಲ್ಲ and rose through its ranks to become
They have done
director
it very
from
well.
1994
Just
to
making sure.
1998.
Can you copy and give me the pen drive?
Yes, it's industry collaborations and advanced biotechnology infrastructure in India. Dr. G. Padmanabhan is the first student of the Indian Institute of Science, Bangalore, to have become its director. Dr. Padmanabhan's research has illuminated key mechanisms in malaria, such as parasite-independent heme synthesis pathway, positioning it as a potential drug target and demonstrating the anti-malarial protection of curcumin from turmeric as a universal adjunct drug. He has mentored over 50 PhD candidates, including a generation of students, junior and senior scientists, and entrepreneurs. He is the author of more than 180 highly cited publications and has written memoirs on his experiences in doing science in India 2008 and 2018. He has played a pivotal role in introducing recombinant DNA technology to India and contributed to vaccine development, including hepatitis B and AIDS. He has projected India to become a global leader in vaccines and has published an article, India-born weapons against infectious diseases in Nature 2003, and his profile was covered and published in peer-reviewed medical journal Nature Medicine in 2003. He was co-chair of the COVID Vaccine Evaluation Committee of DBT. He was a member of Vision Group in Science and Technology of the Government of Karnataka and has chaired several technical committees for government SMT establishments. His lecture in science in 1998, explaining the background behind India's post nuclear test, was widely appreciated. He was elected He was elected President of the National Academy of Sciences India in 2019. He has been the Chancellor of Central University of Tamil Nadu since 2018. He served as a Senior Science Innovation Advisor of the Biotechnology Research Assistance Council Gujarat of the DBT Government of India since its inception in 2012. His international engagements include visiting scientist desks at University of Chicago, USA, for over a decade, writing summary chiefs in 1986, and Sun Series Hospital Medical School London, enhancing his expertise in gene regulation and parasitology. He has served in international organizations such as World Health Organization, International Center for Genetic Engineering and Biotechnology, and an expert member. He was awarded the UNESCO Chair in Biotechnology in 2000. Among his numerous accolades, Dr. Padmanabhan received the Shanti Padmanabhan Prize in 1983 for biological sciences, the Padma Shri in 1991, and the Padma Bhushan in 2004 for contributions to science and technology. In 2024, he was awarded the inaugural Vijnan Ratna Award, India's highest science honor, recognizing exceptional achievements in early research and biotechnology. Above all, Dr. Padmanabhan is a seamless, spiritualistic, disciplined, and science-dedicated person, lover of Carnatic music, and a very good singer himself. It's truly important to have you among us. Thank you. Now, I sincerely request our beloved principal, Mr. Bharati N, to felicitate and honor Dr. G. Padmanabhan.
ಶಿಫ್ಟ್ ಮಾಡ್ರು ಇದಾ ಚೇಂಜ್ ಮಾಡಬೇಕು ಹಾಂ ಇದನ್ನ ಹಾಕಿರ್ತೀನಿ ಒಂದು ನಿಮಿಷ ಮುಂದೆನೇ ಹೋಗ್ತು ಓ ಸಾರಿ ಛೇ ಕೀನೋಟ್
Thank you, Anu, for that insightful introduction. Ladies and gentlemen, it is a rare honor and a full immense pride for our institution to host the Visionary Scientist of Global Review. Father Bhushan and Padmashri awardee for Mr. Govindarajan Padmanabhan, former director of Indian Institute of Science, Bengaluru. With deep respect and admiration, I now invite her to speak a few words.
ಸರಿ ಮಾತಾಡಾದ್ಮೇಲೆ
ಅದಕ್ಕೆ ಕೇಳಿದ್ದ
ಮಾತಾಡ್ತಿದ್ದಾನೆ ಅನ್ಸುತ್ತೆ ಒಂದು ನಿಮಿಷ ದೇವರ ಇದಕ್ಕೆ ಹಾಕಿರಿ ಅದಕ್ಕೆ ನಾನು ಈಗ ಇದ್ರ ಕೆಳಗೆ ಹಾಕಿ
innovation in your lifetime. And that became to what we have.
Then what? Just speaking of that photo.
But in terms of the operation, I should tell you, the period when they thought chemistry would be predominant, physics would be predominant, and we have gone through an age of life sciences being the dominant area of investigation. Why? Ultimately, it's the health and productive agriculture, sustain life. Therefore, all innovations happen. I can give a talk separate on another occasion. Today, we are talking about precision health. We can sequence genomes. When I was a student of faculty, they would say it will take 50 years to sequence the human genome. So we already have a complete sequence of human genome. Any genome sequence will be done in a week these days. The technology has moved so much. Sequencing has become a child's play. What is the use of this sequencing? And that is where you find we are all 3 times into the 9 base pair in our genome. 3 billion base pairs. And we are all identical. So far as sequence is concerned. Except 1 in 1,000 bases is different. And does it make us so different? The genome so different? These are all exciting questions still not answered. Our genome is very close to chimpanzee. There's only 1.8% difference. So how the genome is influencing expression of cellular level, then proteins, and we know proteins are the ones that are the energy run the whole system of the body. And that's why we are talking today about precision technology, precision health. Genetic disorders are a hope. We used to say genetic disorder is a product of karma. But today we have hope it can be cured. It can be mitigated at least. It cannot be. Best changes can happen. And maybe I have a slide on this, genome editing has become a major tool today. And there are also been ethical issues maybe very time. So I only want to appeal to students to take to science. Unfortunately, slowly, interest in science is disappeared in this country. People want to do economics, commerce, making money is okay. But ultimately, you want health, you want productivity. There's all the genome of today, right genome, wheat genome, cereal genome, all are available. So all this knowledge must be used to improve productivity, improve the gain quality, improve nutrition, health. At the same time, as I said, individuals who have genetic disorders can they be cured? Is it possible? It didn't look possible now, today, it looks very much possible. We always speak of the 5,000 genetic disorders, and all infectious diseases also. Susceptibility to infection also is genome. All of us are exposed to malaria person. All of us are written by hospital. But we don't get everybody doesn't get. Only 1 to 2% of the population has even first. But we are all exposed to manufacturing. What is it that besides somebody is resistant, somebody is sensitive? It was an exciting opportunity. I mean, I can go on and on, it's an exciting subject. But I just want the students not only to take science and take to life sciences, tremendous opportunity for you to solve global issues, whether it is health, whether it is agriculture, whether it is environment. These are the 3 major areas you have to construct and have great off. All the best. God bless.
Thank you, sir, for that intellectually interesting and deeply inspiring address. At this juncture, I invite our respected Committee President and Principal, Professor M. Bharati, to deliver the presidential address.
Om Shri Manjunataya Namaha. Narrator of today's conference, Professor Govindarajan Padmanabhan, Chief Convener, Professor Satyamurthy, and Convener, Professor Ramesh, Srimati Adhiya, Head of the Department of Microbiology, Dr. Shweta, Head of the Department of Biotechnology, Srimati K.S. Sukruta, IQAC Coordinator and Head of the Department of Computer Science, other dignitaries of the dais, my dear colleagues and students. Today, I would like to begin saying that I am not from life science background, but I am from the background of physics. But it doesn't matter because in today's world, knowledge does not grow in separate compartments. All subjects are interconnected with each other, and progress happens only when we work together. That's why interdisciplinary, multidisciplinary subjects are of greater importance nowadays, where knowledge acquired in one subject is applied to the another to deepen your level of understanding and extending horizon of learning. For example, in MRI and scanning, CT scan, they all work on the principles of physics, but it serves as a powerful tool in research work associated with life science. Similarly, microscope, lasers, radioisotopes, they were all invented by physicists, but microscope, it has extensively, it is used to examine the structure of DNA, structure of cells, and lasers and radioisotopes, they are all used extensively in medical field, both for diagnosis as well as treatment. So that's all that physics plays a very important role or supports life science at the smallest level. And coming to microorganisms, my knowledge about microorganisms is because I told you I am from physics background. There we deal with microparticles like the pen, proton, soil the pen, proton, electron, neutron, and quantum mechanics deals with very more fundamental and elementary particles. Coming to your microorganisms, virus, bacteria. So whenever I say something about virus, coronavirus relates to the surface, because no one forgets the hazard being undergone. So all those lockdowns, travel restrictions, overcrowded hospitals, no one forgets that how it affected our normal life, and so many of them lost their near and dear ones, and how we struggled to bring our life back onto the tracks. But there are also good fibers. That's what Professor Ramesh sir was explaining yesterday. They help in digestion, they help to develop immune system, and many more. Anyhow, lot of inventions and research in life science has led to the advancement in medical field, as Professor Padmanabhan said. And nowadays, gene therapy, all of us used to study in newspapers, gene therapy where DNA is altered to treat diseases, and also it is extensively used in agriculture to improve the crop yield. And lot of debate is nowadays going on about the future of the world. One thing is very certain. I'm sure about it that in the near future we are going to face shortage of food supply, shortage of food. Maybe because climate change or growth of population, increase of population. Here where biotechnology comes into picture, it enhances agricultural production at 4 different levels: quantity increase, improve the quality and production of growth prospects, and also pest resistant. Here also to the plants also, gene therapy is given to become tough to develop more resistance, because nowadays pests destroys a lot of crops, and so it becomes more pest resistant. And remember this, in many, even in medical field, okay. Medical field is life science plays a very important role, but people think that medicine begins with doctors. No, doctors just examine and prescribe the medicine. The real journey of a medicine, whether it is tablet, syrup, or tonic, injections, they are developed in the laboratories where biochemists work, and they develop the medicines, and a lot of experiments is done, and lot of research work is done before they could bring the medicine out to the world. Because human body, there is a lot of works, a lot of chemical reactions, the chemistry of the cell, chemistry of the human being. All these are taken into account by developing the tablet or developing medicine. First, the biochemists, they design the medicine, they experiment it on a target, experiment it for safety, experiment it for all other aspects are taken into account: purity, safety, and adverse effects, side effects of the lot of tests is done. And finally, after certification from BIS, it is released to the outside or market, whatever is there. So, without biochemists or life science, pharmaceutical company does not exist. So, the paramedical, it plays a very important role. The doctor just prescribe the medicine. Everything is developed by them. Who developed coronavirus? Biotechnology people work day and night to develop coronavirus, which we thought it was very hazardous, very 2 3 years back. So, now another important thing is about waste management. That is also very important. So how you have all studied biofuel? Why we have to go for alternate sources of energy. In biofuel, organic waste, plant waste, animal waste, everything is taken, and it is without oxygen. It is called anaerobic digestion, And methane gas, if you study it in detail, methane gas is there, which is used as a biofuel, but because it is a hazard-free, environmental friendly, doesn't release any harmful materials to the atmosphere, but energy density is low. Compared to fossil fuels, it takes millions of years to produce from nature, natural through nature. So millions of years, but this is not like that. But energy density, even in solar cell, everywhere energy density is low. If you burn this much of fossil fuel, you will get large amount of energy, but not so. Even in solar cell, from one single cell, you will get point 0.1 amperes of current and point 0.1 volts. But here, fossil fuel tremendous energy. So lot of it is emerging areas for research also to improvise. Electroplastics are used to take biofuel. So lot of research is going on in this direction also. So there are many more research areas. Okay. Once a few days back, I was seeing a gold interview with football player Pele, and they were asking him. He was awarded for hitting large number of goals, and he was asked in the interview, "How come you hit so many goals?" They asked. So for that, what he told, everyone runs behind the ball, where ball is there, but I will go where ball goes. So he predicts where it will go, and that's what the meaning. So there are a lot of other stuff. Bioinformatics is another emerging field. We have conducted one ad-hoc course on bioinformatics where you can't say initially biology people used to say I don't want mathematics. Now it is mathematics also comes. So you have to find large amount of data for that computer science is every branch computer science is required for analysis. So bioinformatics is another emerging field. So you have a lot of opportunities. You have to go where which you are passionate about, which you are interested in, and today, on behalf of our college, I express my sincere gratitude to Professor Padmanabha sir for inaugurating this conference, and your rich knowledge and inspiring words have motivated our students. And we are truly honored that you took time off from your busy schedule and made your day to be with us today. Thank you very much, sir, on my personal behalf, and thank you very much. And Satyamurthy sir from Day-1 is giving guidance how to carry out this project, and even Ramesh sir patiently he sat with us and designed how this conference should go on. From on behalf of our college and on my personal behalf, I thank you, sir. And my appreciation to all life science departments. They are from past 2 months they are working tirelessly and make this happen. Whenever we ask something, no, I have to organize some work associated with the conference. They used to tell Srimati Atiya, Raja Rajeshwari, Dr. Shweta, everybody, Dr. Sri, and Abhishek, everyone. Everyone has worked, so my appreciation to all of them, and wish the conference to be a great grand success and make today's discussions ignite your mind, ignite your passion, and propel you towards a future filled with endless possibilities. Thank you, one and all.
Thank you, ma'am, for your motivating work this leadership. Your unwavering commitment to empowering women through education continues to be a strength of this institution. Gratitude is not merely an expression, it is an acknowledgement of connected work. To propose the vote of thanks, I now invite Srimati K. Suzuka, Associate Professor and Head, Department of Computer Science, and IQAC Coordinator, to address the gathering.
Good morning to 100 all present here. Respected dignitaries on the day, distinguished speakers, members of the advisory board, organizing committee, sponsors, faculty colleagues,
researchers, dear students, and all the participants. Very pleasant good morning to one and all. It's my proud privilege to propose the vote of thanks on this memorable occasion of the inaugural ceremony of the National Conference on Recent Research and Innovations in Life Sciences 2026, organized by the Department of Life Sciences and IPAC of LFA and SDM Madhya Mahavidyalaya in association with SDM Research Institute for Biomedical Sciences, SDM University Dhaka, under the aegis of RUSA. At the outset, we express our deep sense of gratitude to our Chief Patron, Padma Vibhushana Awardee, Puja Dr. Veerendra Hegde, President, SDM Educational Society, Ujjain, whose visionary leadership and commitment to education continues to guide and inspire us. We express our heartfelt gratitude to our esteemed inaugurator and keynote speaker, Professor G. Padmanabhan, former Director, Indian Institute of Science, and also Padma Bhushan and Padma Shri awardee, for gracing this occasion and for delivering an inspiring keynote address that has set the perfect academic tone for the conference. Your profound insights into research and scientific advancement have truly motivated young researchers and students. We extend our heartfelt thanks to our president of today's program, Professor Bharati S., Principal, MFA and SDM Madhya Mahavidyalaya, for her constant encouragement, administrative support, and motivating presence, which have been instrumental in organizing this national conference. Thank you, ma'am. I am extremely thankful to our Chief Convener, Professor K. Satyamurthy, Director Research, SDM University Dhaka, for his academic guidance and for being us about the conference and its research significance. Thank you, sir. We gratefully acknowledge our distinguished advisory board member, Dr. Prakash Halami, Chief Scientist and Head, Microbiology and Fermentation Technology, CSIR, CSTRA, Mysore, on his absence, and Dr. Ramesh, CEO, Chief Scientist, Bhuja Memorial Himachal Education Center, Mysore, for their scholarly guidance and valuable support in shaping the academic framework of this conference. Thank you, sir. I also thank eminent speakers from premier institutions and industries who have agreed to share their knowledge and expertise during the conference. Your presence enriches the academic value of this gathering. We gratefully acknowledge the efforts of our respected session chairs and duties of poster presentation who are going to steer the sessions with scholarly insight and thoughtful moderation. I also extend sincere appreciation to all the conveners, Srimati Adya Samin MP, HOD of Microbiology, Dr. Shweta S., HOD of Biotechnology, Srimati Meghna M., HOD of Biochemistry, and co-conveners, Srimati Radharajeshwari, Mr. Abhishek S., Ms. Kantashri BL, and all organizing committee members, student coordinators, Ms. Kushi I. Saudari, Ms. Maria Anisha, Ms. Shama, and all executive members whose tireless planning and teamwork since some 2 months made this program smooth and successful one. Thank you all. A special note of thanks to all our sponsors and supporting organization, industries, and institutional partners, RUSA, Rashtriya Uchchatar Shikshana Abhiyana, International Academy of Reproductive Sciences, Jeevanaya Blood Bank, Terra Vertex Innovation Hub, Microbiology Society, Rajiv Life Sciences Private Limited, Bangalore, GK Unit, a Vedic probiotic food, Karnataka Fine Chemicals, Megalo, Management Consulting from Canada, and Ms. Pooja, an alumna of this college, is working in NVIDIA Bangalore for their generous support and encouragement towards student development and research promotion. I thank the faculty members, research scholars, non-teaching staff members, and student volunteers, media persons whose dedicated efforts behind the scene ensure flawless arrangement. I also appreciate the technical team members, Sri Suresh Babu and Sri Akshay, for managing the hybrid mode of the conference and enabling wider participation. My sincere thanks to every participant who has traveled from different places and joined us to make this academic gathering meaningful. Finally, I thank each and everyone present here for their valuable time, enthusiasm, and contribution in making the inaugural ceremony a grand success. Thank you all. Have a good day. Thank you all for that heartfelt expression of gratitude. With deep respect and admiration, I now invite Professor Govindarajan to deliver the inaugural keynote address.
ಅದೇ ಕಲರ್ ಮೈಕ್ ಕೇಳಿ ಸರ್ ಕಲರ್ ಮೈಕ್ ಬೇಕಂತ ಕೇಳಿ ಕಲರ್ ಮೈಕ್ ಬೇಕಂತ ಕೇಳಿ
Thank you again for inviting me to give this talk very briefly. I would like to tell you my own journey with the science and IIT. Which I am going to IIT and trying to do something. I joined the Indian Institute of Science after a master's degree in soil science and agricultural chemistry at the Indian Agricultural Research Institute. I don't know why I joined because I was a basic chemistry student. I did my bachelor's at Residency College Madras. But then they started a PG school at that time, and with the Ratan strength support and anything abroad, coming from abroad was considered very new, and so I thought. After my master's, my guide told me, if you want to do biochemistry, you have to go to the Institute of Science in Bangalore. So I went to Bangalore. My first thesis in IIM was on trace elements in availability of trace elements to plants. Trace elements, molybdenum, copper, manganese, these are all trace elements. they are very important in agriculture. And how do you measure availability in a plant? There will be plenty of metals available, but it is available to the plant. So there was a assay where the expected is bioassay. Just one example I will just give. If you have I used to make a copper deficient medium. And then, if you have very little copper, and then you add the soil. For example, here you can see the moisture is all white, very little copper. So increase the copper, force the forward, as well as it becomes black. So that was better availability based on that. That was fine. But this is, I looked at availability of copper and molybdenum. That's better based on growth of the stuff for better sport. Then I went to physical science. They joined the biochemistry department for my PhD. And I worked with Dr. P.S. Verma. Great biochemist is particularly Verma Verma is from South Canada. Unfortunately, he died. He had gone to Switzerland to attend my conference, and by heart attack, he then died on the railway station. I joined him, and he was the one who guided me for his PhD, for my PhD. Initially, I also worked on the toxic principles in Gasolina. It is still grown, it is still available, the cheapest source that is available, but we isolated separately causing paralysis of the legs. There is no proof, but we isolated a compound, enough to the diameter property and established its neurotoxicity. But we do not know that that is one responsible for the disease in the human. But in animal model, we could produce current. Then I isolated this compound called death very clopidine. These are highly cyclic peptides. They can bind iron enormous, very high affinity. But interestingly, if you reduce the molecule, iron will be released. If you compress, then it will be released. And if you grow almost like neurasthenia, in iron deficient medium or copper toxic medium, it will secrete this molecule. Without iron, looking for iron, and if iron is available, it will be very available. So I thought this molecule can be used to treat leukemia. As you all know, because of iron toxicity in the patients, iron toxicity in children, they get leukemia. Whether the iron can be removed using this molecule. So I started with the animal model and I added this physicoprotein, and we could see that the urine was blood red because it was binding the iron in the body and secreted in the urine. So we thought it can be used for treatment of the to remove iron. But in those days, of the 60s I'm talking about, there was no industry interested in your molecule, and I didn't know how to take it forward. So left it there, like that. I'm saying this because today you have a lot of industry, lot of startups who are interested in your research, you are interested in your results, and they are prepared to take it forward. But we went through a period, we didn't know how to take it. We had interesting findings, but we didn't know how to take it forward. So, desire from this very cooperative will be given to him, This is the structure of B. The tetrapyral nucleus is protoporphyrin and iron becomes D. As you all know, hemoglobin has D. D is the prosthetic that we want. But I found D by itself has many interesting properties. Then, I became assistant professor at 1969. 73 only I went abroad. Perhaps I was the only one at that time to be given a job in the institute without postdoctoral experience outside the country. Till I went abroad after I became assistant professor. I worked with the University of Chicago. I worked with Mother Rabinovich, you see, he's in a wheelchair with muscular dystrophy. He was motivated because he was completely paralyzed, hands, legs, but still he will come to the lab 5 times a week in the wheelchair. And his wife, she's a pediatrician, she resigned her job just to take care of him. So I went to his lab in the University of Chicago. At the end of the year, I went to the celebrate party. And they said, why are you going back to India? There's nothing here in your country. I told them, no, sir, it's not true. I want to build a lab, but please help me. And they were very nice. They appreciated my stand. So it doesn't mean, India, even that time I told like that. But today India is so much developed. A lot of opportunities are there. So even if you go abroad, all the keep saying, come back, some of this is very more. When I went to this lab, when I said that, then he said, okay, I appreciate your stand, go back to the country, but come to my lab anytime if you want. So I went there, I took this offer literally, and went to his lab 10 times between 1973 and 86. So whenever I have time, 1 month, 2 months, I see those very liberal in terms of knowledge building. In summer vacation, I used to go there. And 10 times I picked up all the technology at University of Chicago. And techniques like gene cloning, sequencing, hybridization, protocols, recombinant DNA, all the technology I picked up there. So I brought those technologies. I didn't take my system. My system was cytochrome P450 liver, which is involved in drug metabolism. I was interested in cloning this gene, cytochrome P450 gene in liver. And because drugs will induce cytochrome P450. And drugs, whether it is a toxic drug or a therapeutic drug, they are all metabolized. And one of the important steps in metabolism is hydroxylation. The drug is hydroxylated, then conjugated, and then secreted in the urine. So I was interested, and when I started research, only 2 people were known. We used to call them as phenobarbital reduced, methylpolatinate reduced. But today, hundreds of P450s are known. How the science has exploded. We used to think there are only 2 species. Today we find P450s all over, in plants, animals, bacteria, everywhere. Well, I will not take you to the science part. This is the first PNAS paper I could publish from India. I have half a dozen PNAS papers, all published from India. Nothing I did, I used to bring technology from US, but work in my institute and publish these papers. How the gene is regulated. This is what it is. So I could establish that he molecule, I told you he is in the prosthetic group, but still it can regulate protein synthesis, it can regulate protein translocation, it can regulate transcription, it can regulate differentiation in hematopoietic cells. So we published this review. He is a multifunctional cellular regulator. Can control many pathways in the body system. At that time, Astra, Sweden, came to India. And I don't know why, while I was working like this, they said, please help us, we want to set up a laboratory next to Minnesota. So I had the AstraZeneca, it became AstraZeneca. Of course, they served for 25 years in Bangalore. Even today, you will see when you go on the airport road, you see the building there, but they have closed down after 25 years. Not only in India, many states, many global capitals. But anyway, after when we start, I started 4 labs, 4 projects for AstraZeneca, and one of them was on malaria. Then I start, why not I start malaria myself? And that's how I got into malaria research. And my one of my students, Samraj, broke the entire pathway of chemosynthesis in the malaria parasite. Malaria parasite, although gets a lot of heat from hemoglobin, also makes heat. It's interesting. Why does it make heat? It's getting so much hemoglobin. Parasite, as you know, when it infects the red blood cell, gets heat from hemoglobin, but also makes heat. We didn't know why it should be making heat. And the then pathway is present, we clone all these genes, we sequence all these genes. Why are they present? Then we realized, of course, after we started, there was initial skepticism, but then many other groups also started working in this area. Patton's group, Rose group, Wilson's group, Goldman's group, Akhil from Drexel University, all of them started working in this area. And the parasite, connection gene is what? This is what I hope I was able to see. Parasite has a life cycle in the animal model. In the animal, this is the asexual. And then in the mosquito, when it bites, it not only sends parasite, it also takes up the blood. And the parasite now develops with the sexual mode. So the parasite follows the sexual mode in the human blood, and sexual mode is the mosquito blood. And then they infect. Parasite infection causes malaria. It is simple malaria is curable. But when it becomes, it becomes resistant, then it becomes dangerous. It binds to the red blood cells or it binds to placenta in pregnant women. And that is why it becomes a huge problem for pregnant. Malaria is a huge risk in pregnant women. Well, I don't want to get into this. We wrote many reviews on the Western methodology. The entire heat kinetics or heat dynamics in the cell, we could figure out here that heat not only comes from hemoglobin, this is the parasite, this is the red cell. It gets heat from the parasite, from the red blood cell, it makes heat. Now, why does it make all this? Multiple pathways from hemoglobin, it gets. It gets heat also because the red cell makes heat and then it imports. Then it has a biogenic pathway. Then we are wondering why do we have so many pathways? Why heat is so important for the function? The parasite is in the sea of hemoglobin, but it makes heat. Because I told you again, after all more details of this, we showed that parasite got an organelle called food vacuum. In the food vacuum, the hemoglobin heme is converted into Amazon, because heme is a very dangerous molecule or toxic molecule. And we found that biosynthetic heme regulates this conversion of heme to Amazon. Very interesting. Just to explain it in a figurative sense, this is biosynthetic heme. Small amount is made by the person. This is hemoglobin heme which it gets done, but it's very essential that this heme is required for this heme to be formed. For this heme, hemoglobin heme to be detoxified, this heme is required because it regulates the food factor, where the conversion is taken. Therefore, I went on to see whether I can use the molecule to learn this cerebral malaria session. So, I will not get into it. Curcumin, turmeric, which some of you may know, there is no disease which has not been tested with curcumin. And curcumin has been shown from turmeric has been shown to be very effective. What you are not able to read is all these various pathways which curcumin affects, which is present in turmeric. But anyway, I will just show you the concluding slide on this research which we have done. The cerebral malaria in the animal model at least is caused by this phosphorylation, less phosphorylation involves inflammation, parasite infection. The inflammation is very essential for causing cerebral malaria and animal diet. If you cultivate, it will prevent inflammation. But still the animal will die. Animal will die of anemia. So, we found a combination of artificial and curcumin is very effective in curing cerebral malaria. So, the upshot of all this research is to show that curcumin is a very important adjunct drug along with so, before research, in a lifetime identify two therapeutic molecules with potential to prevent and treat cerebral malaria. One is curcumin, the other one is glyceoprolin. I will not get into it. I accomplished data. Both of them are inhibitors of heme synthesis in the first line, and we find that they can cure cerebral malaria. So, I was involved, apart from these two molecules, curcumin and heme, I was involved in developing hepatitis B vaccine. Professor Rangarajan was my student, and he developed this vaccine in my laboratory. And I served as chair, member of several technical committees, EBT, GSP, CSIR, ICFR, DRDO, UGC, Department of Atomic Energy. There was a period I was chairing so many committees, and that took a lot of my time, but still I felt it was my obligation about my science. So I closely interacted with the DBT, Department of Biotechnology, especially when it started National Biotechnology Board. The department came only in 1986. So I was present at that time in So I also believe, apart from all the more cloning work which we did to unravel the gene pathway, genes you can see, recombinant DNA research I could establish in this country. What I used to do, I go to Chicago, learn the techniques, come back home, and then run a workshop. I will invite all the people all over the country to come, and they will learn all the techniques and go back. And some of them became head of the department, few Few of them became vice chancellors, the Prasad University, Delhi University, so many of them. They still understand Guruji. Very interesting people there. They have. But anyway, these are makes you feel very happy. There's nothing more than that. I'm a supporter of modern technology in agriculture, genetically modified crops and plants. Not only recommend being a health is important for human, and health is important for agriculture also, for modifying the genes. And I remember criticized there were activists who will say recombinant DNA will get into the plant and then you will consume it and you will cause all kinds of problems. I used to write in current science newspapers and despite all the personal criticism I have to face. I'm saying this because in your career, even if you want to think you are doing good, you should be ready to face criticism. You may be right, you may not be right, but if you feel you are convinced your stand is right, stick to it. That's very important. Anyway, I wrote how why India needs biotechnology to ensure food and nutrition security. It was a good chapter in a book by edited by Aswat Krishnaraj. Published this book. GM food. I wrote a native biotechnology necessity for GM food in India. Now, GM, all the genetic modification had problems. Slowly, that objection has gone away because genetic modification is getting replaced by gene editing. For students, you know, I'm sure some of you know what is gene editing. Gene editing is change the gene present inside already. You don't bring any gene from outside. You bring a gene from outside and introduce it becomes genetic modification GMO. Genetically modified organism. But this is also a genetic modification. But you don't bring a gene from outside, you modify the gene which is already present. That your single base change is not possible today because of the CRISPR-Cas system. I think students should go and read up what is CRISPR-Cas. It's very, very important technology in CRISPR-Cas. I have just told you from the internet what it means. CRISPR stands for clustered, regularly interspersed, short, value-growing repeats. It's a revolutionary technology, gene editing technology, adapted to the bacterial immune system. It's like a molecular scissors. You cut the gene wherever you want. Guide RNA to directly catch protein to a target DNA location. And then you want to remove the base, it will remove. That means how do you modify a gene in a human or in an animal, but you can use this CRISPR-Cas technology. That enzyme is so specific. Only thing is you should know where to direct your gene for which you use the RNA, which is complementary to that gene. And you go there and then cut, and then that was the thing. And then earlier years there was some chair of this net level report, closed in cavities, appointed by the DGs. I worked with many of them. Directly general, but I was sharing this biotechnology industry assistance council since its inception. It supports startup companies and has played a major role. There are around 10,000 biotech companies, 10,000 biotech startups in India, and 10% of them are in Bangalore. Bangalore has got 1,500 plus startup companies in my school. Therefore, I'm saying this because you have an opportunity today to start up, to become a startup. And Bayrak will give you 50 lakhs of rupees if you have an interesting, good project. They have a committee, they will have a site visit, and they find your project good, they will give you 50 lakhs. That's how so many startup companies have come in India today. Startup companies in other areas, AI and computer science and other areas. But in Microsoft, they have 1,500. So it's a very, India has got 150,000 startups in all areas put together. So there is a lot of opportunity. So let me end. I have thanked a lot of students, 50 plus PhDs inside outside the campus, and I have interacted with a generation of scientists, politics, entrepreneurs. That's how I came in touch with Satya and Manipal and interacted with them. Whenever I get an opportunity, and the research on malaria vaccine is now continued by Dr. Arun Nagaraj at Institute of Life Sciences Foundation. So it's a great opportunity for me in a lifetime to have worked in this area, and my contribution apart from science, I think, is to enthuse startups in my technology. And keep saying that these opportunities are available to everyone. Young students, MSc students, I know, who have passed out, they said, Doctor, I am not going abroad, I am not doing my PhD, I want to become a startup. And if they have a good project, they can get supported quickly. But not easy, I can tell you. You have to be very great. But everybody great opportunity. So there are opportunities for students today, younger students. And apart from going abroad and learning, of course, all that you have to do. But you can become a startup. You don't have to work for anybody. You start up and give job to others. Thank you so much.
Thank you, sir, for that intellectually interesting and deeply inspiring address. Your words have not only enlightened us, but also reminded us of the responsibility that accompanies scientific pursuit. With this, we formally conclude the inaugural ceremony of the National Conference on Recent Research and Innovations in Life Science 2026. However, this marks only the beginning of 2 days of meaningful dialogue, research exchange, and academic engagement. All delegates and participants are requested to stay seated as further instructions regarding the technical sessions will be announced shortly. On behalf of the organizing committee, I thank each one of you for your gracious presence and participation. Let us move forward with curiosity, collaboration, commitment towards scientific excellence. Thank you and have a great day ahead. It's time to refresh and recharge. We will now take a short tea break. Tea is arranged in C314, room number C314. Kindly return to your seats in 20 minutes so we may proceed with the next segment of our conference. Thank you.
I will do that thing.
Wait. Where is Abhishek sir?
Huh?
Abhishek sir.
Abhishek sir, he went outside. Why?
I'm saving the file.
Save in one folder. That's
Sir,
one.
here this pen drive. I'm saving in this pen drive.
ಹಾ ಒಂದು ಇದರಲ್ಲೂ ಸೇವ್ ಮಾಡಿಬಿಡು.
Save as well.
ನ್ಯಾಷನಲ್ ಕಾನ್ಫರೆನ್ಸ್ ಅಂತ ಒಂದು ಫೋಲ್ಡರ್ ಮಾಡಿ ಡೆಸ್ಕ್ಟಾಪಲ್ಲಿ ಅದ್ರ ಸೇವ್ ಮಾಡಿಬಿಡು ಇರಲಿ.
Where is Isha?
She went.
You have to give this.
ಏನು ಬ್ರೋ?
ಇದನ್ನ ಇಟ್ಕೊಂಡಿರು.
I will give up.
What will you have? The lady looks like this. Let her sit there.
I know who is this.
ಏನಾದ್ರು ಸರ್ ಯಶಸ್ವಿ ಇಟ್ಕೊಂಡಿಲ್ಲ. ಅಪ್ಪನ ಅದು ಆಕ್ಸೆಪ್ಟ್
ಇದೆನಾ?
अपना
that is बता दे नहीं
कौन सी फोल्डर में डालूं? नया फोल्डर बनाओ.
ಡೆಸ್ಕ್ಟಾಪ್ ಅಲ್ಲಿ ಒಂದು ಹೋಸ್ಟ್ ಮಾಡ್ಬಿಡು ನಾಟ್ಕಾನ್ ಅಂತ ನ್ಯಾಷನಲ್ ಕಾನ್ಫರೆನ್ಸ್ ಅದಕ್ಕೆ
dollar extra. Let me be there.
ಅಲ್ಲೇ ಇಟ್ಕೊಂಡು ಹೋಗಿದ್ರೆ
नहीं, she wanted to write something. I'm just giving her.
Now I want to do something. Oh my ಚಂದ್ರಕುಮಾರ್ ಚಂದ್ರಕುಮಾರ್ MG
ಓಪನ್ ಆಗ್ತದ
ಕಾನ್ಫರೆನ್ಸ್ ಮಾಡಿದ್ರೆ ಸದ್ಯ ಬರೀ ನಾವು 3 ಜನ ಗ್ರೂಪ್ ಇದ್ದೀರಲ್ಲ ನಾನು ಖುಷಿ ಅಲ್ಲೆಲ್ಲೋ ಒಂದು ಕಡೆ ಇದೆ ಕಡೆ ಸಿಗ್ತಾ ಸರ್ಚ್ ಮಾಡ್ಬಿಡು ಅಲ್ಲೇ
You send it to yourself.
ವೇಟ್ ಗೊತ್ತಿಲ್ಲ ಎಲ್ಲಿ ಇದೆ ಅಂತ
तरफ उधर है इंट्रोडक्शन का
उनका पीपीटी आया था नहीं क्या
Only the profiles are sent in the holiday, not.
उनका पीपीटी पूछना पड़ेगा तभी नहीं कि तभी मैं अंदर डाल लेंगे
वो सर
अन्य और बर्तन व्यक्ति
Whenever I speak,
ಇನ್ನೊಂದು ಫೋನ್ ಖಾಲಿ ಇದೆ ತಕ್ಷಣ ಓಪನ್ ಮಾಡೋಣ ಅಷ್ಟೇ ಇದೆ ಹಿಂದಿಯಲ್ಲಿ Tell me again once even the conversation ಹಾಕಬಿಟ್ರೆ
I call her, nothing.
ಮಾಡ್ಕೊಂಡೇನೆ
I'll
ಬೇಕು ಮಾಮೂಲಿ ಈ ಸ್ಲೈಡ್ಸ್ ಬೇಕಾಗುತ್ತೆ ನಿನ್ ಸ್ಲೈಡ್ಸ್ ಅಲ್ಲಿ
After the session, only letter and moment for both of the moments.
You handle that, I handle this one letter. I'll go arrange there. What did you say? What did you say?
It's like
Sit in this extra sofa behind of it. Uh, 2025
ಇದಕ್ಕೆ ಹೋಗ್ಬೇಕು ಪ್ರೆಸ್ಸಿಗೆ ಅದಕ್ಕೆ
ಅದೇ 2025 26 ಇಸಿ ಟುಡೇ ಎಲ್ಲ ಹಾಕಿದ್ದೀನಿ
ಅಪ್ಪ ಯಾರಿಗೆ
ರೆಕಾರ್ಡಿಂಗ್ ಕಳಿಸ್ಲಾ ನಿಮಗೆ ರೆಕಾರ್ಡಿಂಗ್ ಫುಲ್ ಮಾಡಿದ್ದೀನಿ ಅವ್ರು ಮಾತಾಡ್ತಾರೆ ಸರ್ ನಾನು ಇವಾಗ ಕೊಟ್ಟಿರಿ ನಾನು ನಿಮ್ಗೆ ಇವತ್ತು ಫುಲ್ ಆದ್ಮೇಲೆ ನಿಮ್ಗೆ ಕೊಡ್ತೀನಿ ವಾಪಸ್
ಅಲ್ಲ ಈಗ ಓರಲ್ ಬೇಕಲ್ಲ ಅದಕ್ಕೆ
ಅದು ಮಧ್ಯಾಹ್ನ ತಾನೆ
ಶೇರ್ ಕೈಲಿ ಹಾಕಿಸ್ಬಿಟ್ಟು
ಓಕೆ 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 ಹಾಕಿಸ್ಬಿಟ್ಟು ಕೊಡಿ ಹಾಂ ವಾಪಸ್ ಬರ್ತದಾ मुझे ये टेबल जरा गाड़ी हो रहा था ना ये टेबल खाली हो जाता है ಈಗ ಇದೆಯಲ್ಲ ಅಲ್ಲೇ ಬಿದ್ದಿರುತ್ತೆ ಅವ್ರು ಏನು ಸಿಸ್ಟಮ್ ಮಾಡ್ತಾರೆ ಅಪ್ಲಿಕೇಶನ್ ಕೇಳಿ ಓ sent it to Abhishek sir. ಏನ ಬರೆದ್ಬಿಟ್ಟು ಕೊಡ್ಬೇಕಂತೆ ಏನಂತ ಆ ಚಾಟ್ ಜಿಪಿಟಿಗೆ ಅದನ್ನ ಹೀಗೆ ನಾನು ವಾಯ್ಸ್ ನಂಬರ್ ಹಾಕಬೇಕು ಚಂದ್ರಕುಮಾರ್ ತುಂಬ ದೊಡ್ಡ ತಪ್ಪು ಮತ್ತೆ 7 ಚಶ್ಮೆ ಬೇಕಂತ ಅನ್ಸುತ್ತಾ ಯಾರಿಗೆ ಬೇಕಿದ್ಯಾ
This is not required here.
Then you can save in your folder. हाँ यार न्यू फोल्डर ಮಾಡಿ ನ್ಯೂ ಫೋಲ್ಡರ್ ಅಂತಾನೆ ಇದೆ ಎಲ್ಲವೂ ಒಂದು ಎಲ್ಲ ಆದ್ಮೇಲೆ ಒಂದ್ಸತಿ ಆ ಪಾರ್ಟಿಸಿಪೆಂಟ್ಸ್ ಫಾಲೋ ಮಾಡಿ ಅಪ್ಲಿಕೇಶನ್ ಕಳಿಸ್ಬಿಟ್ಟು ಮಾಡಿ ಅದಕ್ಕೆ ಈ ಬ್ರೌಸ್ ಇದೆ ತಪ್ಪಿಟ್ಟಿದ್ಯಾ ಟೀ ಕಾಫಿ ಬೇಕಾ ನಿಮಗೆ ಇಲ್ಲಿಗೆ ನಿಮಗೆ ಬರ್ತೀನಿ ಅಲ್ಲೇ ಬನ್ನಿ ಬರ್ಕೊಂಬರಿ ನಿಮಗೆ ಎಷ್ಟು ಕಾಫಿ ಇದು ಯಾಕೆ ಬೇಡ ಖಾಲಿ ಆಗುತ್ತೆ ಬೇಡ ಅಂತೆ ಅವರಿಗೆ ಅವರೇ ಮಾಡ್ಕೊಂಡ್ಬಂದಿದಾರಂತೆ ಟೇಕ್ ಮೈ ಚಾರ್ಜರ್ once gone is always gone but ನಾವು ಅಪ್ಲಿಕೇಶನ್ ಇಟ್ಕೊಂತೀವಿ ಅಷ್ಟೇ ಇದಕ್ಕೆ ಇದು ನೋಡ್ಕೊಂತಾರಪ್ಪ
लंच का बोले रेडी है वो नीना बोला नहीं है तेरे पास नीना हाँ मैं बोल देती हूँ
You have to only say
I don't know at which places they are kept. तू ही बोल दे, no problem.
2 सेशंस के बाद आएगा, एक दो
Hmm.
बोल.
No, like the number at where the lunch is being kept. That
Jesus, and the digital stuff. उनको भी देना है क्या? Because she's a co-chair.
She's the coach, she has to be there. What you have to? She's talking. She's talking with the chair. All of this. And they're glad. They're glad to have Dr. Shitti's They're just happy that we have joined conference and we're happy that we are healing her.
He will he wants green tea चाहिए उनको. Green tea पीने के बाद वो शुरू करेंगे इतना टाइम. ಇದನ್ನ ಸ್ಟಿಕ್ ಹಾಕ್ಬಿಡು, ಅದನ್ನ ತೆಗಿ. ಬೇಡ ಬೇಡ, ಈಗ ಇಡ್ಲಿ ರೆಡಿ ಮಾಡ್ಕೊಂಡು ಕೊಡ್ತಾರೆ ಅವನು, ಅದನ್ನ ಸ್ವಲ್ಪ ಇದೊಂದು ಸ್ಲಿಪ್ ಇಟ್ಬಿಡು. मेरे बच्चे ಪರವಾಗಿಲ್ಲ ಸರ್, ಇಲ್ಲ ಇರೋದು
ಕಾಂಪ್ಲೇಂಟ್ ಮಾಡೋದು ಈಸಿ ಆಗ್ತದೆ.
स्टेज में इरोದಾ? स्टेज में इರೋದಾ?
ಅದಕ್ಕೆ ಗೋಪಾಲ್ ಅವ್ರು ಕೊಡ್ಬೇಕು. ನಮ್ಮ ಸರ್ ಅದೇ ಟೈಮ್ ಬನ್ನಿ ಅದು ಕಲರ್ ಮಿಕ್ಸ್ಬೇಕಂತ.
A very nice afternoon to one of our residents. Immediately, a key resource person, honorable chair and co-chair person, faculty members, young participants, and my dear students. I extend a warm and hearty welcome to each one of you present here. I, Tushar Kasarji, serve as the Master of Ceremonies for today's celebration session. We gather here for this enriching academic event, and I'm certainly sure that today's session will provide us with valuable insight, knowledge, and inspiration. To begin with, I request our esteemed resource person, Dr. Shinichi Acharya, and chair and co-chair person, Dr. Yashasvi Balaraju, and GS Sama, to kindly take this information. Now, I call upon D2R of 3rd BMD to formally introduce our esteemed chairperson, Dr. Yashasvi Balaraju, founder of Therapeutic Innovation for this session. Good afternoon, respected dignitaries, esteemed faculty members, and dear participants. I am Ritual of 3rd BMD, and it is my privilege to introduce our honored chairperson for this session, Dr. Yashasvi Balaraju. Dr. Yashasvi Balaraju is a researcher, innovator, and a consultant with expertise in anthropology, fungal biotechnology, and applied microbiology. Ma'am is the founder of Altimana and director of Share World Innovation Hub, driving innovation in sustainable biotechnology. Ma'am is the ex-co-founder of Microvation, Asia's first mycelium-based innovative company in Singapore, where she contributed to advancing fungal-based solutions as the chief product officer. Our work focuses on fungi forms, material, and bio-based remediation applications. Dr. Yashaswini is passionate about translating research into scalable business solutions One mentor and innovators and researchers and specialists in community building activities in the ecosystem. Our vision supports sustainability, circular bioeconomy, and industry-friendly collaboration. On behalf of our college, I extend a warm and respectful welcome to you all. Next, I would like to introduce our honored co-chairperson for this session, Srimati K.S. Uttama. Srimati K.S. Uttama is an associate professor and head of the Department of Computer Science at MLK and SBM Mahila Mahavidyalaya. With 24 years of teaching experience, she holds an MCA from the University of Mysore and an MPhil from Madurai Kamraj University. She has published research papers in national and international journals and has received the Best Paper Presenter Award twice. She is a member of DOE and DOE of the University of Mysore, a nominee to the University Science and Technology Council, a life member of the Computer Society of India, and currently serves as the IPSC coordinator of the college. On behalf of everyone present here, I extend a warm and respectful welcome to you, ma'am. Thank you. Thank you, Ritu. I request Srimati K.S. Uttama to welcome Dr. Yashaswini Balaraju. With no further ado, let's introduce our resource person for today, uh, which is none other than Dr. Shirish K. Acharya. So I would like Dr. Yashaswini Balaraju to welcome Dr. Shirish sir. Sir. We are even glad to have his life partner, Srimati Mangalama. So we are very happy that you are attending our conference and we welcome you as well. So now I would like to call Mrs. Ma'am to introduce our resource person, Dr. Shitish K. Acharya, Professor of Archaeomics and Reproductive Biology, IIT Bangalore. Good morning, everyone. I understand that it's a very packed session. We had an exciting session with Professor G. Padmanabhan earlier this morning, and I thank everybody for a warm welcome here and introducing me. And it's my pleasure to introduce Professor Satish K. Acharya and welcome him to proceed the session. He's a professor at IBAT with nearly 3 decades of research experience in molecular biology, bioinformatics, and systems level data analysis. He has a PhD from Banaras University, followed by postdoctoral research in IIT Bangalore and University of Virginia. Since joining IBAT in 2003, his research has focused on mechanism of gene expression regulation under normal and disease conditions, leading to discovery of candidate diagnostic markers, including gene infertility. Professor Acharya has published nearly 50 papers, supervised few PhD candidates, and made over 100 research. He is also involved in national efforts on genomic architecture of hereditary deafness. And Professor Acharya combines molecular biology with integrated data analysis, is an active educator, visiting faculty at several institutions, and a strong educator of science outreach and hands-on training. I welcome you, sir. Thank you, ma'am. Building on what has been shared, I now invite Dr. Satish K. Acharya to take over the session.
Okay. Is that better? So, the process some periods of I am also trying to respond to all of these for documentation. And as okay, so this is the topic I want to talk about. Small research and what is the content of my talk. I'll give you some background. And I talk about some of the basics. Then I talk about the main story how one of the main researchers of my group who actually did the research and developed the invention of outcome and developed a diagnostic assay before he completed his PhD. Mainly talking about that and work today, and briefly I also mentioned other research, particularly focusing on algorithmic approaches. In a nutshell, I tried to all of them. But the most important thing is we need to talk here. So we need to ask questions in the middle of the talk also. I suggest the 25 minutes, not a problem. But it is for you, and I want you to ask questions. If I see people talking to each other, I want to ask you questions. Okay? Can you get it clear? Okay. You get it clear, I want to ask you questions. Okay? So there's a lot of effort by everybody and yours also. Let's try to do something. Okay? You are free to ask me any question anytime. Thank you. So this is the overall overview of the talk. First, let's understand what is the biology. It's very simple. If you look at some of these things, like somebody with a DC, it's not right. Is this okay? Yeah, so I think this is okay. It's required I share. Okay. So when you see these are marks and luxuries, some people say, and if you see a running nose, like I have a very bad allergy now, so that's the symptom called an association. So when you see an association, it has a potential diagnostics or symptoms or lot of other results could be there. An association like that would be meaningful in biology also. So for example, there are mainly two types of molecular features which we use as strong associations and indicate a lot of biological events. Okay? So an expression of a gene would indicate change. The expression would indicate a change in biological phenomenon. Similarly, a change in the sequence, like mutation, can also be something happening inside the system. So let's see a little bit more in detail. Feature of some molecules, feature when I say change, either expression, change in the structure, or mutation somewhere, all these types of features can be associated with different biological functions or diseases. Now, you all know the central law of molecular biology. We say DNA RNA protein is required going inside direction. Cells are using the DNA to generate RNA molecules and we can use it cells. And proteins are actually the nano machines which function inside cells. They are all going aware of that. Then I explain now all of the DNA molecules we study together nowadays, we refer to this entire set of DNA, representative set of DNA molecules as genome. Similarly, we say the representative set of all RNA that's not genome, representative set of all proteins is the proteome. But that's not because you know earlier people were studying one gene at a time, one RNA at a time, one protein at a time. But nowadays technology has improved, we can study everything together. And when you look at concepts of molecular biology, you stop looking at one gene now, you should realize that inside the chromosome there are a lot of genes, and that produce our RNA molecules and our protein molecules, and different combinations of protein molecules actually result in different functions. You know, hemoglobin is produced only in some cells, right? Keratin is produced only in some cells. Like that, your food delivery enzymes are produced only in some cells. So that uniqueness of protein combination is what we think is function are well known now and last is the structure of the body. Some of the biomarkers, for example, CD90 cells and helper T cells, you have got PDL of certain types of proteins. So they serve as biomarkers for those cells, cell types. There is a unique association because of the unique expression of these proteins are molecules. That's the fourth point, right? So changes in the expression patterns and sequence mutations, which might result in also if that can be a common expression. So you can use these type of changes, these features, as I have told, as potential markers. Okay? So you, I hope you are with me. I'm just researching the basics you are doing. I'm just establishing the concept of biomarkers. So we could use SNPs and other mutations as potential markers, and differential expressed genes also as potential markers. Now I mentioned about DNA. Now you are familiar with the chromosome pattern of the DNA or the chromatin or even more loosely packed DNA or the double-stranded DNA. We like the genome when we say red cytosine. So you have the chromosome sequence of the one end is one strand of the DNA right now, one strand of the other end. Like that, all the 46 chromosomes of E, along with my mitochondrial DNA, right? That is my sequence. Okay. So similarly, transcriptome also comes into picture. We know the SNPs or essentially a type of substitution which is more on the numbers. So whatever feature we have more common like that or susceptibility to obesity or side effects of the drug, these are all common traits which are not unique to one person, right? Only two people might have. So mutations which are recurring among us, recurring in the population, are referred as. So each individual possesses different features as we know, and essentially it would be one of the possible reasons for that variation. Especially there can be other types of patients because of the they will also be done. So they are often used as biomarkers. They could be important for a lot of life aspects like not effective recorded for biomarker. Now, you of course classical example of sickle cell anemia. You know, one small molecule in the DNA has resulted in shaping the protein, and then function of that protein changes. This is something you are very well aware of. Sometimes it's not that simple. Multiple recipes not really provide. But anyway, my research usually, if I talk today, will focus on that's okay. Okay. So that is another major thing, as I told, how these expressions happen. For example, not supposed to be right? Some other proteins are supposed to be expressed more. And that changes the same thing. You now, because of some changes, you have variation like this. Each 4 is not expressed, each 3 is expressed in 4 quantity. Something like this. When it happens, we see this happening. Okay? So that means you can use this pattern or this pattern, change the pattern of expression. And that's the story about our research. So what we do for that is we look at transcript stories. Transcript flow is population of all our all the transcripts. But then the transcript flow and proteome are different compared to genome. Genome is same in all the tissues that cells, right? It's the same thing everywhere, but the transcript flow and proteome represent functional. So they vary from tissue to tissue, or even cell to cell, or even the same cell from condition to condition, they vary. Okay? So that is important. So to address that, what is the transcript flow might not be delivered blood in normal condition, how it changes in the disease condition, or study as studies for transcript flow. Okay? So we want to look at expression profile, meaning expression pattern of all the genes, normal and disease. And there are massive techniques available today, for example, RNA sequencing. Basically, the next generation, right? If you extract all that you convert it to CPA, then you will be that's what we do, and we might profile, take a list of all these, and take a what we do in the laboratory is we use certain experiments techniques to extract all the items we use. We get the read data because it's raw data. We get small pieces of DNA, fragments, sequences. You align the computationally, you align those sequences, and you get computation workflow. I will not get but what is important is this expression profile. Take first and then you identify the expression. What do I mean? Very quickly. For example, in the normal tissue, you might get 50 copies of the environment. When you do all this experiment, in the end, you will get this establishment. Number of copies of the subject environment for that gene and under normal conditions. Similarly, you have a list of all the genes. But I do this for the other condition also, this condition also, same tissue. I might see almost similar expression pattern or little different. For example, you see something. So you can take this and finally you get a list of different genes. So I take this, if you get, for example, before these express more in the disease condition or B6 less. So like this, we get differential equilibrium, and this is important. What do I do by transcriptomics? I try to list genes which are expressed below the gene. That means I now can understand why this disease is happening, right? So this is the end. So I might have two different types of samples. Like this one actually is an average of certain number of samples. So let's say I have this is an average of 10 samples. And in the disease condition also, I have taken 10 samples from the patient from blood and compared to the. So finally I get this. What we do in our laboratory, we have done this type of experiment. Analyze the data, but we also do all the different tests. So this is outcome of 20 samples from one study. But if we do meta-analysis, we could actually take from one study 10 samples here, but another study we take, for example, that. So basically, I could use one study for 3 samples over, another with 30 samples. Now, I can make a better assessment of the disease. That is the third we developed. So that's our research. We developed a new quantification method which can do a better. Not everything is comparable, right? So you have to find out what is comparable, what samples are comparable, and then make comparable. So that's what we do in our research, and with that we believe we will be able to identify more reliable genes that are expressed. That is our research. We are not getting details of geography. When you identify this type of molecules, they could be having a potential value as well. And we actually explicitly validate and identify what can be valuable markers, and they can have applied values as target genes for therapies or target genes for diagnostics. That's right. Okay? So I hope you understood biomarkers. You can get your different types. Based on expression you can get. Not this transcriptomics, but you get a large number of things. You have to do some more experiments before you can all that as biomarkers. Even the small promising ones. What we identify via meta-analysis, transcriptomics, meta-analysis, and some more experiments. We identify one of the most promising biomarkers. And that's what we identify as a diagnostic. Okay. So now I will tell the story of my PhD student who actually developed diagnostic assay. So initially we needed a lot of research to compile what is the data that already exists. We have applied a lot of data. And we developed databases which actually a lot of time about 2 years we used about 40 people like you compiled all the data from one week at a time. Very laborious task and very time-taking task, but in that compiles all the data that are out there world. So we have done that. We have done similar analysis of our so like vertically we have developed and optic we have developed so here we developed a meta-analysis. What is the meta-analysis method? Here is very simple. So what is it? So for example, say that in one gene, this particular gene is expressed in all the three studies. This is also expressed in all the three. This is also expressed. And you basically derive a consensus score. So what is it? Very simple. So you basically say we 1, 4, and 6 are, I can say they are equal to all of them, I equal 4. This is something very simple. Anyone of you can do this. We can do technology to go well and approach that. This is something very high-fi, very actually can do it. But that is anyone of you can do that. Okay? You have to start thinking on your own and understand the basics. When I started my career, before the getting from but we because we had done something. So you all can do this level of research point. So similarly, we have actually identified the technology which can test more reliably their assets. So this is how we develop technology which are more reliable than the now, when we develop that on micro we are not really sure whether that will work when we, because that time harvesting that's a new thing. And what we did is we did an experiment on the same disease condition. We collected some of the samples and flow samples. And we tested the genes which are having higher reliability score, lesser reliability score, and least reliability. So the hypothesis was that both which are having least reliability will also be reproduced to a lesser extent. Right? That's what happens. The top ones are actually more reproducible even with so our methodology was correct. So we went on doing certain research like this, and eventually this is what we went on to next to identify potential biomarkers. What we did is, we collected clinical samples by collaborating with the doctors and there are procedures our procedures collected the samples and we extracted all of them, because of them, all the and then we try to identify interesting research specifically, alternatively, twice forms of our technology we established. And we also developed a special software because there was not reliable software designed by us for as a very important for our so we developed our own software and then validated it as our and then we should not so this is the software which we call it Express and this is probably one of the very few software of this after experimental so we developed the clear differences can be clear to this tested that was so only can be tested so we take your number because of that. We have had that we did that. Finally, we did our experiments and this basically shows the artificial PCR first constitution followed by quantitative PCR. So, people say that there are some things tests, you just usually can use more samples and do the experiment and it shows that what we test is not everything was good, not everything was improved. But our 100 research clusters is selected, we were able to get 16 of it consistently 100%. So, and this is going now I don't know and he developed, he applied for the biotechnology how to do a search to see that sort of and we continue to do that. We continue to see how that goes. But this is the collaboration with the company I started. Now I can see that I use that. But now but there was some collaboration there, but there's a new meta-analysis. Second meta-analysis developed for the and with help of all of that together, so we also identified new transcription of big data vessels because what expressed only the that is something we established. Suppose some genes are expressed more or less consistently. Among them, there are two types of those which are expressed everywhere. So, when you have one that are expressed more significantly, then they are upgraded. So we wanted to start the agreement of big data. And we established the type of and then made our platform. We identified this specific area we have already. We use them for transmission factors. So I not get to the part of the we also have database for operating platform and cloud. And this is what we did this. That's the world. Alright, so overall, our research goal is try to look at which and to do that, we have to look at mechanisms. Have so all the levels we try to understand. That's how actually we made research goals. Between biomarker discovery and so far I have told you about the biomarker discovery. Okay. I hope you have understood it. What we are now doing. As I told, we are looking at so transmission is regulated by example. But whether of transmission factor -5 or any smoker, the word transmission factor. So we have to and then have but we also have other mechanisms. So there could be enhancers here to be able to move along and some other proteins from the mind and that's the so the DNA duplicate strategy that's the second mechanism. Then there are duplicates for example, this is with the now we also have credit patterns in the DNA data. MicroRNA the way the chromatin is back, I told you at the beginning, it's not random. Every sequence is different. The way we are trying to use we are trying to look at all the data that is available for all types of these mechanisms. Okay? People, for example, look at bulk architecture, bulk okay? Where you can also use single cell RNA. It's separate cell and there are two kinds of that plasmid-clone, single cell RNA, the cells with RNA, or they take the cells wherever they are. So that is spatial process. Then there is also non-coding RNA like those they are also used. All of them plus proteomic data that are available, computational solutions that but we are working on software domain also. We are trying to use that data and attach this data. That is an experimental technique which actually tells you where the somatic you are trying to make use of that data also, and then there is sequencing data which basically tells you if you have a protein of interest, that protein will go and bind to which all regions in the entire one protein binding. So we get data from that. We also get data from thyroid, tissue, and that. Whatever is available, including chromatin packing, which comes from so all of them we are using today in multi-omics, and we are trying to apply this analysis, and we are also trying to apply artificial intelligence so we use various type of techniques computation and nowadays we are looking at multi-omics. These are all each one is a one type of parameter. It's a lot of give about title. All of this is massive. We try to put together all of them. That is the way we are. Fully appreciated to me. So we try to establish new positions. We are all existing. We all that that's what we are doing. So I think I'd like to thank my team because it's always been one of my company. That's me. Thank you very much.
Thank you for sparing your valuable time to share your knowledge with us. We are deeply honored by your presence. Following with the next session, I request our esteemed I request Arthur Ma'am, Department of Microbiology, to facilitate Srimati K.S. Subrahmanyam. The moment I will open up brackets. Now, following with the next session, I request our esteemed resource person, chair and co-chair, Dr. Rashmi, Assistant Professor, Department of Biochemistry, JSS College of Arts, Commerce and Science, and Ms. Dr. Srimati Ranjita to please come on the dais. First and foremost, I would like to welcome Mr. Chandrakumar Devji, the resource person, and he is the System Analyst Deputy Chief Information Security Officer, MITC, New Delhi. Following with that, I would like to welcome Dr. Rashmi, who is an Assistant Professor, Department of Biochemistry, JSS College of Arts, Commerce and Science, to give for the session. I request Srimati Ranjita D to welcome Dr. Rashmi Lord.
ಕಳಿಸ್ಲಾಗ್ಲಿ ಕಳಿಸ್ಲಾಕ್ಷಿ.
ಇಲ್ಲ ಇದೆ ಮಾಮಿ, ಇದೆ ಒಂದು ನಿಮಿಷ.
ಲೆಟ್ ಇಮ್ ಜಾಯಿನ್. ಅವ್ರು ಜಾಯಿನ್ ಆದ್ಮೇಲೆ ಮಾಡ್ಬೇಕು. ಒಂದು 5 ನಿಮಿಷ ಲೆಟ್ ಇಮ್ ಜಾಯಿನ್. ಅತಿಯಮ್ಮ ಲೆಟ್ ಇಮ್ ಜಾಯಿನ್ ದ ಸೆಷನ್ ಸ್ಟಾರ್ಟ್. ಅವ್ರು ಸ್ಟಾರ್ಟ್ ಮಾಡ್ತಾರೆ. ಅವ್ರು ಮಾಡ್ಬಿಟ್ಲಲ್ಲ ಈಗ. ಲೈಟ್ ಆಫ್
ಜಾಯ್ನ್ ಆಗಿಲ್ಲ ಮಾಮಿ.
ಜಾಯಿನ್ ಆಗೋದು ಕಾಯ್ತಾ ಇದ್ದಾರೆ. ಹಲೋ ಮಾಡ್ಬೇಕಂತೆ. ಹಲೋ ಮಾಡಿದ್ದೀರಾ ಸರ್?
ಮಾಡ್ತಾ ಇದ್ದೀನಿ.
ಮಾಡ್ತಾ ಇದ್ದಾರೆ ಮಾಡ್ತಾ ಇದ್ದಾರೆ ಮಾಡ್ತಾ ಇದ್ದಾರೆ ಮಾಡ್ತಾ ಇದ್ದಾರೆ. ಆಯ್ತು ನಿಮ್ದು ಆಯ್ತು.
ಎಸ್.
ಎಸ್.
Now, I welcome Sanjana MP from 3rd PMDP to formally introduce our esteemed chairperson, Dr. Rashmi, for the session. Good afternoon, everyone. I am Sanjana of 3rd PMDP, and it is my great privilege to introduce today's state person, Dr. Rashmi R, who is serving as Assistant Professor in the Department of Biochemistry at Jesus College of Arts, Commerce and Science, Mysore. Dr. Rashmi R completed a PhD in Life Science from Jesus Academy of Biotechnology and Research, Mysore. Her doctorate work focused on the role of NRF2 activator melatonin in experimental diabetes, highlighting her strong research focus in diabetes and cellular protective mechanisms. She holds a master's degree in biochemistry and bachelor's degree in biochemistry, biotechnology, and microbiology from the University of Mysore. She brings around 8 years of teaching experience at both undergraduate and postgraduate levels. She has been actively involved not only in classroom teaching but also in guidance programs, supporting academic programs, and contributing occasional and field-based courses under UGC initiatives. Dr. Rashmi has taken on several academic responsibilities over the years. She served as a member of Board of Examiners and Board of Studies in Biochemistry for multiple colleges and the University of Mysore. She is also an examiner, paper setter, and evaluator, contributing to maintaining academic standards and quality assessment. Her research interests mainly include diabetes mellitus, protection of pancreatic beta cells, medicinal and novel plant therapies, NRF2 signaling pathways, and the use of micronutrients and nutraceuticals in managing chronic diseases. She has also published multiple research papers in peer-reviewed journals and has also contributed to chapters and conference papers. Among her recognitions, she has received the University Research Fellowship and PhD Fellowship from the Department of Backward Classes Welfare, Government of Karnataka. She was also awarded Best Oral Presentation at National Conferences for her research on the anti-diabetic effect of melatonin. In addition, she has participated in completing several faculty development and training programs to continuously update her academic and research skills. We are happy to have you here, ma'am, and we look forward to learn from your experience and knowledge. Now, I would like to welcome the co-chairperson of today's session, Srimati Vaishravati,
Associate Professor of Computer Science. She holds an MSc and MPhil in Computer Science and has several years of teaching experience with PhD and CPS students. She is actively involved in student mentoring and academic activities. Her areas of expertise include artificial intelligence, image processing, biometrics, and blockchain technology. She has presented research papers at national and international level conferences and published her work in academic journals. We are glad to have you here, ma'am, as a co-chair of this session. Thank you, Sanjana, for introducing the chair and co-chairperson for this session. Now, I request Dr. Rashmi to introduce our esteemed resource person, Mr. Chandra Kumar, for the session. Very good morning to all. A very warm welcome. It's my immense pleasure to introduce our esteemed speaker of today's session, Sri Chandra Kumar MJ, Resource and Systems Analyst, Deputy Chair, Informative Secretary Officer at NICOM, Ministry of Education, New Delhi. Mr. Chandra Kumar is a dedicated technology professional and academician with 9 years of experience in managing IT infrastructures, leading digital transformation initiatives, and overseeing institutional cybersecurity as the Deputy Chief Information Security Officer at NICOM. Currently, he pursues his doctoral research on the applications of artificial intelligence in learning analytics, particularly within the framework of NEC 2000. Sri Chandrasekhar completed his Master of Technology in Digital Electronics and Communication Systems from BPU Regional Center, Mysuru, and beyond that, he earned his Master of Engineering in Electronics and Communication from NIE Institute of Technology, Mysuru. He has served as a resource person and invited speaker in the domain of artificial intelligence, and he has delivered expert lectures and workshops on artificial intelligence, machine learning, and cyber security to students, faculty members, and research scholars across various esteemed universities. His professional profile is further strengthened by allied NPTEL certifications and comprehensive suite of Google certifications in advanced domains such as computer vision, Internet of Things, and generative AI. These credentials not only validate his technical expert, but also position him the lead and innovative in the field of educational technology, driving an impactful digital transformation in academia. With this information, now I request Sri Chandra Kumar to deliver his talk on generative AI in the life science. Over to you, sir. Thank you.
Uh, Thank you. I hope my voice is loud and clear.
Yes, sir.
Thank you so much. Thank you very much for inviting me to this national conference on life sciences. As the introduction has been given briefly by Dr. Rashmi and other people, it's my privilege to speak on this national conference related to the very important buzzing topic.
Yes, sir, visible.
Yeah, I'm loading one more time. Once again.
Yes, sir. Side by side आएगा.
Yeah.
Okay.
I hope my screen is visible.
Yes, sir, it is visible.
Okay, thank you. So the topic is on convergence of generative AI in life sciences, advancing the predictive healthcare and digital pathology. And just wanted to say something about the generative AI, what we are heading and where we are heading earlier. And the things are like shaping in very drastic phase. And this is where the transition has to be taken care on any subject. The AI is going very narrow to the subject to subject based racks, that is called retrieval augmented generation. Now, in the case of an healthcare and the digital pathology, it has been advanced also. Before this boom has been coming, healthcare has witnessed a lot of transformation because of AI, patient monitoring systems, and again in identification of drug discovery or making some advancements in life sciences with the help of generative AI. I just wanted to pitch in that the evolution of generative AI has been taking very rapidly. In next another 5 years, things are very smart and decision makings will be very smart and time process will be very minimal. Latencies will not going to take care about these things, and we are going to get in critical stages. We can able to take the decisions in crucial stages of something related to patient who are suffering from cardiac diseases or some early breast cancer detection. These are the latest advance and discoveries where we can able to act. So, prevention is better than cure now. So we can able to identify the threats before what is happening. We can predict the diseases earlier in the earlier stages of cancer, and we are advancing to the later stages that how we can able to cure with the help of AI and artificial intelligence. Now with the help of 5G and 6G technologies, doctors can able to operate the patients throughout the world using this artificial intelligence. Wherever he is sitting, he can able to work on that particular surgery. You can able to do it from all part of the world. And this is the where the applications of generative AI in life sciences as well as in medical is been shaping up. Let me move to the next slide. The research overview about the generative biology paradigm shift is about a 24 to 26 transition from traditional predictive modeling to generative biology foundations. So that earlier it was predicting, again it has been predicting to the problem wise statement. It is getting narrowing down. The transition is have been taken care with the help of artificial intelligence, machine learning, computer vision. The computer vision is taking very serious inventions with the help of now I think you can able to see autonomous vehicles or with the monitoring patient monitoring system in the ICUs. The computer vision plays a very vital role when it comes for the medical healthcare or in the point of the even for image analysis. We can able to work with this artificial intelligence and machine learning convergence with the computational substrates. Foundation models like AlphaFold or 3 and ESM3 and vision transformers for predictive healthcare. I'll try to give you some small amount of a demonstration how we can able to get these things with the help of the research point of view. And breakthrough in the application analyzing agentic AI orchestration and virtual staining in digital pathology applications. So agentic AI orchestration is very important today, and now adding the brain to the requirement like a virtual brain to the analysis is very important, and we can able to predict and we can able to analyze and act in prior to any problem. Future healthcare transformation discuss data-driven feedback loops and proactive prevention strategies in modern healthcare. So we can able to act a data-driven decision-making mechanism. Data is not only meant for analysis, like any patient data or any protein data or any life science data can be fed to AI models, and AI you can able to predict and give lot of footprints to the next what we can able to take the decisions based on data is not only meant for analysis, it is meant for predictions and forecasting. With the help of the medical data is a very important data. We have to handle it very carefully also. This ethical concerns will also be there. We have to take a proper appropriate clearances before we take up for analysis. Then the paradigm shift to generative biology from 24 to biological language models, vision transformer, image-based diagnostics. So images will be fed to the transformers, and it will be identified. This feature extractions will start, and it will be giving a digital output with the help of vision transformers. Now, integration framework we can use like a machine learning and computer vision, like unified predictive healthcare systems, generative AI substrate, de novo design capabilities, data-driven loop, continuous improvement cycles. When we feed this data to any AI model, it will going to continuously we can able to see the efficiency of any model increasing, getting the error rates of 10% or whatever the error rates we get, we have been able to minimize by continuously training the any AI models. So by this, the 50% of R&D reduction and 60% latency cut, if we start using the computer vision and the AI into this subject, this especially for life sciences, for R&D, this will going to impact and research, and we can able to good amount of we can able to get good amount of results in in upcoming research. So core technologies and integration, generative AI as a computational substrate for life science innovation. Deep neural networks plays a vital role, like input layer, multiple hidden layers will be there, and output layer. What we try to do when we train any data, there are two types of data, that is structured and unstructured data, labeled and unlabeled. The data should be fed, like suppose if it is structured data, the output will also be structured, and when it comes to unstructured, the model has to analyze and see what kind of essence can be drawn from that data. That will going to take it up is artificial intelligence comes to the role. The machine learning will going to try to train these kind of data with the help of algorithms like decision, random forest, decision tree, and depth first sorting algorithms. These are the summary bubble sort. Other algorithms will be there. These trees or these algorithms are used to analyze the data and take the appropriate decisions with the help of machine learning algorithms. Scikit-learn for machine learning, we call the library for scikit-learn. And ML training and computer vision plays a very vital role, as I told earlier. Multimodal prediction for unified representation here. Integrate diverse biological data streams and cross-modal learning, transfer knowledge across modalities. So transfer learning is very important in the machine learning. We have to use these kind of neural networks for any data predictions. When we are started using this data networks, this will going to give us throw us a light on our output. Now novel molecule generation, AI-driven molecular design from the scratch, or reduce the R&D time by 50%. So these are the things we can able to check it out with the help of a generative AI framework. Bringing generative AI is again a new approach to the any problem. Started working on it, and it is a subject to subject, it will vary, and problem to problem the solutions will be different. And undruggable target like protein structure prediction, AlphaFold 3 breakthrough capabilities, and binding site identification unlock previously inaccessible targets with the help of the AI and ML. So the methodology for materials and methods, multimodal foundational models for predictive healthcare, you can able to use AlphaFold 3 for protein structure prediction, advanced deep learning for accurate protein folding prediction and drug target identification, and biological language models, large-scale transform models for biological sequence and analyze and function prediction. So these models I'm going to give you in demo in a while. With the help of some Python, and I'm going to show you this how this can be achieved also. And medical image and high resolution tissue slide processing and feature extraction. With the feature extraction is very important in image processing. Even slight deviation from the feature extraction, it will deviate the output. And image processing plays a vital role in image analysis. And digital pathology, automated detection of cellular abnormalities and disease markers, and multi-scale analysis. 60% latency again will going to get reduced, and R&D time also will get reduced by 50% from this kind of using this kind of multimodal foundational models, AlphaFold 3, ESM, and vision transformers. Not only this, there are some other things, some other techniques also there. I think this is a need of the hour. We need to adapt as per the problem statement. Again, getting into methodological framework, we'll go to the flow matching, continuous normalizing the flows for de novo molecular design with smooth latent space interpolation. Again, we can able to go for physics, physics informed neural networks, quantum mechanical constraints, unified latent space. You can see with the representation, physics based constraint for accurate molecular property predictions. and integrating into the next, predicting the absorption, distribution, metabolism, and the toxicity profiles early in drug development pipelines. These are all the things we can able to do in the generative AI. Molecular dynamics AI accelerated simulations for understanding protein aligned interactions and binding affinity estimation. We can able to make a simulations with the help of generative AI. I'll also try to show this simulation now. End-to-end computational workflows from design to validation in pharmaceutical research. This is very important. Validation is has to be taken care with the real-time example. That is where the AI generated to real-time and physical real-time requirement should be validated, or else it is should not be taken up further. So latency reductions, again, R&D timeline will be very minimal. Agentic AI orchestration, real-time interoperative guidance will be given. Autonomous decision support systems will be there, and virtual staining, label-free tissue imaging, and high-quality equivalent diagnostics will be there. Slides. Targeting undruggable proteins with de novo design and novel molecular generation, predictive ADMT property predictions, or proactive disease prevention. The integrations is very important. That multimodal AI system deployed in predictive healthcare will be very strategic step to take the decision. It is going to play a vital role in taking the decisions. And getting into the agentic AI orchestration and virtual staining technology, real-time guidance we will going to get it in agentic AI. Now AI as a co-worker, you're going to get support in taking the decisions. Computer vision will going to take up what is happening. It will going to make the analysis. Automated system, self-directed computer vision orchestration workflows. Labeling in images, transform tissue images without traditional staining. Again, diagnostic grade quality, generative the H&E equivalent slides for pathology, and rapid processing, 60% latency reduction in digital workflows. Core transformation about in the conclusion and the impact, I would like to add here that synergy of generative ML and vision-based predictive models are re-architecting the life science into continuous data-driven feedback loop. This will be major step we have to take. Now, getting into the platforms and understanding is very important. Healthcare paradigm shift, predictive healthcare, security and trust, and technology enables like a generative AI foundation, computer vision is very important. Data-driven, data is to be is a main role. It will be playing on any life science part or getting database decisions on. I'm going to give some demonstrations with the practical how we can able to do with the help of data-based decisions also. So the black box in there is a gaps also here, limitations from it. Gaps like black box model, billion parameter model, lack transparency in decision, complex representations, again clinical adoption barrier. And one more thing, now today the GPU clusters will be costing like anything. I think you know about India is deploying a lot of data centers to get attract the foreign direct investments on data centers, infrastructure investment, and environmental impact. This is very, very important. Significantly, energy consumption of any GPU or data center will going to take a lot of the energy and electricity for this. And future directions, we can able to see quantum classical hybrids like subatomic molecular dynamics simulations, combining quantum computing power with classical AI for unprecedented accuracy in drug discovery, and autonomous laboratories like Lighthouse Lab, fully automated experimental facilities operating 24/7 without human intervention. And agentic AI control, intelligent a system orchestrating complex multi-step experiments autonomously. And real-time optimization, continuous learning and adaptation to the of experimental parameters for optimal outcomes. For this, I would like to say research impact and key metrics will be reduced with the help of generative AI. Let me go to the real-time demonstration. I'm going to take it up to the demonstration part. One second, I'm going to share my screen once again. Here the activity, I would like to show it in practical in front of you, that whatever we are trying to do with this the proposed things which has been shown in the PowerPoint, that has to be done it in front of you. Now, generative AI, generative biology demo is like this, like in de novo molecular design. The traditional drug discovery is like looking like a needle for a stack. Generative AI allows you us to print the needle. We are moving from searching to the engineering. What I'm trying to do here in the collab, this is a collab is also the Google DeepMind project. If I run this, there is a code, it will going to generate me the completely biomolecule demo from this. Can you see? I can agentic AI generated valid structure for latent space ID with this. This is the output we can able to AI generated lead compound. This has been generated for the first activity. Now, one more activity I would like to demonstrate, that is called computer vision demo. In many parts of the world, access of chemical reagents for pathology is limited. AI can perform virtual staining, taking a raw label-free image of tissue and instantly generating a diagnostic grade slide. So what I'm trying to do, we're going to give some raw tissue as as an input, and we use OpenCV to apply color mapping and transformation, explaining the real world of P200 GPU cluster. This would be a vision transformer analyzing the morphology. And the key, this is not just an aesthetic change. Our vision models are trained to identify the cancer cell nuclei that are invisible to human eye in the raw image. This art of agentic AI in the operating room, you can able to see. I'll try to generate one more then. Can you see this is a generated candidate from this molecular generation, de novo molecular generation from the output. This is how, by putting the code and by putting, this is a platform where you can able to generate these kind of platforms outputs. And let us run one more activity, that is predictive analysis data loop. An AI might be designed perfect molecule, but it might be a toxic to human liver. So pins into simulate physics and biology before we ever physics lab. So I'll go to predictive toxology or real physics constraint. We have to think about it. As you can able to see, I'm going to make one more output demo using computer vision. Can you see this now? We can able to input as a label free image raw tissue, and the output will be virtual staining H&E equivalent. These are all the real-time practical approaches we can able to adapt and implement. And one more something related to the alpha fold, we can able to see virtualizing the structure of 7p6a. I hope you can able to see this. High quality images are getting with the help of AlphaFold, complex human protein structure. Even not only this, we can able to achieve the demonstrations with the help of simulations of DNA unfolding or protein structure with the help of AI. Just a small glimpse about how we can able to do. This is also Google DeepMind project. We're going to see the output about DNA simulation or protein simulations we will going to take up, or any other simulations you need, we can able to generate with the help of AI, and we can able to see this in real time. It will going to take minimum 1 or maximum 20 to 30 seconds. I'm going to show the demonstration. Meanwhile, I'm already, okay, I got it. It is generating this. I'm going to download it immediately once it has completed the generation, the simulation, DNA simulation. It has done. Let me open this. Can you see this? DNA simulation, we 3D LX. Even we can make it interactive also, this simulation. I'll make it interactive now. It will make an interactive simulation, and I'm also having some downloaded code, DNA related. I hope you can able to see these scores like DNA related. It is there. Uh, One more is there with me. You can see the major groove. I hope you can able to see. These are all the simulations we can able to do it with the help of generative AI. Even protein structures can also be done. So you can able to see the genetics lab. These are all the capabilities of generative AI we can able to do. And we can generate about the protein structure also we can able to do. And I'm going to show this demo for you. If you need collab, we can able to work with collab and generate whatever you read it for protein analysis or for biological simulations, generating the generative biology data. I'll download this one more option. And you can able to see this, I can able to make the speed and basic pairs. You can see I can able to make geometry, helix width. I can able to change. I hope you can able to see this helix width and the twist amount. You can able to see a total height and color. We can able to change from here. So these are all the simulations and capabilities of generative AI, interactive DNA, and we can able to work on that protein simulation. If you need any simulation, you can put in the chat box also here. I try to put the simulation. Anybody having any queries, you can put it in the chat box. In collab, in deep dive, you can able to upload the data of any medical data. We can able to do the analysis. I'll try to download any medical data, medical CSV data. I'll take it up from any data from Kaggle healthcare data set. A small data set, I'll take it. So I got one more simulations where you can use this for yeah. I hope you can able to see this. This is a related to protein structure, amino acids. You can see. With the help of generative AI, we can make this kind of simulations for your life sciences, and we can analyze the data of any medical data in Colab. For example, I'm going to upload one data set here, life science CSV file. Yes. Now, friends, if there is any queries, since as per schedule, we must take care of this time schedule. Now, if there is any queries, do let me know, and I'll going to give one small short demo within 2 minutes. 1 second. This is a very advanced thing we can able to work on your side. And for research, it is very important. And we can load the data set here. I'll upload one data set. I'm loading a healthcare data set. Provide possible machine learning predictions along with the interactive visualizations. Any doubts? If anyone having any doubts, do let me know. Any doubts? Do anybody having any doubts? They can ask. Another 1 or 2 minutes, I'll wind up by showing this output. You can see I have downloaded the data is preparing, loading healthcare data, processing data for ML, and training machine learning model, generating predictions or evaluation. It will going to you're going to see it in real time, whatever it is happening in front of you.
Are there any queries to us, sir?
Oh, any Q&A, Dr. Rashmi, please let me know. Oh, I can able to wind up now. If any queries, madam, you please let me know if any queries or anything.
Sir,
Because
they were asking if they could share your email ID so that they can share their queries with you.
okay, thank you, and let me stop sharing.
Thank you, sir. We were deeply honored by your presence. Now, I would like to request Dr. Rashmi ma'am to come forward and speak a few words on her exam. Second opportunity to engage with Mr. Chandrakumar and explore artificial intelligence and its usage in the life science field. Thank you, sir. Thanks so much.
Thank you.
Thank you, ma'am. So now it's time to express our gratitude to a small token of appreciation. First and foremost, I would like to acknowledge Mr. Chandrakumar MJ for his spend valuable time and giving the knowledge to us was a very great thing. So thank you, sir, and we would like to give felicitate with a moment as a token of gratitude. So thank you, sir.
Thank you.
And furthermore, I would like to thank Dr. Rashmi for supporting us as chairperson. And I request Srimati Rachita Ji to thank Ma'am with a moment as a token of gratitude. Thank you, ma'am. I would also like to thank Srimati Rachita B as being the third chairperson for the session, and I request HOD of Meghna ma'am, Department of Biochemistry, to felicitate ma'am. Thank you, teacher. I would like to call upon Shama on the stage.
With that, we conclude the morning session. We will now be leaving for lunch. It will be from 1:30 to 2:30 p.m. The lunch for delegates and resource persons will be provided in Vidyosava dining hall, and student participants are requested to go to G006 in the ground floor. G006, room number G006. As we pause for lunch, let us carry forward the spirit of today's discussion beyond this hall. May this meet be a space for enjoying your conversations, collaborations, and new beginnings. Thank you and have a great day ahead. For correction, the student participants should have lunch in room number T314. I repeat, room number T314. Thank you.
ಇಬ್ರದ್ದು ಇದು ಓಪನ್ ಇದೆ.
ಹಾಂ ಹಾಂ ಸರ್.
ಅವ್ರದ್ದು
ಹಾಂ ಅದೌದು. Wow. She left her brain back at home only.
ಆಫ್ ಮಾಡಿದ್ದಾರೆ.
ಡ್ರಾಮ್ಯಾಟಿಕಲ್ ಎಕ್ಸ್ಪೀರಿಯನ್ಸ್ ಅಲ್ಲ. ಇವತ್ತೇ
ಒಳ್ಳೆ ಗಂಡ ಸಿಗ್ಬೇಕಂತ.
ನಿನ್ನೆ ಏಕಾದಶಿ ಅಲ್ಲ, ಇವತ್ತು
ಅವ್ನು
ಏಕಾದಶಿ.
ಸಿಗಲ್ಲ ಈ ಜನ್ಮಕ್ಕೆ ನನಗೆ.
ಏಕಾದಶಿ is ಮಾಫಿಯಾ ಮಾಫಿಯಾ. He wants a mafia guys. You know, we'll make a scenario for her. Okay? We'll arrange a guy for her.
I will arrange a guy.
ಸರಿ ಸರಿ ಮಾಡು.
Guess what? First we'll arrange a guy who will pretend to get married to her. Okay? We'll arrange a marriage. ಏನ್ ಬುಸ್ಗಳೇನೋ ಬಿಟ್ಟಾಕಿ. Okay, we'll make an arrangement. Then we'll make, you know, we'll take it to marriage venue and all. ಅವನೇ ತಗೊಂಡು ಹೋಗ್ಬೇಕು ಕಣೆ. ಅದೇ ಕೇಳಿಸ್ಕೋ.
ಏನು ಮದುವೆ ಇದ್ದಲ್ಲಿ that random guy I will find with that very random guy.
ಏ ಚೀಟು.
ರುಚಿ ಗೌತಮ್ ತರ ನೀವು ಬೇರೆ you should find some find them very good guy for
ಯಾರ ಮುಗೋಡಿ
ನಿಂಗೆ ಗೊತ್ತಾ ರಾಜಸ್ಥಾನ
ಅವ್ರನ್ನೇ ನಾನು ಟೆಕ್ನಿಕಲ್ ಅಸಿಸ್ಟೆಂಟ್ ಟೆಕ್ನಿಕಲ್ ಆಗಿ ಮಾಡಿ ಆಫ್ಲೈನ್ ಸೆಷನ್ ಆ ಕಡೆ ಅನ್ಸುತ್ತೆ ಆಫ್ಲೈನ್ ಆ ಕಡೆನಾ ಆನ್ಲೈನು ಸೆಮಿನಾರ್ ಅದಕ್ಕೆ ಅಲ್ಲಿ ಕೆಳಗಡೆ ಮಾಡ್ತೀವಿ
ಲ್ಯಾಪ್ಟಾಪ್ ಇದೆ
it?
Yes.
Let's.
This one.
Okay, fine.
Not yet.
Okay, fine. Okay, I have 25 minutes to make them awake.
Does not respond, you know, unresponsive, right? Or exit. Exit means close and open. Or open in this one. If I do that now, I don't expect them to teach me. Government rules ಪ್ರಕಾರ ಅಷ್ಟೇ ಬೇಕಲ್ಲ cheap to go the dresses everything. My mom ಇಲ್ಲ actually Day-1 cost. Day-1 sent it. They start my mom he was not following all this
ಊಟ ಆಯ್ತಾ ನನ್ನದಿಲ್ಲ ಊಟ ಆದಾಗೆ ಇದು ಈ ವೈಫೈ ಇದೆಯಲ್ಲಿದ್ದು
ಉಂಟು ಸರ್
ಕೊಡ್ಬೋದಾ
Oh, we can't give that. ಕೊಡ್ರು ಒಂದು ಇಲ್ಲಿಂದ ಆಯ್ತು ಒಂದು ಅವಳು ಕುತ್ಕೊಳ್ಳೋದು When I go to SDM, when you get 70 SDM. ವೈಫೈ ಅದು ಕನೆಕ್ಟ್ ಆಗ್ತದೆ ಏನು ಹೇಳು ಪಾಸ್ವರ್ಡ್ ಇಲ್ಲ ನಿಮ್ಗೆ ಯಾವುದು ಕನೆಕ್ಟ್ ಆಗಿಲ್ಲ. ಹಾಂ ಹಾಂ ವೈಫೈ ಇಲ್ಲ ವೈಫೈ ಸಿಗ್ತಾ ಇಲ್ಲ. ವೈಫೈ ಸಿಗ್ತಾ ಇಲ್ಲ ಮೊಬೈಲ್ ಸರಿ ಕನೆಕ್ಟ್ ಆಗ್ತಿಲ್ಲ. ಕನೆಕ್ಟ್ ಆಗಿದೆ
Everyone once again. Let's continue with our session and stop. Now, I request our esteemed resource person, Dr. Srinivasa DM, Chair and Social Person, Dr. Sarah Paul Fatima, and Ms. Sanjay Sri to take your seat on the dais. First and foremost, I would like to welcome the resource person for the session, Srinivasa DM, MBBS and Scientific Manager, ECS Scientific Manager, Professor AGS National Institute of Malaria Research, Denmark Garden, ICMR Complex, Bangalore. We welcome you, sir. I request Dr. Sarah Paul Fatima to welcome Dr. Srinivasa DM, sir. I would also like to welcome the chairperson, Dr. Sarah Paul Fatima, Associate Professor and Head Department of Botany and Microbiology, Government College, Copenhagen, India. We welcome you now. I request Ms. Sanjay Sri ma'am to welcome Dr. Sarah Paul Fatima. Sorry. I would also like to welcome the social person, Ms. Sanjay Sri ma'am. We welcome you ma'am. Now, I would like to call upon Sandana next from 3rd AMP to formally introduce our esteemed chairperson, Dr. Sarah Paul Fatima, to this session. Good afternoon, esteemed members and the attendees. I'm Sandana, guest of 3rd AMP, and today to introduce our organization, Dr. Sarah Paul Fatima. Dr. Fatima is currently serving as professor and head of department of botany and microbiology at Government College, Copenhagen. Since her appointment in 1992, she has dedicated more than 3 decades to academics, shaping her mind and strengthening the field of microbiology through her commitment and leadership. Ma'am PhD from the University of Mysore and a doctoral research focused on biology and management of cosmosis data or means reflected a strong academic foundation. Throughout her distinguished career, she has published 16 research papers in peer-reviewed national and international journals, authored 2 books, and presented her research at several national and as a committee member, she has guided Temple scholars and numerous MSc students. She has also successfully completed a UGC-funded minor research project on airborne fungi from Mysore. Her leadership roles include serving as the president of the Association of Microbiologists of India, the president of Microbiology Society, the president of Alumni Association, and chairperson of the board of examiners and board of studies in microbiology. She continues to contribute actively to the academic development of University of Science. Now, as a life member of the Science Knowledge Association and Association of Microbiologists of India, and serve as the general reviewer for the African Journal of Microbiology. With such an inspiring journey and remarkable contribution to science and education, we are truly privileged to have her with us today. On behalf of the college, I extend a warm and respectful welcome for you.
Thank you, Chandra, for the speech that project. Now, I request Dr. Sarita for the part of the key resource person, Dr. Srinath P.M.
ಡಿಗ್ರಿ ಆಫೀಸಿಂದಲೇ
Participants from various colleges and various schools. A very good afternoon to one and all present here. At the outset, I would like to thank the organizers of this national conference for providing me this opportunity to introduce the speakers of this day. It is indeed an honor and a proud privilege for me to introduce the distinguished speaker of this session, Dr. Srinivasa P.M. to the August gathering. Doctor by profession, his area of expertise is community medicine. Dr. Srinivasa obtained his MBBS from Vijayanagar Institute of Medical Sciences, Vallaki, Rajiv Gandhi University of Health Sciences, Karnataka, in 2008. Then he did his BPH in community medicine from Government Medical College, Thiruvananthapuram, University of Kerala, in the year 2011. He obtained MD in community medicine from Government Medical College, Thiruvananthapuram, University of Kerala, in the year 2012. Dr. Srinivasa has obtained has served as public health physician and medical scientist with extensive experience in community medicine, infectious disease research, and national health programs, contributing significantly to malaria, tuberculosis, and climate health research. Doctor has many awards and recognitions to his credit. To name a few, he has received Meritorious Exceptional Scientist Promotion Award on 25th June 2025 for his outstanding contributions in public health research. He is also the recipient of DHR International Fellowship. Further, he served as the principal investigator for the subnational survey of CFAPM malaria impact across 11 states of India. He is a core team member in India's first COVID-19 zero survey and contributor to landmark
COVID-19 vaccine trials. Further, he has contributed to major studies including subnational malaria surveys, TB sentinel surveillance, diagnostic accuracy studies, and climate vector disease research. His scientific contributions in the field of community medicine are highly commended. He has published more than 50 research papers in peer-reviewed national and international journals. With this brief introduction, I request Dr. Srinivasa to take over the session. Thank you so much.
Give this to him. Tanu. ಇಲ್ಲ PPT ಆಫ್ ಮಾಡಿದೆ ಇನ್ನು
Are you all with me? Let's sleep together.
ಲೈಟ್ ಆಫ್ ಮಾಡಿ ಲೈಟ್ ಆಫ್ ಮಾಡಿ
Will you all have to sleep? ಊಟ ಮಾಡಿದ್ರೆ ಇಲ್ಲ?
ಸೆಕೆಂಡ್ ಒನ್
Are you feeling sleepy?
ಏನು ಮಾಡ್ತಾರಮ್ಮ
Those who are not saying yes are lying. For normal human physiology, high blood glucose should induce sleep. If not, you should get tested.
ಬಂತು ಬಂತು ಆನ್ ಆಗಿದೆ ತಗೊಳ್ಳಿ ತಗೊಳ್ಳಿ
ಸೆಕೆಂಡ್ ಒನ್ ಅಲ್ಲ ಥರ್ಡ್ ಒನ್ ಅನ್ಸುತ್ತೆ ಥರ್ಡ್ ಒನ್
Am I audible? Am I audible?
ಬಂತು
If you want to sleep, I should be audible to you. Because for the next 20-25 minutes, I'm just going to say some narrative stories. Before I start off, I would like to thank the organizers for the opportunity. I'm grateful. Being a Localite, it's a privilege and prestige for me to stand in front of this audience. And I have a lot of seniors in front of me. My research career is very small compared to the stalwarts here in the audience. But at the same time, I feel like I am equal like you all. So, am I audible?
So you would be session
Am I audible?
presentation to judges.
Okay. So, how much money will you spend on an average per month to kill mosquitoes?
ಆಮೇಲೆ
ಬೆಳಗ್ಗೆ ಸಂಜೆ ಮೂವತ್ತು ಅಲ್ವಾ? ಎಷ್ಟು ಖರ್ಚು ಮಾಡ್ತೀರಾ? I get paid to rear mosquitoes. I'm not joking. You can visit my lab at Bangalore. We are paid for rearing mosquitoes. Are you all with me? Okay. So let us start with some stories. Basically, if you do not understand, I'm thankful for something of sir's elaborate to talk on malaria work which sir has done. So that will make my presentation easier. Okay. So what is malaria? Vector-borne diseases. You should remember three things. One, man who is getting infected. A mosquito which transmits it. What is it transmitting? A parasite called plasmodium. Okay. So if you look these three things, now as I told, you will tell the story. How many of you cannot follow Kannada?
ಹಾಕಿದ್ವಿ ಯಾಕೆ ಆಗ್ತಿತ್ತು
All of us. ಕನ್ನಡ ಬಂದಿಲ್ಲ ಎಲ್ಲರಿಗೂ? ಸಾಹಿತ್ಯ ಓದೋದು ಕೇಳ್ತಾ ಇದೆಯಾ? I'm proud to stand in front of stalwarts who are working in Mysore University. Mysore University has produced the greatest of greatest writers only for Kannada. Okay. How many of you have read? See, this is one of the wonderful novel which is written. Starts with a character called Nai Gutti. Interval is with the Nai and ends with the again Gutti. Okay. So research is integrated with ಸಾಹಿತ್ಯ ಬಗ್ಗೆ ಈ ಪುಸ್ತಕದಲ್ಲಿ ಎಲ್ಲೋ ಮೆನ್ಷನ್ ಮಾಡ್ತಾರೆ ಕೊಯಿನ್ ಕೊಡ್ತಾರೆ ಅಂತ. There are narrations at least 4 to 5 times in this entire book. Coin ಕೊಡ್ತಾರೆ. This is somewhere around 60s or 70s book. Okay. What is coin? Whoever answers, I will take you to which of the hotel you ask for dinner today. Because I am confident you will not answer. Okay, shall we start? Are you all with me? See, but I don't know about you, but I keep forgetting. If you remind me at the end, I will answer this question. Okay? Okay, fine. Next slide. Okay, as I told you already, I believe I am a Gen Z person. But I got Kannada Sahitya anta gottilla andri. Who is she? Mike is there. Give somebody if somebody is answering. Who is she? So you do not belong to Gen Z. Or I am the outlier. Or she has become old enough. Who is she? Okay, whoever she is. Right. Maybe this audience doesn't like a beautiful women speak. I should have posted the other gender. Okay. I have posted for the chess part in this. This is one of the wonderful web series chess. Queen's Gambit. Okay, she is somebody called Taylor. If you get time. My interest is what is so beautiful about chess.
Search for this now.
See, chess is the only game where you know the number of blocks are fixed and number of pawns are fixed and the type of move is also fixed. But even then, the probability of outcome is somebody maths here. I learned maths 15 years back. Infinite probabilities. Okay. Even when there are fixed number of blocks, fixed number of pile, fixed type of movement, the outcomes can be of infinite type. So how it is linked with malaria? Again, the homework for you all. What is the homework? I will forget what you should do. See, if you are not responding, I will not move forward. I will, after 25th minute, I will say thank you and I will go. It's fine, sir. So what is it linked? How is it linked with the how chess is linked with malaria? So will you remind me at the end? So I have 2 homeworks for you already. Okay? Next slide. Okay. See, basically you have to know something about the mosquitoes. Previously, the classification of which mosquito causes malaria in which part was defined based upon its bionomics. Something called bionomics, where its habitat, where they breed, where they feed, and all those things. See, if you look at the map, it says when I was doing my MBBS, maybe in 2007-8, we were taught that Anopheles ulysses is a rural vector, which is commonly present in villages because of animal dwellings. And something called Anopheles stephensi, which is a urban vector. But do you think the rural-urban difference still exists in India? If I ask sir, what was the burden of Mysuru when he started his college days? People say Rajshekhara Hospital. What is there? Nagadi was a forest. Okay, but now till Vadodara you are getting 30-40 sold. As taken it's in Mysuru. Okay, so the transition has changed the dynamics of mosquito as well. I will tell you why it is important. Okay, and this is about the malaria of what are those primary vectors, secondary vectors, the parasite types. I think beyond the scope of this. My presentation I have tried to limit it to the spectrum of malaria and what, as a scientist from ICMR and representing a important institute from ICMR, what we are trying to do. Okay. How many of you know government of India is planning to eliminate malaria? By 20? Yeah, sir. Even to not in the net. 2030. Okay, that is the target. So when we say we are eliminating, we want to say there is no person is affected with malaria. Maybe you all are lucky now with the improved quality of living. Nobody might have been exposed to malaria, but previously, maybe a decade back, the malaria was highly prevalent, especially the sugarcane growing belt and where there is a lot of stagnation of water. Especially Mandya and all these places are endemic for malaria. Okay, but now due to change in habits, change of quality of living, and especially the good treatment available, how many of you have faced DDT spray? Sir, I think he's from the program and he has a lot of exposure. So they used to do spraying in all the endemic zones. I studied in a hostel which was endemic for falciparum malaria, and every year they used to spray DDT and it used to cause lot of nostril burn and epistaxis. Nostril bleeding is called epistaxis. It used to happen. But now because DDT is resistant, they have stopped spraying. The logic for spraying was previously because the house used to be dark, the mosquito after taking blood meal used to rest on the walls. But now mosquitoes have started waiting outside. The behavior of mosquito has changed from indoor waiting to outdoor waiting. Okay, that itself is causing elimination of malaria challenge. Okay, so this is about the cycle of the parasite, which you can. I don't think I want to go too deep into this. Next slide. See, as I told, in the entire spectrum of malaria, first is diagnosing. Our prime interest is to prevent any person getting affected with malaria, but if he is affected, to prevent his death. How many deaths occurred in Karnataka due to malaria? The deaths in malaria, Karnataka. So actually, the deaths have come down. Say, all over India, the deaths were reported only 84 last year. But not in Karnataka. Karnataka has only 4 places where malaria is still reporting: Bangalore, Udupi, KPJNL, Krishna Bhagya Jala Nigama. Okay, Upper Krishna Bank. So there is one particular mosquito which breeds only in the flowing river streams of water. So only in these 4 areas we have malaria in Karnataka, but there are places where the malaria is still a problem. Okay. If you remember, Kutturaman sir saying that the malaria is a big problem among pregnant women and under 5 children, because there will be a lot of death, especially in Africa, because of their exposure to thalassemia as well as sickle cell anemia. The probability of death is more common in African women. So what happens? Now you have got malaria. I have prescribed the treatment. Suppose if the treatment is working, what should happen to the person? The person should be cured, right? What will happen if the drug is not working? What will happen? If the treatment is not working, the disease will become serious and the person may go for cerebral malaria and die. Okay, so that's where the challenge of treatment failure. Next slide. See, as I told, when we are planning for malaria elimination, mosquito is a brilliant, very brilliant parasite, a vector. Okay. See, as I told in the first slide, in India the most common vectors are Anopheles faces and Anopheles stephensi. But in Africa, the most common mosquito causing malaria is MV, Anopheles MV. But what has happened in last 3 years? This mosquito called Anopheles stephensi has adopted the African environment and it is causing more problems. How do you pronounce this place? Jubati, Jubati. How do you pronounce? There is a city called Jubati. Okay, Dijubti is the spelling. If you, I don't know how you pronounce it. Nanbhasha Kannada. Okay, so you can pronounce it the way you want. Okay, Jubati had the maximum number of almost 43,000 cases of falciparum malaria. They were investigating what has changed in that place. When they found out, they found that this Anopheles stephensi has adapted to African environment. Next slide. The second part. Previously, the spray, DDT and other sprays used to work to control mosquito breeding. But now, most of these insecticides are from pyrethroid group. Most of these pyrethroids are resistant. Okay. Now, actually, biological methods are being tried to find out how to control the mosquito breeding. But all these sprays are, in some part or the most part, are not working. Why? Why resistance? Do you think only humans will adapt to, only humans will develop a method to control? Mosquitoes also have developed methods to resist the insecticide. Okay. Next slide. Now the other challenge. I told the treatment part. See, in globally, the treatment for malaria is something called ACT, artemisinin combined therapy. But now there are places in Africa where the artemisinin is not working. So what will happen if the treatment is not working? Number of cases will increase or decrease? Increases. It will increase or decrease? Increase. What if the mosquito adaptation increases? Number of cases will increase or decrease? Increase. Okay. So our aim is to reduce the burden by controlling both these aspects. To make sure that the person who is infected is treated as early as possible and the infectiveness of himself comes down so that the parasite load is less so that he will not become infected to others. Okay? Next. When the Africa started reporting the resistance, there are two combinations of ACT. One is called ACT-SP and one is called ACT-AL. Since northeastern part of India is very much close to the Cambodia, Myanmar and all those border and because of thick forest there, the resistance pattern was very high. So they approached the WHO told you have to change your treatment guidelines in the northeastern India. But ICMR did a study and we found out that ACT is still working in India and they told rather than ACT-SP, we can shift to ACT-AL, that is called lumefantrine. So how do you find out about resistance? See, the initial two talks was more about genes, enzymes and all those things. So in medical research also, we look into the research, the main important mutations will lead to resistance. So especially the PFK13, PFK, all these are enzymes. See, the parasite or the vector will develop an enzyme which will eat up either the insecticidal product or the drug product so that the drug is not working as well as insecticide is not working. Next slide. So how do we prevent? How many of you use mosquito nets? Dengue ಬಂದ ಮತ್ತೆ ಶುರು ಮಾಡಿದ್ದಾರೆ. One of the prime intervention for malaria control was LLI, long lasting insecticide treated nets. Okay. So but all these insecticides are pyrethroids. Now mosquito is resistant to pyrethroid, so it will not die. So there are researchers which are trying to prove what better, how better we can improve the LLI. Okay. Next slide. See, as a team member from ICMR and NIMR, we have collaborated with the Swiss Tropical and Ifakara Institute of Tanzania, and we have worked out this. This was a 5-year research which we did in Andhra district, where see, to do whether LLA, a mosquito net is working or not, they are supposed to distribute it to the people, ask them to use properly, and at the right time point, you collect it back and do testing of whether the chemical in that thread is still working or not. This is the first of the kind in the world where we have done this research and we have proven there is something called PPO, properly butyric acid. If you add that, even the pyrethroid resistant nets will work. And we have developed this, and actually WHO has asked for the raw data from us to make this a policy, saying that PPO nuts will prevent the mosquito bite. Okay, this is one of the contributions. This was published just last year. Next slide. This is what I was telling from Karnataka. As India is planning for elimination by 2030, in Karnataka they are planning by 2027, because the loads have come down, where we have published, we have worked out, and we have shown that how effectively the intervention works in Karnataka. This is published in Malaria Journal. This is what I was saying. Malaria is, see what is common in, if you look at this India map, you can fix that the area into two triangles. Right? One is in northeast, other is east central part of India. What is special about these two areas? Volcomundra. Are you awake? What is common between these two triangles? Say, what is common between those two triangles? Both of those zones are covered with thick forest. Okay. Actually, this is the map of my project which we are working. See, there are 52 identified endemic districts in India. Okay. Where GFADM is Global Fund Against AIDS, TB and Malaria, which funds heavily for control of malaria, TB and HIV. So we are working a project for them in all those places. And there are 52 districts where we are working to look what are the challenges in these areas, why still malaria is. Okay, this the protocol of the work which we are doing, we are published there. See, in exam marks are important. Research is measured in terms of quality publications. When I say we have published, it means our quality of work was good enough to be published. Okay, you A grade, B grade, C grade and so on. Our contribution, scientific contribution is based upon quality publication. Okay, so this is the exact zone where still malaria is existing. The biggest challenge is most of these places are very difficult to reach. If I plan a travel to any of these places, I cannot go in one day and come back. Especially Mysoreans are lucky. You Mysore na Sanjay Bejar is around Bangalore. Okay. Whereas these places is very difficult. See, one of the center, this was the first round, second round we are doing even in Gadchiroli. Remember Gadchiroli? Gadchiroli was infamous previously because of national issues. It's one of the border districts of Maharashtra, bordering Chhattisgarh. Okay. To reach, have you heard of Tadoba? Tadoba Kelidra? Tadoba is Kage. Have you heard of Tadoba? You do not know actually. You can't go to get Mysore ಹತ್ರ ಇದೆ ಅದನ್ನ ಹೇಳೋದು ಅಂತೀರಾ? Tadoba is a wonderful tiger reserve, very largest Chandipur forest tiger reserve is there. Actually, Gadchiroli is just 48 kilometers from Chandipur, Tadoba. Okay, so if you plan to visit Gadchiroli, you cannot visit in one day and come back. Okay, the access is the biggest challenge in these places, and we are working on those places. When we say we want to eliminate the disease, the most important is to look at the last mile coverage, where there is still the disease. So that's why Government of India and all the research institutes are investing in these places to take care, to tackle the challenges here. There's a place called Dhalai in Tripura district, Tripura state. There's a place called Dhalai. In the entire northeast, this one district contributes highest number of cases. If all the districts are counting 2,000, 3,000, Dhalai district counts in terms of 37,000, 38,000 malaria positive cases. They did lot of studies. You know the major challenge there is their main activities. There is something like June cultivation. There is a June cultivation geography. June cultivation kelidira. June cultivation is what they do cut forest, they do cultivation and leave the place and move on. To do June cultivation, they do not wear proper clothes. Okay. And whenever they go to field, they get mosquito bite, they get malaria. So most of the time in the entire India, Dhalai district will have the highest number of mosquitoes. Okay. So this is about resistant mutations which we have looked at. This is from my senior team group who worked on the plasmodium falciparum. See, the actual gold standard diagnostics is to collect the blood, create a slide and look at the microscope. But now there are something called RDTs, rapid diagnostic tests. Okay, that is based upon some enzyme called HRP gene. But the parasite is very clever. Now it is deleting the gene itself. Okay, so what will happen when you test if the parasite is there in your body and you are testing it by RDT, but the parasite does not have the enzyme, what will happen to your test? It will come positive or negative. Okay, so you will have more false negatives, in turn increasing the case. So that's what this study is. Next slide. I have four more slides. Okay, as I told, pyrethroids are resistant everywhere. Now that's why what they are doing is there is something called repurposing. So there are already so many pesticides which are used in agriculture field. So we are doing research on these pesticides and how they work on vector control. This is one of our studies we have submitted published, and actually this will play an important role in dengue control also. This spinosad is based upon something called spinosyn. Okay, it is derived from Actinomycetes. It prevents the mosquito to grow. Okay, so this has wonderful impact for all kind of mosquito-borne diseases. So this is something about the vaccine. You might have heard just 2-3 months back, two Indian scientists, two senior scientists, Dr. K.C. Pandey and Dr. Sushi, have developed a new malaria vaccine molecule. Both of them come from ICMR, one from my institute, one from Bhubaneswar, where they have developed a molecule. See, when we talk about malaria vaccine, it's a huge area. If you look at the blood stage, human stage, in human liver stage, and in mosquito stage also, there are multiple agencies which are working on different parts to generate vaccine for malaria. Okay? So this is about vaccine derivatives. Next. So what was the first homework? This was the second homework, right? Okay, so second, I will complete the second homework. See, even though you know what the parasite which causes, even though you know the vector which transmits the disease, even though you know what the treatment for malaria, but when we go locally, as I told you about the Dhalai issue, there are so many places like this. So this is not told by me, please correct it. It's somebody who has told in 1937. Okay, and and just read it for completion. Say everything about malaria is so dominant and altered by local conditions that it becomes 1,000 different diseases and epidemiological puzzle. Like chess, it is played with few pieces, but is capable of an infinite variety of situations. Okay, so when I say Karnataka, we are successful in every place, but not in Bangalore. Why? Bangalore has harbor. There are a lot of migrant people. If you visit the harbor, you can see a lot of young people from, you do not know which language they speak. Across the globe, they are there, they are working in the harbors. Okay, so it's very difficult to track them and treat them. Like if you go and visit that place, you will know they have this cycle of 21 days. They will go on a boat for fishing for 21 days, where they will pay, I think, per day 3 to 4,000 rupees. So 21 days they will be in a ship. After that, they will come back. So for this work, there will be a lot of rotation. Everybody is not interested to go in every cycle. Okay, this leads to a bigger challenge in Bangalore. So that's what I want to say. When you add the local challenges, it becomes a puzzle. And that's why, as I told and showed in the two triangles, there are places where still the malaria is challenge. Excellent. I think it's time to play. Shall I thank you all? This is in a nutshell what I can say about malaria. What is it? And if you have any questions, please. Yes. That is why COVID instances have come down. Malaria instances have come down. What is your opinion? As I already told, is that if COVID has affected two important diseases and two important programs, it was malaria and other was tuberculosis. Okay. Tuberculosis present is cough. COVID also presented in cough and fever. Fever is caused by malaria as well as COVID. And at the same time, to add to what sir told, it is very if not anything, COVID made one name very popular, one medicine very popular in the area of household. What is the drug? Dolo 650. Everybody knew Dolo 650. So that was altering people's presentation to malaria, sir. And I agree with whatever you told. Maybe that was the one reason why the reported cases of malaria were very less. So, why the hydroxychloroquine was more effective against COVID? Sir, this is very only 3 of the studies told that hydroxychloroquine was successful, but unfortunately, there are more adverse events due to hydroxychloroquine. It was stopped, and maybe the vaccine group was more stronger than the hydroxychloroquine group to communicate and say that if you give hydroxychloroquine, the people will be safe. But irrespective of this concept, which is documented across the globe, and especially even in India, hydroxychloroquine was used in many places. Maybe people
saved from malaria before they got the not COVID.
Sir, I am a scientist. I cannot say one thing today and change my words tomorrow.
You're all scientists.
So, and sir, I agree. Researchers, always the researchers' dilemma is, I can say what has happened. I cannot say this happened because of this or this will happen because of this. Exactly. Sir, I would like to add one concept in the same. What happens to a researcher? See, we did a BCG for COVID study. We are doing in 6 cities in India. In Kochi was one of the sites. Right? There was an apartment building. There were 16 flats in that building. One grand lady had she had registered and we had given her a BCG. The second wave, the entire building became positive, but only this grandmother was comfortable. And she called when she was scolding me. You know the treatment already, but you are not giving. See, as a researcher, I told madam it's at a research condition. It's my duty to document that it's working or not and report to the government of India and the policy makers. As a researcher, I cannot tell everyone, but actually that helped me to complete my study very quickly because I had more people registered into my study. Now, I request about this grandmom became my ambassador and I got all the 200 recruitment happening in 15 days. So this is the researcher's challenges. We can just say the research mode it works, sir.
It's actually the difficulty putting 1 and 2 or mutated. They cannot be used for diagnosis or diagnosis. What is the option? What is the choice?
Sir, one, they are working on the MDH part and they are the concern will come only when the deletion exceeds 5%.
So there is also for the analysis and the research. So we have some tests which we are willing to discuss with you in between. We will talk about it. Thank you very much.
And again, deletion is not much important, but even cross 2% we have done. Sir, thank you very much for enriching my
Thank you so much, Karen.
But the basic difference is malaria is a parasitic disease. Okay, parasite is very slow multiplier, it is lazy like me. Okay, it takes time. Whereas dengue is your viral, and in dengue also the problem is dengue has 4 brothers. Dengue 1, 2, 3, 4. If you are affected with one brother, it will not cause anything. You can have wild fever. But if one brother attacks the same house with other brothers, then the immunological reaction will cause the dengue problem. If you look at the spectrum of dengue, we have dengue fever DF, dengue hemorrhagic fever, where you will have bleeding points, DM, DHF, and DSS, dengue shock syndrome, where people need a lot of care to be treated for fluid care. So the major difference as is presentation and other important thing is that the transmission of dengue is by Aedes group of mosquitoes, whereas malaria is transmitted by Anopheles group of mosquitoes. Now there is a huge strong association of construction work, growth of Aedes group of mosquitoes, and higher dengue burden. Okay, whereas Anopheles group of mosquitoes are very clean. ನೀವು ಸ್ನಾನ ಮಾಡಿದ್ರೆ ಬೆಳಗ್ಗೆ ಎಲ್ಲರೂ ಮಾಡಿರ್ತೀರಿ. ನೀವು ನೀರು ಹೇಗಿದೆ ಅಂತ ಟೆಸ್ಟ್ ಮಾಡಿದ್ರಾ? ಅದ್ರ pH or calcium ಇದೆಯಲ್ವಾ ಅಂತ? As I told in the beginning, I get paid for rearing mosquitoes. ಹೌದಾ? Mosquito is so clever and so clean. If I increase the pH by 0.01, my entire batch of mosquitoes will die. I thought somebody last time you ಸೊಳ್ಳೆ ಯಾಕೆ ಸಾಕ್ತೀರಾ ಅಂತ. ಯಾಕೆ ಸಾಕ್ಬೋದು ಹೇಳಿ ಸೊಳ್ಳೆನ? I told you that the presentation which we have published, to test one mosquito net, I need almost 9,000 mosquitoes. So if I sell one mosquito by 100 rupees, I will be there near my market.
We are trying to control mosquito population. tolerance as far as their viability is concerned and also their ecological habitats are also concerned. And one of the methods that was tried over decades is sterilization technique, where the mosquitoes are sterilized, male mosquitoes are sterilized, and they are left into human system so that they will compete with the normal mosquitoes. And this kind of control process was very popular some decades ago. And ultimately, it so happened that 1 or 2 decades ago, it came down. That was not that popular. But again, we see that there is a surge in the technology to induce sterility in males and then release them into ecosystem. What is the current status now?
So thank you for bringing this question. Actually, one Australian group was very active in doing this research. They introduced this, and one of the PhD students from Bhubaneswar was also part of that. You might have heard of Olbachia. Olbachia is being added to that. See, the Latin American countries are affected with more physical challenges. So now the Australian group is trying to work in Brazil, sir, the basketball team from Brazil was working on that. So the challenge is how it affects non-native or non-target organisms. The important challenge which none of the research group are able to address. See, your concern is this mosquito, but what happens is several mosquito after taking care of it. So non-target organism issue is being, I think we can discuss in detail. The ethics issue of this is the non-target organism. That's why the research people are not very supportive about the biological control module. Thank you.
Excuse me, sir. Sir, I have one question. As you have heard on COVID vaccine, I have two small questions. One, whether this vaccine has any impact on bringing down the incidence of malaria. Secondly, sir, there are many notions about this COVID-19 vaccine. As a professional doctor, I'm asking you, sir, whether there are many negative impacts of this vaccine. They say clotting of blood and all this due to COVID vaccine.
Thank you for asking this question. So my honest answer, which I have answered in almost every speaker I have done. So that's why I have stopped writing COVID vaccine trial in my field. See, actually ICMR has done a huge study to prove whether the COVID vaccine is linked with the cardiac issues which are happening. Out of the way, what I want to say is there is no actual association, but COVID virus as such may have an impact because the cause of death in COVID was something called cytokine storm. If somebody is from immunology background, you will understand cytokines are actually protective mechanism in your body. But what happens if the protection is overprotective, it will attack your own lungs system and people will collapse. So the virus itself may have some immunological trigger. That's one aspect. Second aspect is for mathematical back of the hand calculations. In India, we have vaccinated more than 1.2 crore. Like 20 lakh people have not taken. Okay? 1.2 crore people have taken. Suppose if vaccine had impact of at least 1%, so physics maths people please tell me what is 1% of 1.2 crore? 120 crore. Sorry. What is 1% of 120 crore? 1.2 crores. 1.2 crores minus. No, this is just for clarification. See, the major challenge is, see, if you have the habit of reading books, you can read. Lot of books are there. See, the epidemic of cardiac deaths in US happened somewhere around late 60s, 70s, and 80s. This was 20 years after their point of view improved in 40s and 50s. Something similar, the quality of living, the food consumption, and our standard of living changed in the 30 years to 90. How much is that? So the cumulative behavioral aspect of individuals are one, heavy consumption of sugar, processed food, physical activity. COVID ಅಲ್ಲಿ ಮನೆಯಿಂದ ಆಚೆ ಬರಬೇಡಿ ಅಂದ್ರೆ ಎಲ್ಲರೂ ಬಿಸಿಲೇ ಇರ್ತೀವಿ. ಈಗ ಹೋಗ್ರಪ್ಪ ಆಚೆ ಅಂದ್ರೆ ಯಾರು ಹೋಗೋದಿಲ್ಲ. Physical activity and most important, stress. Madam, there is study is going on here where they are trying to find out. See how many of you switch off your mobile by 8 or 8:30 p.m. and sleep properly. How many of you switch off your phone at 10:30, 11:30, okay, 7:30, 8:30, 9:30? This is the problem. Quality sleep has a major impact on cardiac. Irrespective of the death which has happened, so many doctors also have died, and what they are trying to find out is quality of sleep. Quality of sleep has a lot of impact on your cardiac and mental health. I think we can have some decision on this. We can talk. Now there is a question. I cannot hear everybody. Come here, please. How India is going to identify by wonderful question. So what he is saying is when I say it's just how we are going to eliminate. As I told, we are reaching to those 52 districts where it is still existing. We are not going for one action plan fits all. We are going locally. We are addressing locally. Actually, I was in Delhi for a program yesterday and I landed only yesterday midnight, where we were developing a program for traditionally. Okay, so localized program for district specific intervention we are planning, and that's how we will be successful in the coming days in elimination. The definition of elimination is there should not be any indigenous cases. That means there should not be no local transmission for which government of India is investing in many parts. I think I have exceeded the session. We'll postpone. I am available till evening. We can discuss. Okay, if you have anything.
Thank you, Dr. Your knowledge and experience really needed. So I would normally request our chairs of the case community and talk with the master. Nearly 1 hour, we are very good and very informative talk about elimination of malaria from India and also from the world. It happens, it will be really interesting. Started with the life cycle of mosquitoes and after mosquitoes. He presented very nice interaction and also there with the delegates and also the students. So I, on behalf of the management and organizers of this today national program, thank you very much for the nice interactive session on elimination of malaria. Thank you so much. It is now time to express our gratitude to a small document. So I request Dr. Neda Fatima to come forward and appreciate our yes, with a memento and a token of gratitude for the I would also request Mr. Manjushri Ma'am to thank Dr. Neda Fatima with memento as a token of gratitude. I request Dr. Shweta Chori, Assistant Professor, Department of Biotechnology, to certificate the contesting us with the letter. Before our next, going on to our next session, which is presented by our resource person, Jalpi Koya, I would like to call upon Shama and Lavya to introduce the guest of the oral presentation, please. Good afternoon, everyone. I'm Monisha from the I would like to introduce our judges for today's oral presentation, which is Dr. D.R. Jagannath, who is a distinguished academic and researcher currently serving as a professor in the Department of Study in Botany and as director of the International Center at the University of Mysore. With over 25 years of experience in teaching and research, his expertise spans specialized fields such as mycology, microbiology, and molecular plant pathology. His academic foundation is exploring having secured first rank in course independent in botany and helped us in seed technology before earning a PhD in applied botany from the University of Mysore. Throughout his profile career, Professor Janardana has been recognized with numerous prestigious honors, including the Dr. Abdul Kalam Lifetime Achievement Award for his contributions to education and the Matsumi International Fellowship in Japan. He has successfully led multiple high-impact research projects funded by the major agencies like the UBC and DSP, focusing on areas such as microbial diversity and molecular diagnosis of plant diseases. Furthermore, he has mentored direct PhD students to completion and authored a vast array of research publications in renowned international journals, cementing his status as a leading authority in the world botanical sciences. I would like to call upon Ms. Anand Samin, HOD and Assistant Professor, Department of Microbiology, to welcome Sir Florence. It is my privilege to introduce Dr. Sukhman, who is currently serving as a professor in the Department of Studies in Medical Science at the University of Mysore. He completed his bachelor's from Governor College, University of Mysore in 1976, followed by an MSc in Zoology in 1978 and a PhD in Zoology in 1988 from the same university. His doctoral research focused on the economic approach to identifying high-yielding breeds of the silkworm or its molds. With over 3 decades of teaching and research experience, he has significantly contributed to postgraduate education and advanced research in cell culture. His major research interests include silkworm breeding and genetics, quantitative genetics, molecular genetics, aging studies using body coding and the body of animals, and the geotoxic effects of radiation and pesticides. He has received several national and international recognition, including the Commonwealth Academic Fellowship and the Dr. Leroy Merrill Award from the Zoological Society of India. Dr. Subramania has guided numerous PhD scholars, completed multiple funded research projects, and published extensively in reputed national and international journals, establishing himself as a distinguished expert in agricultural science and genetics. A hearty welcome to you, sir. I request Mr. Assistant Professor, Department of Microbiology, President Sir.
of you postdoctoral research at the Mason Eye Institute, University of Missouri, United States of America, who is investigating apoptotic changes during embryonic eye development. With around 20 years of research and 9 years of teaching experience, he is a recognized PhD guide under the University of Mysore and also serves as scientific advisor at Psychology Life Sciences and biomedical labs, Bengaluru. His expertise spans neurodegeneration, in vivo and in vitro toxicity models, Drosophila and rodent behavioral studies, dietary neuroprotection, and biochemical analysis. His research primarily explores the molecular and behavioral mechanisms underlying Parkinson's and Alzheimer's disease, with emphasis on biochemical and dietary neurotherapeutics. He has guided over 110 project students and currently supervises 3 PhD scholars. Dr. Chandran has authored international research papers, review articles, and book chapters, and has presented extensively at national and international conferences across Germany, South Korea, Austria, USA, Sweden, France, and Dubai. His work has earned multiple awards, including Best Poster and Platform Presentations, International Talent Grants from IDR and ICMR, and recognition at local neuroscience forums. He has also organized numerous workshops in Drosophila research, HPLC, biostatistics, and bioinformatics. We are very grateful to have you with us today, sir. I request Srimati Meghana, Head Department of Biochemistry, to welcome Sir Floridi. We are delighted to welcome Ms. Nirmal Grace S.R., Assistant Professor and Coordinator, Department of Studies in Biotechnology, SDRR Mahajana's First Grade College, Mysore. With over 90 years of experience in teaching and research, she holds MSc, MPhil, and PhD degrees and specializes in phytochemistry, enzyme kinetics, and nutrigenomics. She has published several research papers and guided many postgraduate students. In addition to her academic excellence, she has been actively involved in organizing workshops, conferences, and training programs, and plays a key role in institutional development through IUAC, NAC, and alumni coordination. Her dedication to mentoring young minds and promoting research culture makes her presence truly inspiring for all of us today. I would like to call upon Srimati Shweta, Head and Assistant Professor, Department of Biotechnology, to welcome Ms. Floridi. Once again, I heartily welcome all the judges of oral presentation today and all the best to all the participants. Thank you. Here is an announcement. The oral competition for UG and PG students will be held in the seminar hall first floor. I request for all of the UG and PG students to join in the seminar hall first floor. The research scholars and faculty's oral competition will be done post the session of Dr. Zan. Thank you.
Hey, the session start ಆಗುತ್ತಾ? ಎಲ್ಲಿದ್ದೀಗ? Wait, wait. You put ma'am.
I would like to welcome our first person for this session, Dr. Randeep Koya, who is Professor and Institute for Biomedical Science in Chandigarh University. I would like to call Mr. Deepak to welcome Dr. Randeep Koya to the stage. I would also like to welcome our third person, Mr. Deepak, Associate Professor, International Academy of Interactive Science, Mysore. Congratulations, sir. I request Srimati Rajarajeshwari ma'am to welcome Sir further. Now, I call upon psychiatrist Sir Day-1 to formally introduce our esteemed faculty. Mr. Deepak. It is my privilege to introduce Mr. Deepak Bhatt, our leader academician and experienced professional in the field of epidemiology and reproductive sciences. He holds a distinct microbiology from the University of Mysore and postgraduate diploma in clinical education and assistant reproductive technology from the University of Ireland. He is currently serving as an associate professor at the International Academy of Reproductive Sciences, Mysore, where he has played a significant role in shaping future professions. With over a decade of extensive hands-on experience in human embryology, his own expertise lies in ICSI and embryo. For the last 8 years, he has successfully trained more than 400 students, making a remarkable contribution to education and skill development in this specialized field. Mr. Deepak has also been a faculty member at the ACE Conference 2016 in Bengaluru and is a lifelong of ACE India, reflecting his active involvement in professional academic bodies. In addition to his academic excellence, he has proudly represented India in ultimate World Championships across multiple countries, highlighting his discipline, leadership, and dedication to your academics. Welcome, sir. Thank you. Thank you, Sarita, for introducing me. Now, I will request Mr. Deepak to introduce our esteemed Dr. I would request Ms. Srimati Rajarajeshwari ma'am to introduce the esteemed resource person, Dr. Good afternoon, everyone. Om Shri Mati Narayanama. It's my immense pleasure to introduce Dr. Jagdish Goya, Assistant Professor, FJ Research Institute for Biomedical Sciences, Goya is a biomedical scientist working in the area of hypoxia, signaling, and cancer biology and computational drug discovery. Sir has completed his PhD in biochemistry from Arnold University and is currently an assistant professor at HGM RITS HGM University, Mysore. His research mainly focuses focus on understanding how cells will respond to low oxygen conditions and how its process can be targeted for cancer treatment. And we work on mainly on H1F1 alpha study, PhD D2 and small molecules that can regulate oxygen sensory pathways in diseases such as cancer and stroke. He is the principal investigator of a 52 lakh research project which is funded by the ANRF from 2025 to 2028, aimed at developing new treatment strategies targeting oxygen-related pathways. Dr. Boyer has published more than 25 research papers in well-known international journals. His work was listed 323 citations, and he has an H index of 7, and he also serves as a peer reviewer for several reputed international journals, contributing to maintaining research quality history. He has extensively research experience in hypoxia signaling pathways and cancer biology, combining molecular biology, computational drug discovery, and experimental approaches with our innovative therapeutic strategies. We are happy to have you here, sir. We welcome you for the session and thanks for accepting my invitation to all you, sir.
Good evening, everyone. I am I'm basically scientific background and civil knowledge about this project combining these two research areas and I'm working on the object testing in the cell and basic conditions. Now, this is now, what is often connecting mean? Because we have targeted examples that means now that variable and rainbow and negative mode. Now, we compare these three data periods and parental measuring superiority by taking all from that we can go off the training, but it may last for 3 weeks, okay, not late. So it's the rainbow light, exactly it may last up to 15 minutes and any movement away almost 80 minutes. Now, what makes us able to resist or metabolically adapt to the low oxygenation? That really quite interesting because this is the total molecular metabolic alterations happen in the system actually. They get alive in the low oxygenation. For example, to understand these things, okay, we have to understand how cells are very low oxygenation. For that, we write your studies. After you actually we are starting at Thursday. Now, I can need to tell a story to student. Now, what to offer is actually main purpose of the energy and it has been spent without balancing the self-efficacy and even self-reliability. Now, this is actually two purposes. We have two enzymes, okay, which we call dioxin. Dioxin is nothing but is a molecule enzyme can directly used to place for the function instead of one oxygen atom, both the molecule and to the protein. Now, so they can observe the provide hydroxylase, provide protein hydroxylase, and back to the and then there is global protein and protein and take protein, take any electrons of it, provide hydroxylase, and other hydroxylase. These are few dioxins very popular. It changes the oxygen level in the cell and modify isotopic protein and protein source transformation and based on the oxygen level. Now, we come to dioxin. It is a class of what is called oxygen reduction enzyme. Now, it actually, as we were earlier, it actually incorporated up to atoms which is the inorganic substance. Now, in out of these 6 years, 5 years, we are going to run one person pathway for different pathways. Where the ACE or protein hydroxylase between E and N, which are regulating the histamine alpha or we call hypoxia chemical factors. Okay, we have little calories, little things. Now then regulated by the pH phenotype. Other things that we have either variant amino proteins which are used in our comment remodeling or direct gene manipulation and other things that we use. Now we just have all those regulated regulated by the one thing we made is metabolic pathway model, T3 cycle or T3 cycle stuff. We have intermediate metabolites, whether the first one was constantly like now, if you come to the how icons basically pathway or atmosphere work in human system. Example, human actually is a transcription factor. Okay, engaged by factor, D53 transcription protein, conventional OA kinase. There is induced by expression of the hexagonal. If the hexagonal is stable in the system, okay, you have to undergo hydroxylase where these layers are CH2 or 1 to 3. Then hydroxylase different answer and once the hydroxylase it recognizes by pH factor and it comes out to producible DNA. Now, it is low oxygen condition. Now, with low oxygen available for hydroxylase and methanol, where the pH is 2. So, the different frequency changes that undergoes translocation to the nucleus, then it enters the expression infinitely because the microbiome is by the DNA and the DNA. Now, do you have any role in nanotechnology? Basically, let development chemical we have projects which have been introduced by the network that is on those network that is also and also there is a basic now, one of the biggest problems in and we call cell adaptation with the reason adaptation with the primary lactate in pathway. All of this happens due to the cell in this country, till the COVID time, we have any of these things like cancer or polycythemia or anemia, chronic kidney disease and stroke and all these things are really and even COVID time people are active in and we are working in a one particular area called liquid smoke and because story in cancer, what happens is example we know that in the last year, those are called last year as per the figure. Now, what happens oxygen and the nutrition not reachable to the central point of human. Now, human being has the lack of oxygen nutrition kind of central point of temperature and being now, in that case, let's say that we have these particular limits, the pathway figure where we have point equation of variable level pathway where the variable and metastatic markers and DHM only and neurotransmitter screen problem. Now, these are things positive are things that we get adversity. Now, problem is here actually people are really working on cancer, but actually in not in alpha and post cancer, we are at both rather than target. So, emotions are in all kinds of related diseases. The cancer is only there's no oxygen to hydroxylate and intercellular disease gets triggered which is very pathways either called rubber headache or metastatic signal pathways all these. Now, we have one application called CHP. Now, this particular have as earlier. Now, we have three icons, one and two and three. Now, the one has to be the icon issue and these are second one that we have mind rating for most of the one and two. And these are fluid to most of the different. Now, if you look at the how it works, we look at the domain aspects of the ESP enzyme 1, 2, and 3, and all of them are equal, iron and equal 4 factors, along with 2 also equal to 8. And there are these two things actually big figure of protein, and people that actually key factors in taking that taking oxygen, iron, and throughout the meter or we call it intermittent. Now, in this left side, you can see that we how the economic development by these two, and the two side, one that we 564 of the provide or 413. These two combinations under and after that, the mark by this, eight factor and equal to now, people are working on thinking about the very long time, particularly and taking the local skills, who are moving around for equal people. And people look at the fact that the Indian people for people that for now, if you look at how people different proteins, they are alpha, two alpha, three alpha. Now, these are we call as oxidative protein. Now, this one is something because beta, two beta, beta. In fact, it's not different oxygen. It actually called beta, other oxygen protein. Now, if you see oxygen here alpha, 2 alpha, and 3, and the hydroxide PNG2 and PNG2. It is low this stable and alpha is neutral and activated most of the now, now, what does that tell you? Let's say one of the things there is a greater than the health activity. Wherever so one larger part is we talk about the most significant changes in medical treatment. Making the changes of the least takers to private, where least takers is not people. People are the new recognition of the species. I don't know the private and network. So we can not convert private instead of private into the that we call water. Now, what the problem here is one of the basic problem is basically any molecule, private into people, there is some high and even sometimes even also modified mitochondrial actually it is the chance of relation now we and even we have one more thing that is the finite object we have so all these things are actually relevant we are creating the spirit in this all these metrics developed by technology. So these things directly we may require these specific pathways or specific other things so far it's or a now what we do we have actually made this long protein level we read the publication by the book and published papers we have these molecular information and even now we are also we talk about the inhibition of PNG2 enzymes as well as the hormones peptide. It can help in the induction of the inflammatory pathway. It can beneficial for the patient or now, these are the publications we have published these were publication on these now, why we can talk about slope? Slope actually we know that due to the officers are being called the obstruction of the downfall. Now, there are two cases. One is the rate of related or post-treatment rate. Now, what are the benefits in this case people are going to get now? And actually, I would be talking about now we have looked at two aspects. iron as a cofactor. Okay. So if you talk with the high iron silicon, which has a direct contact, that is very beneficial. And another aspect of the hypoxia we can combine H2 so that it becomes stable in the cell and it can trigger the path. Now, we have in our institute master degree students, made with the project. Out of that, we can go to the one student has to work on that we have to do both as an individual for our and we use some particular operating strategies because there is no actually the bacteria can use it iron required for the pH to function proper activity. So if this pure point impact with pH iron, everything for the element for the pH level is reduced and it becomes inactive. This is one logic. And we see in the cerebral life, cerebral life, and we using glide and sodium and everything is normal time. And the thought made these studies are quite different than studies actually. The two different methods we follow are writing both utilizing and writing and from the university data. So starting and then we and this data is published in the secondary and so this is one thing we established actually one of the guidelines for actually and we have shown the very stable interaction now this is the same result we studied next week. It interacts with the pocket of the pH and it requires for the interaction with the IC and the activity. And this table represents the economic pathway in most cases there is a neural protection because as we know if there is a low high oxygen feature there is a repercussion injury may repercussion is the oxygen body is more the oxygen flow may damage the cell neural cells along the shock and experience. Next one. Now, one more thing that we instead of looking for the math is the cell must have some limitations. So, we follow the design global performance looking into the series data model. We should feed data from the very easy and separate and create a few molecules. Out of one molecule is selected and tested for the advanced experiment. We have selected one number for the area of people. Now, these concepts actually interact with the pH of 4 active sites and here is required that has been binding to the efficient energy and activity individual. Now, we tested idea. Now, we use we use with the many model analytics to use the neural science we control with the expression after that we treated with the increased number of people. Because we should not head off the level of substance scale, whereas the head off level is very lower in the input level of the field level. It is the second range and second level. So, and we tested artificial data actually looking for the size, and this actually, I try to come to the opportunity actually consider the most and where and most of the time especially and so what I'm trying to say is integrating knowledge of impact and may help in to address new matters and always moving hard work
I need my folder. Why do I forget that this exists? Delete tomorrow 1 second.
Auditorium.
What is presentation for academicians and research scholars will be held here in the auditorium. And I think I have probably given the course presentation course. The board has already been sent in the group, so I can according to that will be supporting here, and you will be disclosing your project and the name to be sending your ID. It has been sent in the group. Thank you.
I don't think you can use one. With that man, must be way.
ಒಂದು ಬೇಕಾ ಅಲ್ಲಿದೆ ಅಲ್ಲಿದೆ ಇದು ಆನ್ಲೈನ್ ಅಲ್ಲ ಅಂತ PPT ಓರಲ್ಲ ಆಫ್ಲೈನ್ ಅವ್ರು PPT ಕೊಡ್ತಾರೆ ನಮ್ಮ ಪರವಾಗಿಲ್ಲ 6 minutes ಬರೀ ಮಾಡ್ತಾರೆ ಐ ನೀಡ್ ಟು ಕನೆಕ್ಟ್ ಮೈ ಲ್ಯಾಪ್ಟಾಪ್ I have to connect. Take a print. You take this out. Bro. Online is also okay. ಈ ಕನೆಕ್ಟ್ ಮಾಡ್ಕೋ ಫಸ್ಟ್ ಈ ಕಡೆ ಇದೆ ಕನೆಕ್ಟ್ ಮಾಡ್ಕೋ ಇದು 5 + 1 6 + 1 6 + 1 ಪ್ರಾಬ್ಲಮ್ ಆಗುತ್ತೆ ಶೇರ್ ಆನ್ಲೈನ್ ಐಡಿ ಗೊತ್ತಾಗಿಲ್ಲ ಅಂದ್ರೆ ಆನ್ಲೈನ್ ಅದರಲ್ಲಿ ಇದೆಯಾ ಒಂಚೂರು ಹೇಳ್ತಾ ಇದ್ದಾರೆ ನನಗೆ
Time 4 + 4 6 + 1.
Good afternoon, everyone. My code is AC01. Today I am presenting effects of malnutrition on oral cavity. And this is Pallavi Amma, working as an assistant teacher of science and Samarya Central School, Bangalore. Objectives of my paper: find the nutrition affects oral health in several ways. Malnutrition adversely affects the oral structures, and poor oral health in turn leads to poor nutrition, also known as malnutrition. Thus, malnutrition affects the development of the oral cavity and the progression of the oral disease. Coming to what is malnutrition, before that, we should know the difference between what is diet and what is nutrition. Diet is food what we eat, but nutrition, it is the biochemical and physiological process of consuming food for the support of life, growth, and development. Good nutrition is a intake of food in a required nutrient quantity and is a stepping each step along with the physical activity. Coming to malnutrition, malnutrition is a condition resulting due to the excess intake of nutrients or due to very poor intake of nutrients. And malnutrition which leads to improper mental as well as the physical growth, reduces immunity, and also it reduces susceptibility to fight against infections. Yes, malnutrition is a threat for global health or oral health, and the cause for malnutrition is mainly aggravated by overpopulation, increased family size, gender inequality, and
due to adverse cultural practices and even due to large population in certain countries. This is a table showing the effects of malnutrition on oral cavity, deficiency of many micronutrients, vitamins and minerals, and protein energy malnutrition, which adversely affects the oral cavity. Protein energy malnutrition leads to delayed tooth reaction, reduced tooth size, and also it decreases enamel solubility, salivary gland dysfunction, and deficiency of micronutrients like vitamins A, B, C, and vitamin B complexes, and deficiency of iron, which leads to salivary gland dysfunction, angular, and many other oral diseases. I apologize for the vague pictures, but these pictures clearly indicate that oral cavity is adversely affected by malnutrition. Deficiency of vitamin A, D, and protein energy malnutrition, which severely affects the oral cavity, and it defects and it causes the oral defects as well as oral structure malfunction. And in case of oral defects, enamel hypoplasia and hypomineralization malnutrition. Enamel hypoplasia is a condition is formed due to the lesions where it is characterized by the fix, hypofix on the enamel of the tooth structure, and there is an appearance of the longitudinal fix on the tooth structure. Increased enamel hypoplasia in turn leads to hypomineralization, and hypomineralization increases susceptibility to demineralization, which creates a keratogenic environment. Keratogenic means where there is diminution of the inorganic substances on the structural part of the tooth. And demineralization means there is a decrease in the solubility of the calcium hydroxyapatite, which is formed in the outer structure of the tooth due to the production of more number of organic substances. Along with keratogenesis, enamel hypoplasia, it also leads to vitamin C deficiency disease scurvy, which in turn causes halitosis, battery, painful ulcers in the mouth, then ulcerative gingivitis, fissures in the tongue, and angular chelosis. So the corners of the mouth, you can see that there is a red inflammation, and it may take longer period of time, more than days to years if it is untreated. Coming to the discussion, many studies have shown that malnutrition is a multifactorial disease which can happen early also during the intra-children's life or during childhood. And it has also been shown that due to the deficiency of micronutrients as well as the minerals, there decreases the protective effect of saliva on the oral cavity. And even many studies have been shown that the mineral calcium is plays an important role for the stronger enamel on the teeth. So deficiency of calcium, which in turn affects the density of the alveolar bones, which gives a strong support to the teeth. And even vitamin C deficiency diseases can also lead to many of the periodontal diseases. In order to prevent malnutrition, one must take in consideration that a proper explanation given to the breastfeeding mothers after 1 month delivery, and especially 6 months of breastfeeding must be given to the mothers with a clear explanation. And along with that, most of the non-conventional ways of communicating organize like short videos or games so that it can reach to each and every people. And many of the Indian government organizations programs like ICDS, Integrated Child Developmental Services, and also NIDDCD, National Iron Deficiency Disorder Control Programs, and being various services in order to control the malnutrition. Along with these initiatives have to be taken in order to maintain our oral health, proper certifications must be done with the dentists. These are my conclusions. Thank you.
Sir. ಅದು ಪಿನ್ ಮಾಡಬೇಕು.
Really respected witness, really Devina. I am Arpita, research scholar from Department of Biotechnology, University of Mysore. My ID is RCOC1. Today I am speaking here titled Observed Orientation of Superprocessed Using Sustainable Agriculture. Let me begin with brief background problem. Antibiotics are considered as one of the major refractory emerging pollutants that are primarily discharged from the pharmaceutical industry. However, the persistent in the environment even at a trace concentration, but they develop significant risk by proliferating the antibiotic resistance. There are many conventional techniques which are costly and also the energy intensive. This has motivated our study to develop the efficient and sustainable observatory is known as biochar. Among the biochar, the nanometer attracted attention due to its high surface area to the mass ratio, enhanced activity, and also the presence of functional group, which is very helpful in the removal of the protect from the aqueous environment. In my study, I selected 6,000 antibiotic, which is a broad spectrum immunomodulator antibiotic. Objectives of my study is pilot synthesis of the nanoparticle which is covered by the naturalization to optimize the novel approach for the removal of this processing from the aqueous environment into the sustainable nanoparticle and evaluate its degradation using the HPLC and also the FTIR and assess the reusability of the nanoparticle. Methodology, collection of the waste. Here I selected the waste species of mass which was pre-dried and subjected to the local surface for the fire at 3 different temperatures, 300 degrees Celsius, 550 degrees Celsius and 900 degrees Celsius. And then in a separate batch after the cooling, the nanoparticle is plastic dried, ultrasonicated and centrifuged, which is called as various characterization. And also
also the absorbing and because pH is the temperature and also the concentrate and to assess the reusability. And also we have done the comparative test with the FTI and HPLC. We also carried out absorption kinetics and also the isotops. Absorption kinetics is used to determine how fast the reaction is happening. Either physical construct or the zero second or the elevation or the determine the diffusion and limit the diffusion. And also how is the nature of the absorption. Whether it is a monolayer or multi-layer, more in case of particular cases. So we are conducting macular studies and testing and PR model. Development description. First, heat is very important. Here we can see the heat of the nanoparticle decreasing markedly with the increase in the temperature. You can see here at 300 degrees Celsius because of the 47.5% and 12.4% at 550 degrees Celsius and 2.5% at 900 degrees Celsius. Next, characterization. Here we are conducting energy dispersed to exo spectroscopy to determine the elemental composition. As compared to 300 and 900, at 550 nanometer, the carbon content is very high. FTIR, carried out FTIR, to determine the surface functional variation which is influenced by the pyrolysis temperature. We can see here FTIR 300 degrees Celsius, OH, identification of the alcohol, stretching calculation of the carboxylic acid, and here the water conjugated carboxylic acid. But in 550 there is the absence of functional group, there is no presence of organic functional group. And also we are conducting dynamic light scattering to determine the hydrodynamic size distribution. So hydrodynamic size distribution of 300 was found to be 60 to 80 nanometer and 550 was about 500 nanometer. Next, data analysis was carried out for the 300 degrees Celsius. The particle size distribution was found to be 73.6 nanometer and the cycle was heat has. And the X-ray spectra, we also carried out X-ray. You can see here what T theta value 22.7 degrees. This is the characteristics of amount of carbon and also zeta potential. Zeta potential is one of the important parameters to determine the collider stability and also the surface gap. 300 degrees Celsius nanoparticle has a zeta potential of -48 millivolt. Next, we carried out linear element weather surface analysis to determine the pore surface area outside of the total pore volume and it also exhibited type 4 isotope is known. Here the pore morphology is bigger pores. Next, the absorption experiment. What we also obtain is the initial concentration of the concentrate and also the absorbent. Here, after the optimization, a selected 0.5 gram per liter of the nanoparticle, which carried out the absorption maintained over time and also the absorbent required. Next, also the study effect of 8%. In my study, neutron pH I got the maximum absorption and also maximum remote efficiency because at neutron pH, superprocessing occurs as a interionic form and also the nanoparticle has the surface area negative charge which decreases the electrostatic temperature and increases the electrostatic attraction. Next, temperature. Here the moderate temperature, 28 degrees Celsius to 35 degrees Celsius, I got good remote efficiency. And also the effect of contact time. In real base phosphate system, the effect of contact between the solution and absorbent is very important. So, we can observe that in the initiate there is a rapid increase in the remote efficiency because there is a presence of active sites of the nanoparticle. Next, also we access the reusability test. Reusability test is the first cycle is moderate and the second there is a increase in the remote because of the complete saturation of the active sites and also the photovoltaic. This is the HPLC control. You can see this. This is the control system that is degraded also. You can see here this is in the peak area and also the reduction is about 19.16%. And FTIR is controlled by using FTIR also. You can see the control FTIR spectra and the degraded FTIR spectra. And the absorption kinetics. Here all the kinetics we have studied, but our experiment pattern is better used for the super models where the absorption is getting up. And absorption isotope, natural isotope model is better prepared. So we found that our nanoparticle surface is monolinear and the polymer absorbed as monolinear. So conclusion, sustainable nanoparticle effectively removes the superabsorption from aqueous solution. Absorption was strongly influences the pH temperature in contact and kinetics isotope is not affecting. So this is the effects. So I express my sincere gratitude to our guide, Professor Gitana and co-host Chandrika, and also the Department of Biotechnology, University of Mysore, providing me the facilities, and also the financial support by the officer, Secretary of University of Mysore. Thank you. Good afternoon, everyone. My code is RSOP02. My name is Madhumita Rajan, a research scholar in Division of Molecular Biology, GSW of Mexico. And today here I am to present on a very beautiful, wonderful topic, which is micronutrients with polycystic ovarian syndrome. So I would like to start with a quote that the doctor of the future will give no medicine, but will invest his patients in the care of the human brain in diet and in the cause and prevention of disease. This was followed by Thomas Edison. So basically, this future, we can see this future as current right now. So because I'm going to tell you under the basis of molecular network that is happening in PCOS. And I consider PCOS as a silent epidemic of us women. So PCOS. PCOS is generally we all regard it as irregular menstrual cycle or weight gain. But that's not the only case in PCOS. Because there is an entire layer of molecular underpinnings that happen in PCOS. And that's the reason why we call it as a molecular endocrine disorder. And right now, I can tell that even in this scenario, we might have 1 in 5 Indian women who have PCOS, and I'm sure that most of our symptoms are not diagnosed yet. That's how serious this silent epidemic is. And why micronutrients only? Though the name micro, it plays a major role in the human body. Because, for example, let's keep our body as a high performance engine. And micronutrients are oils or the spark plugs that are needed for the engine to work. So if our spark plugs or the oil does not support our performance in your figure of the engine. So there are major pathways that are disrupted in PCOS. We have PI3K, which is an insulin resistance, NRF2, which is an antioxidant, and NRF3, which is an anti-inflammation. So basically, all of these are involved with each and every micronutrient that we see, which we speak further. So my objective was to be the micronutrients and to highlight the mechanistic and clinical evidence that evaluates potential and enzyme management. So basically, my methodology, this is a narrative review. So we went through a lot of content and scholars and we did this analysis. So starting with the results, vitamin D, most of the results papers that I went through, the major deficiency that we see was vitamin D. Why vitamin D? Because it acts as a hormone. It's a steroid, which means it acts as a steroid in the body. And as we know, steroid release is one of the major pathways in PCOS that is disrupted. And that is why vitamin D also actually pre-beta HSG, which is nothing but important for progesterone. So higher the progesterone, lower the androgen because PCOS has hyperandrogenism. And usually in PCOS patients, we see low progesterone and deficiency of vitamin D is caused out. Now moving on to B vitamins, which are always spoken about in almost every disorder. They do of course have a reason because they are very essential cofactors for one carbon metabolism and mainly ADA mechanism and for homocysteine. Because the higher homocysteine, as we know, cardiovascular risk hormone, and cardiovascular risk has been found in 1 of 5 patients in PCOS. And this is a research that has been and vitamin E and C are antioxidant defense mechanisms that is important for all the ROS, that is reactive oxygen species. So these vitamins protect us against these cell death because granulosa cells go
importance of molecular analysis that will get developed in PCOS. And hence, vitamin E and C play an important role. And as you can see, all the pathways are interconnected. And they are the micronutrients are also interconnected in one or the other way, either directly or indirectly. Now, moving on to the minerals play a very crucial role because magnesium is one of the most common minerals found to be deficient in PCOS because magnesium helps in blood flow transportation, which is for glucose transportation between cells. Hence, what is insulin resistance? Because insulin resistance is one of the major things that are seen in every PCOS patient, that is what the insulin resistance talks about. And since it's important to manage it, this is something that every PCOS woman knows. Hair is like armpits are low, even in the face, mustache and all of that is low. And hence one is needed. Selenium is important for enough science and chromium is again important for insulin receptors. And hence we can see the link of insulin signaling playing a major role in this. So summary of deficiencies, when I went through RCTs and different research papers, I found that the most deficiencies are already vitamin D and magnesium. It is also, we are not sure if it is because of the PCOS that we are getting this deficiency or because of the deficiency do we have PCOS. That is a question of research yet. So vitamin D, magnesium are the major. And these are the clinical and experimental evidence that are mentioned as such. So vitamin D and B complexes have been most high scientific evidence. Coming to the discussion and conclusion, basically, as I said, we don't know the cause of PCOS. It is not a disease. Disease always has a reason, disorder does not have a reason. So we don't know if micronutrients can cure PCOS or can they help us. But I'm pretty sure that micronutrients, not just micronutrients, nutrients also can help us with therapeutic outcomes. And so coming to the conclusion, all these pathways play a major role in PCOS, and as I stand here at SDM Women's College, it's very important that we empower women, and most of us, I'm sure, we would be going through symptoms of PCOS that is still undiagnosed. And nutrition is something that is in our control, in our hand. So if we can modify it before we get PCOS, I think we can achieve a breakthrough and get against the science. And this is a paper, the thing of what I said is actually a paper that was recently published in one year and over the next. So if more information can come on. Thank you. Let's open the
Good evening, everyone. My name is Amrita. I'm a research scholar from Day-1 University. So today my topic is microbial consumption-based nanopriming as a sustainable strategy for enhancing tree growth and yield. So basically, as we see, chili is a major imported crop, and also the productivity is often constrained by several problems, including the delayed germination and also the poor seeding development. But we are depending excessively on chemical fertilizers, so that it is also inherently decreasing the yield of the chili. So we want somewhat sustainable and eco-friendly management strategy in order for the enhancement of the chili yield and growth. So that's why we are approaching microbial conversion-based nanopriming technology as a promising technology in order to solve these problems. However, there are some very limited studies on microbial consumption-based nanopriming. And also, few people are done only on the microbial consumption and few people are done only on the nanopriming technology. But my research is basically on including both the microbial consumption mediated and also using the nanotechnology as a part of it. And also, there is is a lack of integrated evaluation from safety regard to EV. There are only some EV studies has been taken place. EV studies have been taken to greenhouse conditions. And also, there is a need for somewhat eco-friendly nano-renewable approaches in agriculture. These are gas and form. The main objectives of my study is to develop the microbial conversion based on zinc oxide nanoparticle. First one, next evaluate the germination and CPD level chili, and the third one to assess the growth and the yield under the greenhouse conditions. So basically, first I have isolated the bacteria from the residual phase profile of the chili. So I have got the Bacillus anthracis and the Bacillus subtilis as two major organisms. And after that, I have prepared a movement block media and the continuous stable concentration of microbenzoate and native Anthracia by including these two organisms. Once we got the conceptual approach for the zinc oxide nanoparticles preparation. So these are the materials and methods I have followed. So centrifugation, supernatant collection, addition of the zinc oxide then again by adjustment of pH. Then after that, the dryness is Celsius, I want to install the nanoparticles. So in order to confirm it more, I proceeded for the characterization of that. So I first moved for the individual spectroscopy. So the absorption rate required is 7.5 nanometer. It's confirmed the synthesis of the zinc oxide nanoparticles. Then I proceeded for the FDIR. So in FDIR also the broad field spectrum I observed around 426 nanometer, which is the major characteristic of the synthesis of the zinc oxide nanoparticles. Then I proceeded for the cell. In cell also I observed the surface morphology of both the control and also the microbial things, the zinc oxide nanoparticles, which further confirms the presence of the zinc oxide nanoparticles. Then X-ray diffraction spectroscopy, also the broad field where we are observed 37.5 degree centimeter confirms the presence of the zinc oxide nanoparticles. Then when I did the EVS also, I observed the presence of the zinc and oxygen as the engineer composition properties, which confirms the zinc oxide synthesis. Then zeta potential also further it enhances the stability of the zinc oxide nanoparticles and also the charges which is present. So all these characterization finally concluded the conclusion of the synthesis of the zinc oxide nanoparticles. So once we got the zinc oxide nanoparticles, then I moved to the nanoparticle technology. So I improved the zinc oxide, then I did the nanoparticle suspension, then I formed the series and I formed the human nanoparticle suspension. Then I moved to the two methods of follow. One was the blotter release method in order to control the germination percentage, and after very I got the results like this. Second method is followed by the paper cutter method, and also to check the nanoparticle seeds whether it is performing well in the germination or not. I proceeded for this method. Then I get over the series, movement, tuning, and also grinding and threshold. Then I proceeded for the greenhouse studies. So the nanoparticle seeds I have used and also the control I have grown. So these are the images of the greenhouse studies I have conducted. Up to 3 months I have performed this. Then we have some of the experiment regularly. So these are the results. So what all happened? When we are talking to the control, in all the studies I have conducted, be it to be greenhouse conditions, the germination percentages seriously increased in the zinc oxide nanoparticle when compared to the control, and also the germination rate has been increased, mean growth time has also increased in the zinc oxide nanoparticle when compared to the control, and also when we did the paper around the kind of water release method. So out of the 100 seeds also, I have got 87 seeds germinated in the zinc oxide. And when I compared to the research instruments with tertiary dryness, in tertiary development nanoparticles, even mostly very high proportionally compared to the control. So these are the yield I have got for the first, second, and third harvest. So when the particular control, the zebras and animals fully engaged in the meal also is confirmed. So these research concluded that zebras and animals is one of the basically sustainable and eco-friendly management strategy in order to increase the yield and also growth parameters in the tree. Thank you.
How many seeds you have tested? How many seeds you have tested for germination and future?
So I have taken 100 seeds and I have taken the recommendations.
You are doing research. Are you doing research for PhD?
Yes.
You know how many seeds need to be tested for taking the germination percentage? Minimum how many seeds should be tested?
Sir, when I have gone through the survey, sir, there was
International Seed Testing Association, ISTA. It has fixed minimum 400 seeds you need to test. Then only you can results can be analyzed. Minimum 400.
I will do that.
As per ISTA.
Thank you. My name is Iris Teachers, and I'm here to present my paper title, which is Neurological Activity in Human Language in Non-Human Animals and Applications. So, I don't want to repeat the second slide because the second slide is about my title and meaning. So, more to the first slide. So, in the introduction, I wanted to let everyone know why I chose this title. It is because the complexity of the speech in human and animal languages. We are very interested and a lot of researchers are working into the language composition of humans, but not in animals. It is very less and we need to give a side to the animal and language education also. So basically, human speech is a multi-layered system where acoustic, phonic, and prosodic rhythm and emotional balance are interconnected. And when it comes to the animals, the acoustic phonetics are found and the emotional balance are found more than prosodic. And in the intentional stance, recent evidence suggests that non-human animals, such as NHAs, adopt an intentional stance, attributing to social need to human vocal signals. And now I am here to see the cross-species sensitivity is not merely an artifact for the domestication, but it is rooted in the phylogenetically preserved neural mechanisms. The objective of my study is to synthesize the neuroimaging and electrophysiological data regarding the non-human animal responses to the human vocal system. So that to evaluate the shape in neural circuitry, the hypothesis was fixed with a pacemaker line that checking animal utilizes homologous brain regions, that is temporal voice areas for the electrospecific organization. And the third objective is to explore the impact of audio motor learning on the top human vocal synchronization. And the methodology part, we use the keywords of cross-species communication, temporal voice areas, and neurography. In methodology focus, we had the functional MRI, EEG with the delta and theta retrospectivity, and the behavioral application, this application. And phylogenetic scopes were interested in the breeding lineages, especially the macaques and the chimpanzees, and the domestic species like cats, dogs, and horses. Synthesized finding the focus in the sensitivity of phonetic boundaries and prosodic emotional cues. Phonetic boundaries in the sense the way the animals intake verbal cues from the human, and the emotional cues are like the facial expressions and the tones and emotions. And I wanted to know about if everyone knows the brain has two responds more strongly to the vocalization and non-vocalization environment. According to the dog directed speech, human sensitivity slows their speech rate when talking to animals from between the 4 and approximately 3 syllables per second to match the animal's slower neural network, thereby improving the communication. So it shows that the animals can almost relate the words quickly. They are not just listening to the sound, but they can realize the words we are speaking with the language comprehension, which is also related to the EEG study where they explain about the EEG code. And coming to the comparative social cognition, emotional balance is found in that, and if the domestication has fine-tuned ability and reward system that is amygdala and ventral striatum of dogs and horses also to human emotional process. And in the interspecific co-regulation, proximity to the human localization can modulate the HPA axis, that is stress response, hypothalamic pituitary adrenal axis in the dogs, and functioning as a physiological. And together in the discussion, I would like to know about the neural continuity and the symbolic cognition. Actually, the ability to extract identity and emotions from the voice, like we predict the human language, and originates in the pre-Kurosawa network shaped with the common ancestors of the humans. And the emotions are involved in humans as well as in animals, which is also common. And the symbolic boundaries is that while non-human animals exercise the acoustic categorization, that is where they can listen to sound and they can categorize it. For example, if we call a cat with the same name repeatedly, after a few days we can record the meaning, we can understand the sound, acoustic pattern after this, where we can understand the frequency. When you start saying in a few days, and then you start filming in a few days. So brain is talking, and at one point you start the terms filming, they can understand. They are more advanced than they can relate this vocabulary, sounds and frequencies comparatively to the human. And in conclusion, the cross-species communication is facilitated by the shared temporal channel, where the human adjusts their production to keep the animal perspective. Human sensitivity is a part of both evolutionary conservation, like a shared auditory habit, and interspecific adaptation, that is co-evaluationary community. And the future research should focus on the cellular and level network changes induced by the long-term human animal social bonding. And we personally encourage the researchers to also work on the emotional and EEG part of the animal language acquisition. And these are the references. And this is what my slide speaks about. In the one line, we would like to say that communication becomes a shared temporal channel for both animals and humans. Thank you. Room 1406.
Myself, LLC, I am affected in IBS administrator down here in University. So I'm here to talk about our tracking also called the elastic effect. So when you come to the structural elastic effect, so it is
is hard as it is in say. It's somewhat looks like a language structure. They do like so that it is supposed to somewhat looks like no, it's like so like this. So it's not the experiment and the to and the exact sequence and thing. So then we need to look at the four aspects of this the one is the animal activity, and then chemical activity is the test method, and then exactly the secondary and property of the last grade, we will use PHS and property. And then the anti-inflammatory and then anti-inflammatory activities. The methods and method follow. So for the anti-inflammatory assay, that will be is the bacteria culture and then is the treatment and then treatment as prepared as a control is where we control the any underlying molecule and then we will take the distance phase over the plane, the spread phase, the actual spread plane, and then we will integrate the overland for and then the result is room and perform the spread out. So then when it comes to if radical can be assessed. So in respect to pressure, we are integrating the ethanol in the reduced will be the population of the then the CCA directed the compound accepted directed and then pH and then integrated the temperature the work and temperature and then the evaluate results can be versus so then the other test is concerned with antioxidant property of the very reducing antioxidant. So here also it is the fact reagent is called chloride and enzyme and add compound to various concentration and then incubate the measure the solvents at 550 and so anti-hemolytic assay. So prior to that, this assay is for to tell about anti-hemolytic. So I have an example, and then what is the previous and compare the 2% and then by adding the hydrogen peroxide and create this recovery works as a control and then integrate and measure the observation 5.8 and then calculate the inclusion that is of maximally inclusion concentration so perform it certificate and then the last report the antioxidants, the exactly original responsiveness is what is on the nucleic acid. It will be converted into some other derivatives with the production of hydrogen peroxide. So, and integrate with the reference standard is the one which is it was against the responsiveness, the enzyme. So, and it is considered as a standard. And then we have both the intervals and the plastic. So these are the results what the say the aspect showed a larger inclusion zones than the diversity. In order to be at 100 microgram per ml, 19 is seen. When it happens with the epsilon this is what zone. This is so these are all the zones
that material and material as an opportunity. So in the basic so this will be accepted executed over compared to the standard. So for this, there need to be an alternative mode of nanoparticles introduced into the rely on majorly about on the plant extract. Plant products are among the major soil-borne pathogens, in particular in Meladonium genus, especially Meladonium invicta is one of the major species in this genus which restricting crop yield as well as the quality in majority of the agricultural particles of crops. Chemical nematicides, there are many chemical nematicides to control these types of soil-borne pathogens, but they have very large effects on the environment as well as the health risk. So for this, there needs to be an alternative which is eco-friendly and cost effective. So the mineral respiratory life cycle of Meladonium invicta which is being used in this study. The plant order of Zeylanicus belongs to the family Annonaceae, which is a medicinally important plant and also have rich in bioactive compounds that is to make suitable for the nanoparticles in this. These are the objectives which involves the synthesis of simple nanoparticles using the leaf and root extracts from Macropolis mechanisms, along with their characterization using UV, visible spectroscopy, FAR, XRD, and film analysis. So, along with this, I also aim to evaluate the resynthesized nanoparticles for their nanoparticle activity in vitro conditions, along with their ultraspectral changes that is to be caused upon treating with the food non-animal technology. So this is the methodology I followed. I collected the plant parts and made an extract, and using that extract I synthesized the nanoparticles. Upon their color, visual color change, they have been characterized for UV, FAR, XRD, and cell. Along with that, I made anti-mitochondrial activity and the in vitro condition by in vitro human mortality as well as the cat inhibition, and then I studied the ultraspectral changes of the treated egg and the skin. Results showed a visual color change of the nanoparticles which confirms this of silver nanoparticles and is confirmed by using the spectroscopy where stored a sharp peak at 400 to 500 nanometer range. The FAR spectrum showed the involvement of phytochemicals that is being involved in the reduction and encapsulation of silver ions that confirms the synthesis of silver nanoparticles. The film analysis also showed the presence of spherical morphology of the silver nanoparticles along with their lambda size. Under the in vitro activity, nanoparticle activity, negative inhibition is showed, which is equal to a particular functional nanoparticle with lower level in the higher concentration, which about 95% of inhibition is being observed. And in the zonal modality also we used to see the increased zonal modality upon increase in the concentration of silver nanoparticles, which is time dependent, where I used to see 48 hours apart, 48 hours of treating with equal zonal modality with that of the commercial chemical nanoparticles. So this is the images taken under compound microscope where you can see the A and C are the ECMOS X and units of the Meladonium invicta, which has anatomical alterations in them, and B and D are the light nematodes which have which has been like a palm and eggs will be and I used to study to penetrate. This is what the important one. through which we understand the mode of action of the silver nanoparticles in meeting with the eggnomes, where I see the silver information and their structural changes in the eggnomes inside where they once made up the immune being inside the egg, which has entered and then we also see the presence of nanoparticles on the eggnomes. Green synthesis is successful in adopting hand extracts and also showed a great mantle activity and also the study of the mode of action. What I get the result in higher concentration. So in nanoparticles will get you at 90%. Number of units is 400% modality.
My colleagues Alex, Oki, Jeremy, and Bhavani, and I'm from CSP, CSR India in Mysore. I would like to thank the organizers for giving this opportunity to present my answer. My topic is morphological and biological analysis of various molecular genomes. White writing in different economic conditions. Like the only source that is used for the cultivation for cotton production is very possible boundary. So nursery belongs to the family Moraceae and genus Flora. It is called as cultivar because every each and every part of the plant will be used. So mainly the leaf has been used for the cotton production. Still, each and every part, though the bark are being used for furniture and the roots are roots now actually latex of the leaves also has been used for the production of girls and also the latex and some parts of the bark they are used for expressions and celluloid production. Then apart from that, maybe now they are concentrating fruit because fruits are rich in bioactive compounds and are high in nutritional value and plays major role in the human health. And they can due to their natural sweetness, they have been used in jam, jellies, juice production and also wine production. My objective is to evaluate the high-yielding fruit varieties and also to compare the biological profiles across the various fruit types. These are the 8 varieties have been taken, and they are three Indonesia, Fiji, Saraswati estate, Rambari, Modified, Maranadi, Morocco. So these are the 8 varieties I have identified, and the fruits have been harvested periodically for the analysis of biochemical activity. the biochemical analysis, protein oil method, carbohydrate, Dobian control method, TPC for HCl calorimetric method, total polyamide aluminum chloride method, Scott iodine method, ascorbic acid vitamin C, that is 2,6 dichloroquinone, isoquinone, that is 5% for antioxidant DD, EHHA method, and for reducing sugar, BNS. So next, the graphical representation of the morphological and biochemical parameters are below. So first we look for, here we can see the highest number bearing plant is mathematics, that is 224 numbers of fruits per plant, that is the highest recorded. Then the fruit weight is 4.528, that is also mathematics. Next is movement. The highest movement is recorded 7.1 in Anamali variety. The fruit width of highest width 1.2 centimeters. Going for the fruit, here I have been extracting the fruit extract from the fruit and directly the all the fruit extracts contain some acidic method. Highest is 0.3% in brown variety. And in EC it is 0.52 in master variety. And coming to biochemical analysis, for protein it may be estimated because brown variety is highest content protein, that is 95.73 grams per gram of food. The carbohydrate of 36.99 milligrams per gram of fresh protein. Leonard, that is highest was found in 8.57 in DRB variety and Leonard 7.35 in Malus. Starch 8.65 in DRB variety and reducing sugar of 18.56 milligrams per gram in Malus. Ascorbic acid which was 4.49 highest in Malus variety for 85 milligrams and antioxidant in mulberries are highly rich in antioxidant properties. They are showing above 60%. Each and every are above like more than 75% of the foods are showing highly antioxidant. If you include mulberry variety, the mulberry fruit Indian variety, you can increase your antioxidant content in your. So I would like to conclude that the mulberry fruits in these 8 varieties have found DRC, homeopathy, and maramari showing highest range in all the biochemical content. And these fruits can be utilized into DRC, homeopathy, Good evening. Myself Sujana, and I am presenting on the topic effect of abundant feedback on phytochemical retention and antioxidant activities in pain and anti-flower plant extract. My code is RS40 on. It is well known that sustained anti-flower is very fast and consumed as a premium vegetable in India. Due to the many phytochemicals present in it, its
with this background, the present study was designed with the following objectives: to evaluate the effect of drying method on phytochemical retention, to compare solvent efficiency in extraction, and to assess the antioxidant activity. To fresh leaves and flowers were the product was locally used to the center, and it was taken to drying method, particularly outside the clean drying. These freeze-dried and outside samples were then subjected to solvent extraction using with ethanol method, using ultrasound extraction. These extracts were then subjected to phytochemical analysis and antioxidant assays. With respect to CPC or phenolic content, freeze-dried flower extracts showed markedly higher content rather than otherwise flower extracts. However, in these, the outside leaf extracts, that is with the ethanol and methanol, were comparatively higher compared to freeze-dried leaf extracts. This could be due to the heat-induced destruction of the cellular matrices, which enhanced the release of bound phenolics, which improved the extractability. Our time can use the other types of processing methods. This is moderate. This can weaken the phenolic cell wall interaction, which increases the availability of the extractable phenolic compounds. Coming to flavonoids, the highest content was seen in freeze-dried flour methanol extracts. This would be because of the superior preservation of the heat-sensitive flavonoids, whereas in other-type leaf extracts, it has when compared to the other-type leaf extracts, this would be because of the difference in the leaf and flower structure, so there is a difference in their texture. Moving on to tannins, freeze-dried better preserve the tannins in the leaf. This could be due to the reduction in polymerization and enzymatic oxidation, particularly by polyphenol oxidase, which release of the strong tannins. Whereas flowers different phenolic profile and delicate tissue structure, there is a pronounced advantage of over advantage. Freeze-dried leaf extract and freeze-dried flower extract exhibition markedly higher alcohol content compared to outside leaf extracts and outside flower extracts, with complete loss of observed outside flower extracts. So this will be because of the loss of these components in higher temperature. With respect to alkaloids, since they were heat sensitive, they were better preserved in low temperature vacuum conditions of the freeze-dried. Coming to antioxidant assays, in this case, that was measured the IC50 or radical challenging activity. It shows that in this freeze-dried flour and methanol extract, activity between 150 years is desirable. So that was observed in freeze-dried flour extract. Similarly, in contrast, the outside leaf extracts preferably have better challenging than freeze-dried leaf extract. It usually reduces the drying on antioxidant. So, these and markedly enhance the increasing antioxidant activity in flower, and we use a better sensitivity to flavonoids and alkaloids. This improvement of restorative yield suggests that these substances are concentrated on these stages. Classified also follows a similar line of CPS, implying that freeze-dried enhances the antioxidant potential, particularly in the flower tissues. We can establish a strong positive relationship between demand and plant values, polyphenol and CPS cavity, alkaloid presence and enhanced antioxidant forms. This also suggests that flavor and alkaloids may significantly contribute to antioxidant activity. So, if we can freeze-dried increasingly improve the phytochemical potential, especially in the flower extract. Then flower tissues are more than these. Ethanolic extracts are better, and antioxidant activity is strongly associated Day-1 and 12%. And the results are demonstrated in the I can send my results for the assessment. My resolution activity is
but electronics causes both different toxicity in silicon, leading to increase in mortality, growth inhibition, reduce the silicon protein content, and significant decline in thermal and cell parameters. Use of electronic is in semiconductor must be carefully regulated to prevent economic losses and ensure sustainable production. These are some references. As you know, agriculture is at present in our country is providing employment for more than 1 crore population. At the same basis, today, agriculture also has so many problems. But nowadays, we are focusing on the very high technology oriented products, but we are not focusing on very much low organization, create an entrepreneurship in the rural level without any knowledge. So this is one of the which I am going to say that using the normal biogas which is produced by our agricultural waste, we will use this working approach for entrepreneurship. So you can see, in particularly we are going to a 100-year phase of trade, we will have to use 1% age of memory, which produce 10 age of so we can use this based on the working approach production. And this is the base level formula where we can manage the eco-friendly aspect, and by this we can find only polygon fertilizer. So this is the so the relevance of current issues. We all know naturally produce probably use Day-1 in the field having 16 hectares each by hectare per year. And we also have 14 hectares of shoe per hectare per year. Yes, in that where we can see the ratio from the bed waste we are going through, we can get up to 600 to 700, 650 metric tons of waste, biomass waste every year per hectare. So these hectares will now, due to the highly usable content of multiple issues, farmers can decide into composting method or composting method decide what they are burning, and composting of shoes takes more than 1 year. So this is where the farmers are not using it back to their field. This is a point where we can use reverse reverse digestion microbes, and we can make it a commercial model and use some scientific techniques and the other microbes, so we can digest it earlier and we can go for the commercial production. So for that, as an entrepreneur, you can select 20 to 20 farmers and you can collect the waste from the agricultural funds, and you can do like a centralized way. In that way, you don't need to use so much of the high science, you can just manage and you can produce. Same how I can do that. These are some supporting documents or review of literature where we can use it for the byproducts. Our segregation management. Next. So, see, segregation compost drop here. You can see there are details of that. And like this is regarding the economic of composting and segregation waste, how we can be using it. And project also, we as agriculture needs to make more than 20 metric tons of compost per our own product. So our farmers who is having more than 2 acres are using that 20 metric tons of their own production, and they are selling. They are also getting more than 5,000 per year. It is like after that consuming, they are making still it is in the profitable levels. A person who has don't want to the farmers also can produce it in an entrepreneur. This is where you can see because identification of agriculture waste. Because people don't get that. Reason is that Malwa is a high biomass producing plant, and we grow across the area, and no other plant can come or no other crop can compete with Malwa in the biomass production. So people don't know that those are the same thing. Lack of standards protocol, because since it is a every some other work is making a own work for us, again performance to our own work for us. So we need to make the country protocol right, because we need to digest the resource which is facility schools. And insufficient capitalization and the scarcity of environmental values and economic discipline. Next. So after the study, next. The necessity of producing communities governmental society runs to become organized to the world running campus and running age beyond the production. Solution design and biological integrated communities social economic impact and environmental opportunity. So for the one campus preparation, you don't need to use any rocket science. Same production of how every other of our life, every other base what we are using the body campus is basically. The only thing is that our mentioned some methods and some of the materials we need to use it in a different manner. So that can be very different to take this. Next. Then secondly, working on now in the alignment program industry. Per plant they are using approximately 3 to 5 years of working cost. Our working cost is represented. But that much amount of working cost is not being supplied to any farmers, so they are going for the chemical inputs. So we can target by using our waste, we can target the crops like plantation crop like economic crop like coconut and arega. At the same time, horticultural crops also need the high inputs of biofuels or working compost and working cost. This can be managed. So by that we can cut down the chemical inputs. So the compost preparation is also same. There is nothing next. So this is the ways to make product. This is a prototype of how we can make this place and we need to we need to produce your own working compost. So anyhow, so this is how to start is when we have just a issue economic standards we have made this start. So where it is the land we are going for a lease with establishment before establishment also between the basic cost we have made a economic. So when some of the using spending some extra money we can make a use of it where the only money should be we can cover. So if you buy one to another investment you can go more than if you are investing 1 lakh after 2 to 3 years you can change from 1 lakh to 50 lakh. So, establishment of a sustainable commercial working compost unit using multiple ways of raw material, generation of additional income for entrepreneurs, consumers, and farmers, reduction in open culture of number waste, and improved environmental quality, production of high quality, value-added working compost storage to meet market demand, creation of rural employment and entrepreneurship opportunity, generation of private sector, and most important, circular economy and sustainable agriculture practices where we can cut out and use for complete roads and for agriculture. So, next. The regulation where we can use, as I said, for other case for the working compost production and compost preparation, and we can by this we can minimize it and we can reduce the burning of the waste. We can stop this in our polymers waste and we can take it as a model for experimental we can follow it. And we can produce it, we can make our we can make a sustainable agriculture.
ಡಿಸ್ಪ್ಲೇ ಆಗ್ತಾ ಇದೆ.
ಏನೋ
ಡಿಸ್ಪ್ಲೇ
ಆಗ್ಲಿ ಆಗ್ಲಿ.
ಏನೋ
ಏನ್ ಮಾಡುದು?
ಏನೋ
ಮಾತಾಡೋದೆಲ್ಲ ಹೋಗಿದೆ. ಎಲ್ಲ ಜಾಯಿನ್ ಆಗಿರ್ತಾರೆ ಕೈಂಡ್ಲಿ ಅದೇ ಭೂಮಿ ಹೇಳ್ತಾಳಲ್ಲ ಹಾಗೆ. ಬೇಡ ಬೇಡ.
ವಿಡಿಯೋ ಡಿಸ್ಪ್ಲೇ ಆಗಲ್ಲ. ವಿಡಿಯೋ ಡಿಸ್ಪ್ಲೇ ಆಗಲ್ಲ. ಕ್ಯಾಮ್ರಾ ಇದು ಕ್ಯೂಸ್ ಆಗ್ಬಿಟ್ಟೈತೆ. ಇದು ಯೂಟ್ಯೂಬಲ್ಲಿ ಲೈವ್ ಆಗ್ತಿರುತ್ತೆ. ಅದು ಕೇಳಿಸುತ್ತೆ. ಮಾತಾಡ್ಬೋದು. ಮೂರು ಜನ ಬಂದಿದ್ದಾರೆ. ನೀವು ಮಾತಾಡ್ಬೋದು.
ಇನ್ನೊಂದು ಬಂದಿದೆ, ಇನ್ನೊಂದು ಬಂದಿದೆ.
ಆಟೋ ಅಡ್ಮಿಟ್ ಇದೆ.
4 ಜನ 3 ಜನ
ಇನ್ನಿಬ್ರು ಬರ್ಬೇಕು. ಈಗ ಇದ್ದಾರಾ? ಸ್ಟಾರ್ಟ್ ವಿತ್ ಮೇಘರಾಜ್.
ಹಾಂ 5 ಜನ ಬಂದರು.
ಓಕೆ ಓಕೆ ಬೈಕ್ ಕರೆಕ್ಟ್ ಇದ್ದಾರೆ.
ಇದು ಸೇರ ಇದಾಯ್ತು. ಡಬಲ್ ಬರ್ಬೇಕಾ?
ಫಸ್ಟ್ ಮೇಘರಾಜ್ ಡೈರೆಕ್ಟ್ಲಿ ಯು ಸ್ಪೀಕ್ ದಟ್. ಮೇಘರಾಜ್ ಕೋಡೇನು?
Maker Shivraj, ACOP02. Please tell your code and start your presentation.
Hello.
Yes, you are audible.
You're audible. Yes, yes.
Okay, okay, thank you. Wait a minute.
Please tell your code to the judges and start your presentation.
Sure. Good evening, everyone. So my code number is ACOP02. So my research project on the topic isolation, screening and characterization of peroxidesis from algae and its degradative potential on multiple substrates under the guidance of Dr. Pratana Ji, Associate Professor, HOD, Department of PG Studies and Research in Biotechnology, SDM PG Center,
Excuse
Ujjivan.
me, please start with your objective. No need of introduction.
Sure. So my research on the objective of to screen the freshwater bodies for the prevalence of potential algal enzyme for detoxification and to investigate oxidoreductase enzyme activity from the isolated stream and to test the crude enzyme from multi-substrate specificity to carry out the optimization of enzyme activity for a varying pH, temperature, and metal ion concentration, and to detect the structural changes in persistent organic pollutants by the enzyme using the FTIR analysis. So these are the review literature, the research which is already done on the in this field, that is on the lactase enzyme from the different source by the Shaler Valizid and the Oriol Baltron, who done the experiment on the polycyclic aromatic hydrocarbons. So my research is started with a collection of water samples from the different region, Belipalti and the Beltangadi region. And after collecting the samples, we are cultured on the sterile bowls basal media. The ingredients are given in the slide. And after the culture, the sample taken for a microscopic observation by the hanging drop method. So we are taken this method that is for a better study about the morphology and motility behavior of algal cells. And after that, we are done a test for multi-substrate specificity and for determination of peroxidase activity. So here we are taken the two substrate. One is TMB hydrogen peroxide, that is tetramethylbenzidine hydrogen peroxide substrate. So here we are taken on microtiter plate, the 20 microliter of the crude enzyme added with a 10 microliter of the substrate, incubated in the dark for 15 minutes and taken absorbance at 460 nanometer. And if it is given in a blue, it develops a blue color, then it is indicates the peroxidase activity on the substrate. And the second substrate is ABTS. So ABTS reacts with a peroxidase enzyme and it give a green color. For this, we are taken the 100 microliter of 50 millimolar sodium citrate buffer and 40 microliter of 1.2 millimolar of ABTS substrate and 40 microliter of the culture supernatant, that's the crude enzyme. And for a peroxygenases activity, we added 20 microliter of 10 millimolar of hydrogen peroxide and they taken for 20 minutes of room temperature as incubation and measured the absorbance at 480 nanometer. And after the test for multi-substrate specificity, we done the estimation of the amount of enzymes present over the crude extract. So for that, we are taken biuret test. So here we are taken the standard from 0 to 1 ml and 0.5 ml of the sample with 4 isolates, which shows a higher enzyme activity, and they were taken with the 1 ml of distilled water with 3 ml of the biuret reagent, and they are incubated at 37 degrees Celsius for 10 minutes. And we taken the absorbance at measured at 540 nanometer and we plotted a standard curve over there. And optimization of the peroxidase activity, it's mainly done for three influencing factors: temperature, pH, and metal ion concentration. So here we have taken 20 microliter of the crude enzyme extract added with 10 microliter of the TMB substrate, and they were taken in a different temperatures such as 25 degrees Celsius, 27, 37, and 42 degrees Celsius. And after the end of the incubation, we done the absorbance was measured at 460 nanometer. And for the pH, we done a single standard buffer solution was prepared of the pH of 3, 4, 5, 6, 7, 8, and 9 and adjusted with the help of the dilute HCl and NaOH solution. And we are uses of pH conforming by using pH indicator. Here also we are taken the 20 microliter of the crude enzyme extract with a 10 microliter of the TMB substrate, and after the incubation we taken the absorbance measured at 460 nanometer, and last optimization done for the metal ion concentration. So here we have taken the different effect of the metal ions such as magnesium, manganese, iron, and the zinc in the different concentrations such as 1 millimolar, 2 millimolar, 3 and 4 millimolar, and the same procedure were followed here. And at last, we are done the FTR analysis, that is to detect any structural changes happen for persistent organic pollutant when the screwed enzyme act over there. So for that, we are taken the three persistent organic pollutants such as chlorpyrifos, naphthalene, and our ABTS as a substrate. So first, we are taken a 3 cover slip. Over there, we added a layer by layer of this 3 persistent organic pollutant, and again in 3 cover slip, we added the same substrate. Over there we added the crude enzymes and we given a test to a research center in Dharwad for a FTR analysis. So in the result, after the culturing the algae, it shows a green coloration and turbidity, which is suggests there will active photosynthetic activity on the algal cells. And after that observation on the microscope, it shows a green coloration and indicating the chlorophyll A and B having a bonnet, and they have a no flagella, so it suggested that they comes under the chlorella genus. And for the test of multi-substrate specificity, out of the 10 isolates, they choose 4 isolates having the higher enzyme activity in the algal samples. And this is the graph plotted over the biurate method. So the standard curve giving a standard equation. By substituting over there, we get the value of 0.0672 milligram per ml. This is the enzyme amount present over the sample. And after optimization, we get to know that enzyme having a maximum activity at a pH of 6 and the temperature 37 degrees Celsius. And they have an optimal concentration where having iron is more than the manganese, magnesium, and the zinc. And the
Your time is
hello.
up.
Okay.
Please conclude.
So these are the FTI analysis graph that's before and after degradation, and in the graph it shows that there will be a degradation and breaking of the carbon double bond carbon, and that shows that the enzyme actually degrading the persistent organic pollutant. So overall, the study demonstrated the crude peroxidase enzyme. They are derived of freshwater algae possess significant catalytic potential to oxidize and to degrade the multiple types of chemical pollutants. And this research is lays a groundwork for the future studies involving the scaling up for potential integration into our wastewater treatment system, targeting persistent organic contaminants. So these are the reference. Thank you.
Thank you. The next participant, Shashidhar P. Shashidhar P. Let's move on to Saujanya N. Please mention your code and start presenting.
Good evening, ma'am. My code is RSOP 13. Is my voice audible, ma'am?
You are audible. Please start presenting.
Good evening to one and all present here. This is Saujanya, research scholar under the guidance of Dr. Ashwini, Associate Professor and Head, Department of Microbiology, Jain University, deemed to be university. The title of the study is Biofunctional Attributes of Dairy-Derived Probiotic Lactic Acid Bacteria with the Anti-Cancer Potential. Coming to the introduction, as we all know that raw milk are a complex biological secretion which has a very good nutritional
Sorry for the
content
interruption.
and beneficial. Yes, ma'am.
Saujanya, sorry for the
Yes, ma'am.
interruption. Can you just minimize the Google Meet box that you have opened?
Minimize.
It is blocking your presentation.
Yes, ma'am.
Minimize it. Pull it down. And please start from your objective. No need of introduction.
Yes. Yes, ma'am. Yes, ma'am. So the present study aims to isolate, characterize, and identify lactic acid bacteria from dairy and their products. The study also explores the probiotic properties, their safety analysis, and investigates the functional application of partially purified metabolites with the application of antibiofilm assay and anti-proliferatory activity in leukemia cell lines. My slide is actually not moving, ma'am. Yeah. Coming to the methodology, the isolation of lactic acid bacteria from the dairy and its products, the biochemical characterization, safety evaluation, probiotic characteristics, the partially purification of lactic acid bacteria, and finally the application. Coming to the isolation part, where MRSA agar has been used, that are supplemented with calcium carbonate, where 8 dairy and their product samples have been collected. Around 37 bacterial isolates were identified morphologically and also based on the zone that are created by calcium carbonate, and our subculture has a pure colonies, and further biochemical test has been conducted. All 37 isolates were identified as gram positive rods under the light microscope. Out of 37, 27 has been tested negative for the catalase test, and which was further taken for the study because the lactic acid, it is one of the features of lactic acid bacteria. The litmus milk test has been conducted where there is where the observation of color change, gas production, and milk coagulation were observed in comparison with the control. 14 exhibited positive results for litmus milk test. All 14 isolates were tested for sugar fermentation test, that is glucose, lactose, mannitol, and sucrose, and observed for the acid and gas production, and which are mainly species specific. Coming to the safety analysis, out of 14, 9 isolates showed or exhibited gamma hemolysis. Rest all 5 isolate exhibited alpha hemolysis, which were excluded from the study. Coming to the antibiotic sensitivity test, this was carried out using disc diffusion method, and the antibiotics which were taken based on the guidelines to CLSI 2020, out of 9 isolates, 7 isolates were susceptible for all the antibiotics which have been tested, especially SMA9, the first one, the Aramark one, which is isolated from sheep milk, has shown us susceptible to all the antibiotics which have been tested. And these are the pictures of the antibiotic susceptibility method of the selected isolates against the antibiotics which are used. Coming to the probiotic characteristics, the first one is the bile tolerance, where 0.3% of concentration of bile salt is very necessary and sufficient for assessing the bile salt tolerance for a specific strain, where at the different time intervals like 0, 3 hours, 4 hours, and 5 hours, and 24 hours was tested. The graph here representing is 24 hours incubation, where all the isolates were tested for the bile salt, and the percentage showed here is 63% to 100%, where the maximum which is being shown is SMA9 that has been isolated from sheep milk. Nath et al. 2020 has the same study, isolated Lactobacillus plantarum from raw milk, has exhibited 83% at 4th hour. Coming to the acid tolerance test, where it is very important for the probiotic organism to tolerate to the acidic condition, which is the pH lesser than or equal to 3. And here all the isolates were tested for the acidic pH of 3, where the maximum which has been showed by SMA9 and the percentage ranged from 66 to 87% of acid tolerance by all the isolate. The same in the same study at a pH 2.5, Zhang et al. 2020 paper, he showed that Lactobacillus plantarum, which was isolated from sheep milk, and also Lactobacillus brevis has shown 92 to 90%. Coming to the phenol tolerance, it is very important for the lactic acid bacteria to tolerate the phenol, as these are toxic and it can reduce the activity of the organism. Even here at the 24th hour of incubation, SMA9 showed the maximum tolerance towards the phenol. And coming to the Zhang et al. 2022 paper, where Lactobacillus plantarum shows the 88% after 24 hours, and it is slightly lower than our study. Coming to the auto aggregation, the one of the important factor for a probiotic bacteria, that is to ability to adhere to the mucosal lining, where we have tested all the
Excuse me, your
isolates.
time is up.
Okay,
I
ma'am.
request
I'll
you
just,
to conclude.
yeah. So where SMA9 has been done, co-aggregation, the same way SMA9 has shown the maximum ability against E. coli, Pseudomonas bacillus, and Staphylococcus aureus. Even the cell surface hydrophobicity, where SMA9 has showed a maximum. Coming to the partial purification, where the metabolites were purified from the cell-free supernatant using 70% ammonium sulfate, that is followed by dialysis, where the dialysis has shown 8.8 fold purification level, and from the gel filtration chromatography, Luena et al. 2023 showed 3.15 fold purification. Antibiofilm activity that has been tested for SMA9 isolate against E. coli, Pseudomonas, and S. aureus. It is very important for a metabolite, that is to inhibit or eradicate the formation of pre-existing microbial biofilms that inhibit or reduce the biofilm associated resistance, where the maximum results were showed. Coming to the last, anti-proliferatory activity, where the metabolites which I have used has been inhibited proliferation of the leukemia cells, that is MV411. The IC50 value is 3.675 microliter.
I request you to
And
start presenting.
yes, ma'am. And the cell death is 70% when it is very more value compared to the positive control ciplactin. Coming to the conclusion, the study demonstrates a strong biofunctional potential, and among 37, SMA9, which has been isolated from sheep milk, has a superior safety, the tolerance towards all, and the antibiofilm, anti-proliferatory activity, and their cytotoxic against the leukemia cells highlights its potential. These functions will support underutilized dairy sources as a valuable reservoirs of the potent probiotic strains for functional food and biotherapeutic application. These are my references. Thank you.
Thank you, Saujanya. Next,
Yes, ma'am.
I would like to call Namira.
Good evening, ma'am.
One second.
Please mention your.
Good evening, ma'am. Shall I continue? Hello.
Yes, you are audible. Please mention your code and start presenting.
My code is RSOP14.
Please start presenting.
Yes, ma'am. I'm trying.
Namira,
Good
please start
evening, everyone.
from the objectives. Skip the introduction.
All right, ma'am. Okay, ma'am. Good evening, everyone. I'm Namira Khan, research scholar in the Department of Microbiology and Botany under the guidance of Dr. Muhammad Aman from Jain University. My area of research focuses on antifungal or antivirulence potential of Senegalia catechu, which is khair or kach tree, and Ocimum gratissimum, which is clove basil or ram tulsi, against multi-drug resistant candida species. So before I begin, I would like to share some insights related to my topic. While many believe that prevention is better than cure, the reality is that numerous healthy individuals are carriers of multi-drug resistant candida species. Did you know that around 75% of women will experience a candida infection in their lifetime, yet many may carry multi-drug resistant strains without even knowing it? So imagine a world where a simple yeast infection could become a life-threatening condition. Moving forward, I'm not able to shift my slide, ma'am.
Click on save a copy and then start presenting. There's this option called save a copy.
Yes, ma'am, I'm checking.
Save the copy and start presenting.
Okay.
Just above your cursor, there's an option
Yes.
called save a copy. Can you see that?
Yes, ma'am, I'm doing, but it is not happening. Ah. Is it moving, ma'am?
Could you repeat?
Is it moving? My slide is moving.
No, it is not moving. Click on the slideshow button next to the zoom.
Yes, ma'am, I'm trying. Uh,
Ma'am, kindly log off. I mean, stop sharing. Then again you share, ma'am.
All right, sir.
Hello. Hello. ಎಲ್ಲಿದ್ದೀರಾ? ಅಂಟಿಸ್ತಿದ್ದಿಲ್ಲ ಅನ್ಸುತ್ತಾ? ಅದೇ ನಂಬರ್ಗಳು.
It is not moving.
Namira, unless and until you keep that option save a copy, it's showing their upload blocked anta. You have to click on that save a copy. Then it will be available for you to
Okay, sir, I will stop sharing and I'll again do that, save a copy, and then I will do, sir.
Okay. Narmada?
Yes, sir, yes.
Narmada, not Nimira. Namira, you can present at the last.
Yes, okay, sir.
Yes, sir.
Narmada, can you please...
Sir, it is done. Can I continue?
Sorry?
It is done. I saved a copy.
Okay. Yeah.
Can I try it once?
Yeah.
So I tried. It is not moving. I'll present last, sir.
Okay, Narmada.
Yes, ma'am.
Narmada, please.
Yes, ma'am.
Please mention your code and start presenting.
Okay.
Narmada?
Yes, ma'am.
Click on the button next to the zoom option. In the below bar that you are seeing next to notes, there are 4 options, right?
Yes, ma'am. Yes, ma'am.
Not there. Below that. Can you see accessibility?
ಕೆಳಗೆ zoom out zoom in ಮಾಡ್ತೀವಲ್ಲಪ್ಪ ಅಲ್ಲೊಂದಿದೆ ನೋಡಿ ಲೆಫ್ಟ್ ಅಲ್ಲಿ ಇದೆ ನೋಡಿ
ಅಲ್ಲೇ ಕೆಳಗೆ
ಕೆಳಗೆ ಬನ್ನಿ ಡೆಡ್ ಎಂಡ್ ಬನ್ನಿ ಇಲ್ಲಿ zoom in zoom out ಮಾಡ್ತೀರಲ್ಲ
ವಿಡಿಯೋ ಆನ್ ಮಾಡ್ಬಿಟ್ಟು ತೋರಿಸಿ
ಡೈರೆಕ್ಟ್ ಇಂಟ್ರೊಡಕ್ಷನ್ ಇಂದ ಸ್ಟಾರ್ಟ್ ಮಾಡಿ
Okay. My code is PGOP6.
Okay, my code is PGOP6. I am Narmada from MSc Biotechnology STM PG Center, Ujre. Under the guidance of Dr. Pratanaji, Head of the Department, PG Studies and Research in Biotechnology. My topic is
Please
exploring
mention your
agro-based
code, Narmada.
degraded PGOP6 month. Exploring the agro-based degradative ability of cellulose produced from isolated soil screened bacteria and its optimization studies. These are my topic. Uh, Objectives are first we collect the soil samples from agricultural fields and then isolate and screening of the cellular degradation bacteria. Then we go for evaluation of degradative efficiency on different agro residues and we go for optimization by pH and temperature enzyme activities, and by that we confirming the R16S RNA gene sequencing, and that were isolated by cellulotic bacterial strains. In the methodology, we first collect the soil sample with the 5 different fields at Athradi by near Udpi, and then we go for a categorization, then isolate the bacteria by using a serial dilution method and plating technique. On that, we got several sample colonies are formed. By that, we select bacterial strain from high cellulose production and that subculturing it and estimate the proteins by Lawrence method. This estimation was confirm the proteins presence in the strains, and after the confirmation, we inoculate the strains and estimate by DNS method for a concentration production of the strains and optimization of cellulose enzyme activities. These are the congruent cellulose agar media shows the zone of inhibitions of SA3 strains and SE4 strains. Here the clear solutions, clear halo zones or white patches zones shows a maximum activity or maximum production of the agar media, and by this we take 3 samples for subculturing S31, S32 and F41. In that we give a subculturing with using a cellulose broth. In that first Day 1, they shows a minimal activities. As they flies up to 13 days, they shows a maximum activities at absorbance 620. That cellulose broth depicting the growth of the bacteria, and that once we got a subculturing where we form a pure culture that we using an NA media. In that we got a white colonies formed like the image shows. In that we took the 3 strains. In that we for confirming these strains by gram staining. In that we got gram negative bacteria or gram negative rods. By focusing these rods, we can confirms the strains. Then we go for a molecular identification based on the phylogenetic tree. We confirm there are true 2 strains. There is a Stenotrophomonas pavani and Stenotrophomonas betali. Because of this phylogenetic tree, we got a exact or specific sequences by nearer the value of 16S rRNA, and it confirms the 16S rRNA with this Stenomonas pavani and Stenomonas betalis species. And this is the again confirmation of the S32 strains. And then we go, once we get a 63S RNA, then we confirm by the estimation of Lorry's. In that coloration measures, there are 3 different strains. They confirms the production of proteins. As the confirmation, we go for estimation by DNS method. In that, we are took a concentration of concentration by daily basis. In that, we take a substrate for natural agro-based substrate like areca husk, wood chips, sugar canes, and other tools. And in that, we go for cellular broth, or we go for incubation in a broth manner. In that, we go first to second days, they go shows a minimal release of a reducing sugar. As the days increases, the isolation or release of reducing sugar also increases. In that, the sugar cane residues and also the coconut core shows a maximum yield, and also shows a maximum number of absorbance compared to the other files. Then after the confirmation of reducing sugar, we go for optimization method. In that, we confirm by the pH and temperature. In the pH optimization, they shows up around 7 to 8 pH strains, S31 and S32 strains. In that, they shows as the color indication the varieties of samples or a substrate shows a maximum yield at of pH 7 or pH 8 nearest. And after the pH optimization, we go for a temperature. They are preferably good at 37 degrees Celsius growth. And at the conclusion, I would like to say that Stenotrophomonas pavani and Stenobetali show the strongest cellular activity, and they gave efficient degradation of multiple agro residues, and they also called as a multi-substrate specific, and they gave a highest glucose solution, as I mentioned above, sugarcane and the coconut coir. They shows a 3 mg and 2.856 mg per ml respectively, and they shows the optimum growth up to 8 pH and 37 degrees Celsius, and they are a potential use of a bionutrients and agro waste management. They helps to reduce the stub burning, improve soil fertility, and sustainable agricultural supports. These are my reference, and thank you.
Thank you, Narmada. Namira, please, can you share your screen and start presenting?
Yes, ma'am.
Speed ಆಯ್ತಾ? ಅರ್ಥ ಆಯ್ತಾ?
Mention your code.
My code is RSOP14.
Narmada, please mute yourself.
Is my screen visible?
Narmada?
ಸರಿ ಇರ್ತೀಯ.
Hello? Hello?
Yes, you can start.
My screen is visible?
Yes, yes.
Yes, it is visible.
Okay. Good evening, everyone. Sorry for the inconvenience. I'm Namira Khan, research scholar in the Department of Microbiology and Botany under the guidance of Dr. Muhammad Aman from Jain University. So before I begin, I would like to share. Hello?
Mention your code, please.
Yes, ma'am. My code is RSOP14.
Yes, please start from your objective.
Yes. So as I mentioned earlier, that some insights related to my topic, prevention is better than cure, but the reality is numerous healthy individuals are unwitting carriers of multi-drug resistant strains without even knowing it. So based on that, my objective is first to isolate and characterize Candida species from saliva samples of asymptomatic individuals. Second, to study phenotypic and genotypic diversity of virulence factors from Candida species. Third, to evaluate the antivirulence factors derived from Senegalia catechu, which is khair or kach tree, and Ocimum gratissimum, which is clove basil or ram tulsi. And last is to elucidate the structure of antivirulence factors obtained from Senegalia catechu and Ocimum gratissimum. Methodology. This study was carried out in 4 major phases. First, under isolation and identification, a total of 120 saliva samples were collected and stored at 4 degrees Celsius before processing. The samples were enriched in sebrodextrose broth and subsequently cultured on sebrodextrose agar for isolation. Preliminary identification was performed based on colony morphology and microscopic examination. Phenotypic characterization included germ tube assay and capsule staining. In second phase, virulence and resistance profiling was performed. Antifungal susceptibility testing was conducted using BIS diffusion method to identify multi-drug resistant isolates. Biofilm formation was assessed using microtiter plate method. Enzymatic assays, virulence factors were evaluated through phospholipase activity on egg yolk agar, protease activity on BSA agar, esterase activity using twin AT agar, and hemolytic activity on blood agar. Based on virulence intensity and resistant pattern, high virulence isolates were selected for further analysis. Third phase involved molecular characterization. Genomic DNA was extracted from the selected isolates, followed by amplification of the ITS region using ITS1 and ITS4 primers. Sequencing was performed for species level confirmation, and in the fourth phase, plant extract of medicinal plants Senegalia catechu and Ocimum gratissimum were prepared using different solvents including hexane, chloroform, acetone, and methanol. These extracts were evaluated for antifungal and antivirulence activity against selected multi-drug resistant Candida isolates. And last, statistical analysis was performed to determine the significance of plant potency. So based on this, This structured methodology allowed systematic screening, confirmation, and evaluation of plant-based antifungal potential. This slide represents the preliminary phenotypic identification of the obtained candida isolates. In figure 1, germ tube assay results are shown. The germ tube test was performed for all 50 obtained candida isolates. After incubation in serum, microscopic examination revealed the presence of germ tube formation as indicated by this arrow. Tube-like outgrowth from each cell. All isolates demonstrated positive germ tube. Figure 2 shows capsule staining. This capsule staining was carried out for all 50 candida isolates to observe the presence of capsular structure surrounding the yeast cell. Under microscopic examination, clear halo zones were observed around the cell against the stained background, indicating positive capsule staining in all isolates. Next, this slide presents antifungal susceptibility profile and colony morphology of obtained candida isolate. AST was performed for all 50 isolate using diffusion method. The isolates were tested against commonly used antifungal agents. Zone of inhibition were measured and interpreted to determine susceptibility or resistant pattern. Several isolate exhibited resistance to multiple antifungal discs, which is amphotericin B, clotrimazole, ketoconazole, and itraconazole. And figure 4 shows morphological characteristic of the isolate grown on candida isolation agar under appropriate observation. Fluorescent colonies representing other candidas, this candida albicans, and along with non-fluorescent colonies representing other candida species. Biofilm formation was quantitatively evaluated for all 50 candida isolate using microtiter plate assays. The result demonstrated that the majority of isolate exhibited strong biofilm production. This slide outlines the methodology used to assess phenotypic virulence factor in candida isolates. 4 key extracellular virulence enzymes were evaluated, which is phospholipase, protease, hemolytic, and esterase. 24 hours old culture broth were used for all assays to ensure active growth and consistent enzyme expression. Phospholipase activity was assessed on egg yolk agar indicated by opaque precipitation zone, as you can see in this image, representative image. Protease activity was evaluated on BSA agar identified by clear zones around the colonies. Hemolytic activity was examined on blood agar observed as zone of erythrocyte lysis, and esterase activity was determined using twin 80 opacity agar detected by white precipitate formation. So together these assays provided a comprehensive evaluation of virulence potential and enabled the selection of high virulence isolates for further anti-virulence studies. This results and discussion. Phospholipase activity was screened in all 50 candida isolate. Figure A shows bar graph representing phospholipase activity measured as zone of clearance on egg yolk agar. Each bar corresponds to a different candida isolate, and the height of the bar indicates the level of phospholipase production. The results show clear variability among isolate with zone diameter ranging from approximately 30 to 28 mm, as I have mentioned here. PONK 39 exhibited the strongest, highest phospholipase activity, followed by BANK 43 and MDNK 33, suggesting a higher virulence potential. Several isolates showed moderate activity, while a few demonstrated lower phospholipase production. Figure B represents a representative egg yolk agar plate used for phospholipase assay. The opaque precipitation zone surrounding the colony, as indicated by this arrow, results from phospholipid hydrolysis by extracellular phospholipase. The size of this opaque zone correlates directly with the enzyme activity, supporting the quantitative data shown in figure A. So together, both figures shows that phospholipase production varies significantly among candida isolates. This is protease activity of candida isolates. Figure A illustrates comparative protease activity of isolates. Yes, ma'am, I'll conclude. Yes, as you can see, this is for protease activity graph. Same goes for esterase and hemolytic activity. For every graph, representative plate was shown. These are the graph of based on all 4 assays. And based on this virulence profiling, these isolates were selected out of 50, 12 isolated were selected, which is showing highest activity. This is the molecular characterization of those selected isolate showing highest band quality After this plant extract, these two plants extract was selected based on hot extraction. 4 solvent used: hexane, chloroform, acetone, and methanol. And antifungal activity was done for the highest isolates. So, conclusion: saliva samples were collected from asymptomatic healthy individuals, and it is enriched in sebrodextrose broth and isolated on sebrodextrose agar, yielding 50 candida isolates. All isolates were positive for jump formation and capsule staining with colony morphology differentiating candida albicans from other species. This diffusion assays revealed several isolated exhibited multi-drug resistance against commonly used antifungal agent. Virulence, enzymatic assays, phospholipase, protease, esterase, and hemolytic activity were assessed using specific media showing strain-dependent variability among isolate. And based on this combined virulence profile, 13 highly virulent candida isolates were selected for further molecular and antivirulence studies. Hexane, chloroform, acetone, and methanol extract of both plants were prepared and showing notable antifungal activity against selected candida isolates. So expected outcome is it is anticipated that this work will identify potential therapeutic strategies to reduce candida infection, especially in females, addressing the growing issue of multi-drug resistant infection and improving oral health outcome as well. Additionally, by focusing on this, research is crucial for managing conditions such as oral thrush effectively. And in molecular characterization, 2 species, one rare species I found. So that's all. Thank you very much. These are the references of my work. Thank you very much.
Thank you, Narmada. Sorry, thank you,
Thank you.
Namira.
Thank you very much.
The judgment will be given tomorrow. I mean, the results will be out tomorrow. Please attend the validity program online.
Thank you, ma'am.
Thank you all.
Okay, ma'am.
Thank you.
Father is not at home. My mother is not well. Yeah.
Like a law of recommendation. Huh?
sir.
ನಾವು 4 ಜನ ಇದ್ದೀವಿ ಯಾರು ಬರ್ತೀನಿ ಕೊಡ್ತೀನಿ
ಇರ್ಬೇಕಂತ ಫೀಲ್